Hi everybody, good morning. Good morning. Am I perfectly audible? Visible? Hi, good morning. Am I perfectly audible? Are visible, audible, visible? Yes, I am. So, good morning students. Good morning, good morning, good morning. Welcome to the session, guys. Welcome to the session. Good morning, people. Good morning. Hi everybody, welcome to the session. Welcome to Unacademy NEET English. I'm your biology educator Ambika Sharma, and in today's lecture we are going to complete class 12 zoology, right? This is the syllabus that we are going to target today. I hope you guys are doing very well, and I hope you all are ready for the session. Yes, I hope you all are ready for the session. Good morning, good morning, good morning. So quickly hit the like button. This is the first thing that you have to do right now. Quickly hit the like button, and if you are new to this channel, please subscribe to our channel. Please subscribe to our channel. Kindly subscribe to our channel. Yes. Good morning, and good morning everybody. Good morning. Komal Bache, you can watch the lectures on Unacademy NEET English. I have already uploaded it. Rather than focusing on that topic, let's focus here. Let's focus here. So everybody, welcome to the class. Hi Ashraf, hi Hyper, Shrek, Mani, Monica, Tanmay, or yes. Good morning, Jaguar. Oh, bhai sahab. Arpana, Gayatri, Uday, Mariam. So, how are you all doing? First of all, tell me, how are you all doing? Are you guys excited for your exam? Are you guys excited for your exam? Like, what kind of emotions you people have right now? Excited for the exam, nervous, or still struggling with the syllabus? Like, what is there in your mind? What is there in your mind? Good morning. Excited? Okay, so let me tell you one thing very clearly, my dear people. See, today it's the class 12 zoology marathon. So in today's lecture, we will be targeting human reproduction, reproductive health, human health and disease, molecular basis of inheritance, and both the chapters of the biotechnology. So principles of inheritance and variation, your evolution, sexual reproduction in flowering plant, microbes in human welfare, and all the 3 chapters of ecology will complete it under class 12th botany marathon. So today this is what we are going to complete. Alright, this is the first thing that you need to know. Secondly, students, yes, your exam will be there in the month of May, and definitely you can consider the expected exam date to be 4th of May. Right now a lot of things are going on regarding the phase 1, phase 2, online, offline examination. Whatever it is, one thing is clear here, and one thing is that is the most important thing is key, you have to complete your syllabus first, right? You have to complete your syllabus first. If you have completed your syllabus, then there is nothing to worry about now. It doesn't matter what kind of paper is going to come then, right? It doesn't matter, right, the difficulty level of the paper, right, whether there will be, section B or not. It is not going to matter if you people will complete your syllabus properly and if you revise sincerely. That's it. That's all. Okay, so that is why this bounce back series is, that is why we are again exhausting ourselves, and because we really want you people to have we really want to help you in completing your syllabus. So if I talk about today's marathon, let me make it very clear for everybody here. If you guys are preparing for the CBSE examination, right, board examination, again this session is for you. And if you think that I'm going to mark each and every line from the NCERT, I'm going to discuss each and every line from the NCERT, right, bache, I'll make it clear that from all the chapters I will take each and every line and I will discuss everything in detail. No, it's not possible. It's not possible, because it will take around 20 hours, 30 hours, right? If I have to justify all the chapters now, all the chapters, including each and every line of NCERT with proper NCERT marking, then obviously I need 20 to 30 hours. So here in this particular session, what is my plan? I will be targeting the most important questions, and wherever there is the conceptual thing, I will discuss everything in detail there. Is that clear? Is that clear? Yes. So ready for it? If it is a yes, I want to see some fire emojis here in the chat section. Quick. Yes. Yes. Very good, very good, very good. The Josh should be high. Okay, so we are going to target the most important units today, right? So be ready for it. And guys, we are already taking our fast track reloaded batch, right? And trust me, the things are going amazingly here. So if you want a proper batch with proper test practice, with proper syllabus completion, right, this batch is for you. In the description box of this video, you will get the link for the batch, and here you can see you have to pay 3499 and you will be the part of this batch. So we will complete your syllabus till 31st March along with the question practice, along with PYQ practice, plus every week there is a mock test, and after the completion of syllabus in the month of April, you will get 30 full syllabus mock test as well. So without delay, be the part of this batch and let's continue now. So what will be the session flow today? Let me tell you this thing. What is the session flow? So we will go for molecular basis of inheritance first, then both the chapters of biotech, then human reproduction, reproductive health, human health and disease. That's the flow of the chapter. Okay, because I believe from this chapter minimum 6 to 7 questions can come, right? Minimum 6 to 7 questions. So even if I consider 6, 24 marks. So I want you to pay full attention and let's cover it. Okay, let's cover it, and then we will go for biotechnology, right bache? Then we will go for biotechnology. Okay, so we have already completed biomolecules here on this channel, and these are the chapters that I'm going to target. So be ready for it bache, and see this. You have flat 60% off on all NEET UG subscriptions with assured success, right? So if you are targeting 2026, 2027, see, for 2025 you people have our, what do you have, right? For 2025, you know that you have our NEET Avengers fast track batch, but for 2026, 2027, this is what you guys have, right? So it is the Resolve 2025 and it is valid till January 8. So you guys can use coupon code Ambika10 and you can join our batches for 2026 as well as for 2027, right bache? Chali, let's go, let's discuss the topic. Okay, so evolution, human health and disease. So here we go. Molecular basis of inheritance. Yes, molecular basis of inheritance. So ready for it? Yes. Are you ready for it? MBOI, molecular basis of inheritance. Are you ready for it? Done? Okay. So if I talk about this particular chapter, what is important? From this chapter, do not leave Human Genome Project ever. Do not leave Human Genome Project ever. One question you will definitely get from Human Genome Project. One question you will definitely get from genetic code, right bache? Then when it comes to transcription, translation and your DNA replication, right? So I'll suggest you to focus on the enzymes. Focus on the templates part. Do not miss it, because here you people just get confused. Okay, so these are the topics that you should not miss, and yes, one is open. These are the topics that you should not miss. Not at all. Not at all. Other than that, experiments, packaging of DNA double helix is given. But these are the topics that you people should never ever miss. Okay, you should never ever miss. So let's start it. So as per your syllabus, that's what you have. You can see this as per your syllabus, that's what you have, right? Search for the genetic material, DNA as genetic material, structure of DNA and RNA, packaging of DNA, replication, central dogma, transcription, genetic code, translation, gene expression and regulation, genome, and HGP. Okay, HGP. So DNA fingerprinting and protein biosynthesis is there, right bache? So when it comes to the protein biosynthesis, because last time also it was mentioned there in your syllabus, protein biosynthesis, it is nothing. Basically you are just going to talk about the transcription and translation part, right bache? Right bache? Because when it comes to the protein biosynthesis, you know that it is the DNA. DNA is having genes. How will they express? They will form mRNA and then there will be the formation of protein. This is the flow of information, right? This is the flow of information, right bache? So that's what we study under this topic. Okay, So before going forward, what we need to mark, we need to know about the nucleic acids, right? What are we going to cover? What are we going to cover first? We'll be talking about the nucleic acids, right, bache? Right, bache? So when you people talk about the nucleic acid, see, this part is important. This part is literally very, very, very important. Okay, this part is literally very, very, very important. Now, if you think that ma'am is going to highlight each and every single line of NCERT, not possible for me. Okay, not possible for me to highlight each and every line of NCERT right now, right? But obviously, I have to decode the NCERT, and that's what I'm going to do. Okay. Okay, so I have to decode the NCERT, and that's what I'm going to do. So are you ready for it? Right, I'll make sure that you understand everything. Yes. Yes. Okay, chaliye, let's go guys. So we are talking about here in this chapter, obviously we need to know about the nucleic acid, right? We need to know about the nucleic acid. We should know about the nucleic acid first. So when you talk about the nucleic acid, keep it in your mind, it's a bio-macromolecule. What's that? Oh. Yes, what's that, bache? It's a bio-macromolecule. What is it? It is a bio-macromolecule. So when I'm saying it is a bio-macromolecule, it means its molecular weight is, yes, bache, it means that its molecular weight is above 10,000 Dalton. Its molecular weight is above 10,000 Dalton. Its molecular weight is above 10,000 Dalton. So nucleic acid, you will see it in the acid insoluble pool. Where will you see this? You will see it in the acid insoluble pool, means it will be there in retentate. It will be there in retentate. Are you getting my point? Bio-macromolecule, right? These are the nucleic acid. So your DNA, deoxyribonucleic acid, and we have ribonucleic acid. So when it comes to the discovery of your, yes bache, nucleic acid, so we have the name Friedrich Mischer, right? Friedrich Mischer, you know, he was the one who discovered the nucleic acid. Yes, he was the one who first discovered nucleic acid. He was the one who discovered nucleic acid in pus cell, right? In pus cell, and he called it as nuclein. Why? Why? Because it was there in the nucleus, right? He didn't give the name nucleic acid. He called it as nuclein. Friedrich Mischer, right? Friedrich Mischer, and later, bache, Altman, right? Altman renamed it as nucleic acid due to its acidic property. Okay, due to its acidic property, he called it as what? He called it as nucleic acid. This is the first thing. And yes, you are right that Friedrich Mischer in 1869 discovered the nucleic acid in the pus cell, in the nucleus of the pus cell, and he called it as nuclein. And Altman renamed it as nucleic acid. Any doubt here? Any doubt here? So when you talk about the nucleic acid, what you need to understand, as I said, it is a polymer, right? As I said, what is it? It is a polymer. Yes, people. What is it? And I want the session to be interactive, so do answer my question in the chat section. Okay. Do answer my question in the chat section. I really want to feel motivated. Okay, and if you are not interested in class, please don't waste your time here. Go have fun, right? So many other social media platforms are there, right? And other than that, you know that so many movies are available, OTT platforms are there. Go and have fun. So if you're not interested in learning this, then bye-bye. If you're interested, then stay. So what will you say? What will you do? You will stay or you will say bye-bye? What will you do? You will stay or bye-bye? Yes, yes, yes. Done? Voice issues? I think voice is fine. Done? Bye bye, no. Stay, yes, better. Better. Chaliye. So what are we talking? We are talking about the nucleic acid. What are we talking? We are talking about the nucleic acid. So nucleic acid is a polymer. It's a polymer of nucleotide. It's a polymer of nucleotide. So many nucleotides together, right? So many nucleotides together, they form the nucleic acid. So if you want to understand what nucleotides are, so basically what nucleotides are, nitrogenous base, sugar plus phosphate. It is nitrogenous base, sugar plus phosphate. So keep it in your mind, and it was a question in chemistry. Not here in biology, but it was the question in chemistry, and that too in NEET 2023, that which acid is actually involved in the formation of this nucleic acid. So bache, it is your phosphoric acid. Keep it in your mind. It is not phosphorus, it is your phosphoric acid. Which acid is there? Phosphoric acid. Ionic form is phosphate. Ionic form is phosphate. So what is the name here? Nitrogenous base plus sugar plus phosphate. So if you want to understand what a nucleic acid is, what a DNA is, you should know about it. So let's start with the nitrogenous bases. So if I'm using the word nitrogenous bases, one thing is clear, that they are the nitrogen containing compounds. Am I right? Am I right? That what are they? They are the nitrogen containing compounds. Yes or no? Yes, people. What are they? They are the nitrogen containing compounds. And when we talk about the nitrogenous bases, we have purines and we have pyrimidines. What do we have? We have purines and we have pyrimidines. Purines and pyrimidines, that is what we have. So when you use this word purines, the trick here is puga. It's simple and one question will come from this part. It's simple and one question will come from this part. So what is the point here? That when you people talk about the purines, the word here is puga. The word here is puga. The trick here is puga. There is a brand of Puma. So that Puma will become puga, whatever you want to say. So puga means G stands for guanine here. A stands for adenine here, right? So G stands for guanine, A stands for adenine, 9 is the word here, 9. Look at the spellings here, guanine, adenine, guanine, adenine, guanine, adenine, 9. So remember, when you are talking about the guanine purines, they are 9. It is actually a 9-membered ring. What's that? It is a 9-membered ring. What's that? It is a 9-membered ring. Are you getting my point? Are you getting my point? So definitely your guanine is denoted by G, adenine is denoted by A. So it's a 9-membered ring. Understood? It's a 9-membered ring. So if I have to draw it, so it will be something like this, right? It will be something like this. Is that clear? Something like this, and that too alternate double and single bond. Have a look. Okay, chaliye, I'll draw it. But first, then when you talk about the pyrimidines, these are for the purines. Now when we talk about the pyrimidines, this is a 6-membered ring. And my dear students, it is basically, the trick here is cut. C-U-T. What is the trick here? The trick here is cut. C-U-T. Is that clear? Is that clear? It is C-U-T. So one pyrimidine ring is there having 2 nitrogen and 4 carbon. Right, bache? It's a 6-membered ring. The trick is cut. C stands for cytosine, U stands for uracil, and T stands for thymine. Is that clear? Is that clear? So if you don't know what purines are, pyrimidines are, kindly pay attention. Okay, kindly pay attention. So it's a 9-membered ring, it's a 6-membered ring, right? I'll show you the diagram as well. So kindly keep it in your mind. Okay, so remember one thing. Remember one more thing I would say. So purine is the word, pyrimidine is the word. Am I right? Purine is the word, pyrimidine is the word. Right, bache? Right, bache? Purine and pyrimidine. So when you use the purines, so here at position 1, 3, 7 and 9, nitrogen is present. In the ring at 1, 3, 7, 9, nitrogen is present. Pyrimidine mein at 1 and 3, nitrogen is present. Is that clear? At 1 and 3, nitrogen is present. Right, bache? At 1 and 3, nitrogen is present. Is that clear? Is that clear? Sure. Sure. This information is important, of course. This information is important. Now, just pay attention here. Right, please pay attention here. Look at the purines. So, it's nitrogen 1. At first position, nitrogen. Then in the ring, carbon is there. Right, alternate double bond, single bond you people are going to see here. Right. See this. See this. Look at this. Right, so what are they? They are purines. What are they? They are purines. So at 1, 3, 7, 9, nitrogen is there. Look at this. Double bond, single bond, double bond, single bond, double bond, single bond. Is that clear? So when you look at the purines, if you look at this part only, please have a look at this part only. So what can we see? This is just like pyrimidine. Hai na? Pyrimidine. At first and third position, there is a nitrogen. So when you say purines, so they have one pyrimidine ring, one pyrimidine ring, and one imidazole ring. One pyrimidine ring and one imidazole ring. One pyrimidine ring, one imidazole ring. What do we have here? One pyrimidine ring and one imidazole ring. Right, bache? Right, bache? Right? मिस्टर सोको मैम एवेंजर्स रीलोडेड बैच स्टार्ट क्यों नहीं हो रहा बच्चे? क्योंकि आज मैं क्लास ले रही हूँ, इसलिए मैं कल से स्टार्ट करने वाली हूँ. डेफिनेटली, है ना? Definitely. And when it comes to is it a scam or something, we are telling you the batch will start. Right, batch will start. Today I have a marathon, and obviously from tomorrow and day after tomorrow I will take the class. Okay. So we are not denying that batch is not going to start. So rather than just spamming here, please pay attention. Pay attention. It will be useful for you. Okay. Okay. So this part clear. One pyrimidine ring, one imidazole ring will be there. Imidazole ring having 2 nitrogen and 3 carbon. Right. Imidazole ring basically having 2 nitrogen and 3 carbon. Adenine and guanine is the thing. So look at the adenine structure. At 6th carbon, NH2 group is attached. Okay, so these structures are given in your class 11th biomolecule chapter. Okay, so yes, you have to remember this. You have to remember this. And now, next is guanine, right? Guanine. It's guanine. O. It's guanine. O. Guanine. So at 6th carbon, here in this ring, you will see double bond O, and here NH2 is there. Right, but here at 2nd carbon also NH2 is there. So structure should be clear. Structure should be clear. So at position 1, 3, 7 and 9, nitrogen is there. 1, 3, 7 and 9, nitrogen is there. Look at the purine part, right? Look at the purine part. This is your first nitrogen. First nitrogen, then double bond. See, double bond. So here also alternate double bond and single bond is there. Look at this. This is your pyrimidine 6-membered ring. What's that? 6-membered ring. At 1, 3, what is there? N is present. At position 1, 3 in the ring, N is present. So if you have a doubt regarding this, ki ma'am, you are saying 4 nitrogen will be there, but here we have 6. So what am I saying? I'm saying the nitrogens. I'm talking about the ring here. I'm talking about the ring here. What am I talking? I'm talking about the ring here. Okay. Now you have cytosine denoted by C, uracil denoted by U, thymine denoted by T. Okay. So look at the structure of cytosine. Right. Again, you have to remember it. Right. Double bond O is present at 4th carbon. NH2 is present. You have to remember it. Look at the structure of uracil and thymine. Look at the structure of uracil and thymine. See, bache, it is all same. Uracil and thymine, it is all same, except here in the thymine, at 5th carbon, methyl group is attached. Methyl group is attached. So thymine is also known as 5-methyl uracil. It is also known as 5-methyl uracil. It is also known as, please write it here in the chat section, It is also known as 5-methyl uracil. Yes. It is 5-methyl uracil. Okay, what's that? It is 5-methyl uracil. Okay. What's that? It is 5-methyl uracil. Is that clear? Sure? Are you sure? So the structure should be clear to you. Now, next part is sugar. So look at it more clearly. Thymine at 5th carbon, uracil is there. 5-methyl uracil. So let me tell you, students, when you talk about the RNA, in the case of RNA is single-stranded. RNA, purines, present in RNA is guanine and adenine. And when you talk about the pyrimidines that are present in RNA, that will be your, yes, C and U. Uracil, R is there. Uracil is there in your RNA. It is the uracil which is there in your RNA, cytosine and uracil. And when you people talk about the DNA, when you talk about the purines, definitely adenine, guanine, and pyrimidines, cytosine and thymine. So cytosine is a common pyrimidine in both. Again a PYQ. It's a previous year question. If you will solve the questions, you will get this question. Right, bache? And the other thing is in RNA, you have cytosine and uracil. So uracil is only there in RNA. You people will not see it in DNA. Understood? So structure is important. This information is important. If it is done and dusted, I want to see some fire emojis in the chat section. Yes, madam, done. Yes, madam, done. Okay. Okay. Very good, very good, very good. Now the next thing is sugar. Next thing is what? Next thing is sugar. So sugar means carbs. I'm talking about the carbohydrate here, and I am talking about a pentose sugar here. What am I talking? I am talking about a pentose sugar here. Pentose, aldose. What is the meaning of pentose, aldose? Pentose, pent mane 5 carbon. Number of carbon will be 5. If I'm saying aldose, means the functional group here in this carbohydrate is aldehyde. Because in the carbohydrates, when we define the carbohydrates, we say that they are polyhydroxyaldehyde or polyhydroxyketones, or the compounds which on hydrolysis yield polyhydroxyaldehyde or ketone. Right? Yield polyhydroxyaldehyde or ketone. Is that clear? Is that clear? Sure? Sure. So it's a pentose 5 carbon containing sugar. So obviously it is a monosaccharide, and if you need to talk about this monosaccharide, it's very simple, bache. It's very simple that these monosaccharides, yes, they will be there in the acid soluble fraction, right? They'll be present in the acid soluble fraction because it is a biomicromolecule, not the biomacromolecule. Okay, so here the ring is furanose, this. It's like this. Okay, it's like this. This is your carbon number 1, carbon number 2, carbon number 3, carbon number 4. So here you will be having OH, H, H, OH, H, OH, H, CH2OH, H. That's what you have. So it is your ribose. It is your ribose. So bache, it's a pentose. It's a monosaccharide. There is a general formula. CnH2nOn or CnH2ON. That's a general formula for monosaccharides. It's a general formula for monosaccharides. If you know it, very good. If you don't know it, right? If you know it, very good. If you don't know it, then it's my suggestion that please watch the Game of NEET 2.0 or Game of NEET Simple for that biomolecule chapter. This is important. Okay, this is important. CnH2nOn, CnH2nO. So ribose is a pentose because number of carbon, they are 5. 1, 2, 3, 4, and this is the 5th one. This is the fifth one. Okay, so N is equals to 5 here. So if I have to put it in formula, so it will be C5H25O5. So C5, 5 to the 10, O5. That's the formula. Again, PYQ. Previous year question it is. It is again a PYQ. So today I will address all the PYQs from that particular topic. Okay, now, when you talk about the deoxyribose, when you talk about what? When you people talk about the deoxyribose, OH, H, 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 OH, this is the deoxyribose. This is what? It is deoxyribose. So it's all same. The difference here is at second carbon. At second carbon here in the ring, O is, oxygen is absent, right? See, that's the only difference. So you have to be specific here. So when you talk about the RNA, RNA is having what? RNA is having ribose sugar. When you talk about the DNA, it is having deoxyribose sugar, right? So at second carbon, oxygen is absent. That's your deoxyribose. Okay, bache. At second carbon, your oxygen is absent. That's your deoxyribose. Okay, bache. That is your deoxyribose. Okay. Okay. So it's a pentose. So one oxygen is less here, right? One oxygen is less here. So what will be the formula? C5H10 but O4. So this is an exception. This is an exception to general formula. This is an exception to general formula. Deoxyribose, they do not follow the general formula for monosaccharides. Okay, they don't follow the general formula for monosaccharides. This information is important. Again, PYQ. Again, it's a PYQ. Understood? What's that? It is a PYQ. Understood? Yes, understood. So next is the phosphoric acid, right? And phosphate, you can say, right? So what is it? P double bond O. This is something where you need to help me. Is it correct or incorrect? I don't know about it. I don't know about it. You have to tell me. Is it correct or not? Is it phosphoric acid or not? Is it phosphoric acid or not? Please tell me. Is it phosphoric acid or not? Of course it is. It is H3PO4. Am I right? H3PO4, phosphoric acid, right? Because this acid is present in the DNA, that's why it is negatively charged. It is having the acidic properties, right? Phosphoric acid it is. So phosphate is that ionic form. These are the three things that we have. Now what you need to understand, students, you need to understand, right? What you need to understand, you need to understand the next thing. Now you individually know what nitrogenous bases are, what sugars are, what phosphate is. Now we will understand something known as nucleoside. What we need to understand? We need to understand something known as nucleoside. So when I'm using the word nucleoside, N here stands for nitrogenous base, S stands for nucleoside is the word. Do solve the PYQs and you will get the question from it. You solve the PYQs and you will get the question from it. Nucleoside and S stands for sugar. So when your nitrogenous base, when it joins with sugar, when your nitrogenous base, when it joins with sugar, you will get nucleoside. You will get nucleoside. When your nitrogenous base, it joins with sugar, you will get nucleoside. Let me give you one example. Okay, and nitrogenous base and sugar. Okay, let me give you one example. Ready for the example? Yes. Are you ready for the example? Okay, chaliye. It's something like this. Yes, is it something like this? Is it something like this? The ring, I'm talking about the purine here. I'm talking about the purine here, like here double bond, single bond, then double bond, single bond, double bond, single bond. It is like this, yes or no? Is it like this, yes or no? Yes or no? And because it is the 1, 2, 3, 4, 5 and 6. So this is the 6th carbon. So here NH2 is attached. Right. So if NH2 is attached here, is it adenine or guanine? Is it adenine or guanine? Please tell me. Is it adenine or guanine? Please tell me. Is it adenine or guanine? This is the 6th carbon, 7th or 9th. It is the adenine. And now let's take the example of the sugar. What we have to discuss now? Let's take the example of the sugar. 1 second. Okay. Chaliye, so let me draw this again. Okay, so I'll draw the sugar here. I'll draw the sugar here. And one more thing. See, this sugar, this is the first carbon, C1. So, at C1, hydrogen is below. That's the trick, na? Hydrogen is below. So, it is the beta ribose. It's not the alpha ribose. It is the beta ribose. Beta. If this H will be here and OH will be down, then it will be alpha ribose, because at say C1 we have to see the position of OH and H. On the basis of that we say whether it is, right, whether it is the beta ribose or it is the alpha ribose. Is that clear? Whether it is the beta ribose or it is the alpha ribose. This is what we need to keep in our mind. Is that clear? Is that clear? Now I am going to show you the bonding, right? I'll give you only one example, then rest you have to take care of it. This is your first carbon, OHH, HOH, HOH, H, and this is your CH2OH. So, is it ribose or deoxyribose? Is it ribose or deoxyribose? Please tell me. Is it ribose or deoxyribose? Ribose, right? Now, even. Just to make it clear, I am drawing it in this way. Okay? Take. So, what's the mistake here? What's the mistake here? Please tell me, what is the mistake here? Take. What is the mistake here? 1 or 6 के बीच में भी. This is the 7th or this is the 9th one. Take. Right. This is the 7th. So basically it is the adenine. Yes. Basically it is what? It is the adenine. 7, 8 or 9 maybe adenine. Now the first carbon of this sugar and this 9th nitrogen here, they will form a bond. Right. What will they do? They will form a bond. So here there will be minus H2O. This 9th nitrogen of purine and first carbon of your sugar, they will form a bond here. So minus H2O will be there, minus H2O will be there, and they will join. They will join. So it is your N-glycosidic bond. What's that? It is your N-glycosidic bond. What's that? N-glycosidic bond. So when you talk about the nucleoside, nitrogenous base and sugar, they are connected to each other. How? With the help of this N-glycosidic bond. How are they attached to each other? Yes, bache. Maitri, Sai, Sujay, how are they attached to each other? How are they attached to each other? How are they attached? Yes? How are they attached? N-glycosidic bond. Exactly. It is your N-glycosidic bond, right? So that is going to connect your nitrogenous base plus sugar. Now this is adenine, this is ribose. So it will be adenosine. What's that? It is adenosine. Adenosine. What's that? It is adenosine. So you have to remember the nomenclature as well. Now to it, I'll draw it again. Dekho, sugar is sweet. So everybody is like, let's connect with sugar. That's how you can keep it in your mind. Sugar is sweet. So everybody is interested in forming bonds with sugar, right? This is the sugar again. Right, bache? So again, here you have NH. Again, I'm taking the example of, yes, I'm taking the example of adenine and this is the sugar here. Adenine and this is the sugar here, yes or no? This is the sugar here, yes or no? Please tell me, yes or no? 1, 2, 3. Arre. Haan. This is the adenine. That's how it is present. So you know that we will see the which bond here? N-glycosidic bond here. We will see N-glycosidic bond. Why N is the word here? Right? Because nitrogen. One nitrogen and one carbon. No, it is not in between two carbons. CC. No, nitrogen and carbon. So it is N-glycosidic bond, right? Now, bache, here in this particular structure, please keep it in your mind, right? This is the first carbon of the sugar, second carbon, third carbon, fourth carbon, and this is the fifth carbon. Now, next is what? Phosphoric acid we have. P double bond O, OH, 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 right? So here also you will see there will be minus H2O. There will be minus H2O from this part. So when there will be minus H2O, right? When there will be minus H2O, because it is CH2OH. So it's going to be like this. Like this. Right? It will be like this. So it is the ester bond, but phosphate it is phosphoester bond, right? It is phosphoester bond. This is COC. It is your ester bond, right? When it is phosphoester bond, that's how it is. That's how it is. P double bond O, this O and this C here, right? So which bond is there? Which bond is there? It is phosphoester bond. Which bond is there, students? Phosphoester bond. Which bond is there, students? Phosphoester bond. You can even show it in ionic form. No worries at all. No worries at all. Okay? Right? Right? So you know it better than me. Right? You are still reading chemistry. I don't know. So obviously you know it better than me. So when to a nucleoside phosphate group get attached, then you will call it as nucleotide. Nucleotide. So from this T, you have to remember three things are there. So already we have nitrogenous base, already we have sugar, and then there will be the phosphate. So these three things are making your nucleotide. So which bonds do we have here? We have N-glycosidic bond, again a PYQ, and the phosphoester bond is there in the case of nucleotide, in a single nucleotide, right? So adenine plus sugar, it is 1 minute. Right? So it is nitrogenous base, sugar and phosphate. Three things are present. Nitrogenous base sugar and phosphate. So adenine plus sugar, it is adenosine. Because it is a ribose sugar, so it is adenosine. One phosphate is also attached. So can I call it as adenosine monophosphate? You please tell me about the nomenclature because it is also important. Can I call it as adenosine monophosphate? Please tell me. Can I call it as adenosine monophosphate? What is it? It is the adenosine monophosphate. It is the adenosine monophosphate. It is the adenosine monophosphate, or the another name here is adenylate acid. Adenylate acid. Adenylate acid. Is that clear? Is that clear? That's the nucleotide. So when many nucleotides they join, they will result in the formation of polynucleotide chain, and that's how your nucleic acid will form. So if I have to give you the example here only, so see, yes. So when it is a purine, I'll specify it because it is adenine, it's a purine. So nitrogen at ninth position, and in sugar, C of first carbon of sugar, they form bond, N-glycosidic bond. In the case of purine, it is the ninth nitrogen at ninth position and carbon at first position of sugar, they form the bond. So if it is a pyrimidine, then it will be nitrogen at first position of pyrimidine and carbon at first position in sugar, they will form an glycosidic bond. This information is important, right? This information is important. Clear? This information is important. This information is important. Okay, so focus on the nomenclature here. If in the paper the word used is adenine, it is your nitrogenous base. If it is adenosine, it is your nucleoside. If it is adenosine monophosphate, then it is nucleotide. Is that clear? And adenosine monophosphate is also known as adenylic acid. Adenylic acid, but it is the single phosphate. Yes. So what's that? It is adenylic acid. Understood? Right? So if sugar will be deoxyribose, so deoxyadenosine, sorry, if it will be deoxyribose, so it will be deoxyadenosine and deoxyadenylic acid, या deoxyadenosine monophosphate. So nomenclature is important, right? Nomenclature is important. So look at the bonding part, right? Ninth nitrogen, first carbon, N-glycosidic bond. This is nucleoside. In the case of nucleotide, the fifth carbon here is also forming the bond, right? So that is how it is, and it is basically the triphosphate attached, but you need to focus here, right? That is on the monophosphate. Okay. So N-beta glycosidic bond here, phosphoester bond is here, right? Phosphoester bond is here. So bonding part clear? Bonding part clear? Yes, people, bonding part clear? nucleotide, nucleoside, clear. So now when you will form polynucleotide chain, you will see that here, because that's how you can remember. Sugar is sweet, so everybody wants to get attached with sugar. So when it will be the polynucleotide chain, so you can see here, this is the third carbon, bache. This third carbon will attach with the next nucleotide, right? So this phosphate of next nucleotide, right, and this third sugar here, they will be involved in bond formation. Then you use the word phosphodiester. Take, so ye dekho, this is the bonding part that you need to understand. In the NCERT also, when we explain the structure of DNA, so it is written a 3-5- phospho diester bond is there. Then it is diester. So remember, in a single nucleotide, what will you see? In a single nucleotide, it is the phosphoester bond. In a single nucleotide, it is the phosphoester bond. Is that clear? In the single nucleotide, you have the phosphoester bond. But if it is two nucleotides are attached, it will be phosphodiester. It will be phosphodiester. It will be phosphodiester. Clear, bache? Clear? So look at this. This is the, even in the paper, if you will solve the PYQ, X is what? Nitrogenous base. Right? This is your sugar. This is your phosphate. Right? Now this is the next nucleotide. So look at it. 3-OH. And this is the next nucleotide. Right? So these two are involved in bond formation. So your first nucleotide, the carbon at third position, right? This is your next nucleotide, your second nucleotide. So here in the case of your second nucleotide, at 5' in the sugar, at 5' this is the fifth carbon. So they are forming the bond. Look at this. This is the phosphodiester bond. It is connected here and here. That's how the polynucleotide chain will form. Right, bache? So this is what you need to remember. So whenever we talk about the chain formation, the nucleic acid, the polynucleotide chain formation, we say that synthesis is in 5' to 3' direction. Be it's RNA or it is DNA. We always say that synthesis is in 5' to 3' direction. Right? That's what we say. That synthesis is in 5' to 3' direction. Synthesis is in 5' to 3' direction. Yes or no? The synthesis is in 5' to 3' direction. Yes or no? The synthesis is in 5' to 3' direction. Yes or no? Yes or no? Yes or no? Yes, ma'am. It is in 5' to 3' direction. So what is the meaning of that? This is your first nucleotide. Sugar, nitrogenous base, phosphate. So this phosphate is attached at 5' end of sugar. This phosphate is attached at 5' end of sugar. So that's why, like whenever you talk about your first nucleotide, this phosphate is not involved further in bond formation. Right, but it is just attached to the sugar here. So 5' end will always have phosphate. Now we have our next nucleotide. Sugar, nitrogenous base, phosphate. Look at this. Sugar, nitrogenous base, phosphate. Sugar, nitrogenous base, phosphate. That's how it is. Sugar, nitrogenous base, phosphate. That's how it is, students. So you know that this part, that this sugar, it's OH at 3' end or at third position. It's OH at 3' end and third position. It will form bond with this phosphate. It will form bond with this phosphate. It will form bond with this phosphate. Are you getting my point? Are you getting my point? So that's how they will join. So it is showing us that sugar, phosphate, this phosphodiester double bond, and this part is showing us that sugar phosphate backbone, right? So synthesis is always in 5' to 3' direction. So fifth dash, this phosphate is not involved in bond formation here, right? Even I will mark it in NCERT. If in the NCERT they are talking about this bond, what is it? This is the first nucleotide. This is the first nucleotide. First nucleotide here. This bond here is, yes people, this bond here is, yes bache appu, this bond here is, this bond here is, exactly, this bond here is phosphoester bond. Excellent. But when you people talk about this bond, this is phosphodiester. This is phosphodiester. So be clear about the question. Read the question carefully. So 5' end is having free phosphate group. So this is the last nucleotide. So its OH is not involved in the bond formation. So 3' OH. That's how we mark. 5' end is having the phosphate group, 3' end is having the OH group. 5' end is having the phosphate group, 3' end is having the OH group. Understood? Understood? One minute. See this. So you know that DNA, it is anti-parallel. DNA, it is anti-parallel. Yes or no? DNA is anti-parallel. Two strands. They are parallel to each other but in opposite direction. That's what we have. So basically that's how the structure is. Students, that is how the structure is. 1 minute. That's how the structure is, right? So nitrogenous bases, they face the center. They are connected together with the hydrogen bonding. They are connected together with the hydrogen bonding, right? How are they connected? They are connected together with the hydrogen bonding. Yes or no? Yes or no? Yes or no? Right? So that's how the structure is. Sugar phosphate backbone. Nitrogenous bases are there in the center and they are forming the hydrogen bond. So we know that adenine will form 2 hydrogen bond with thymine, because here the principle of complementarity is there that we will study under the Chargaff's rule, and C will form 3 hydrogen bond with guanine. Again, important information and previous year in a question. So if you are a NEET aspirant, you know about it, isn't it? You know about it. Yes, bache, you know about it. Okay, so that is how the structure is. Baki nomenclature, do not make the mistake here, right? Because that's also PYQ. Adenine is a base. Deoxyadenosine means sugar is deoxyribose. So deoxyadenylic acid, deoxymonophosphate, it's same. So it's guanosine या guanylic acid, cytidine, acid, deoxythymidine. You can also write it as thymidine. Why? Because thymine is only there in the DNA, right? And uridine, because it is there in the RNA. So obviously the sugar here is ribose. So that's how it is present. So nomenclature is important, right, bache? Nomenclature is important, right, bache? So let's talk about the DNA. Now you know about the structure of DNA as well. You know about it in detail. You know that the synthesis will always be in 5' to 3' direction. 5 end will have the phosphate, 3' end will have the OH. So overall in DNA, which is a double strand molecule, you will see that nitrogenous bases are connecting. They are joining that 2 polynucleotide chains, right? And when you talk about the polynucleotide chain, sugar, this N-glycosidic bond and 3', 5' phosphodiester bond is there. Okay, 3', 5' phosphodiester bond is there. Okay, so what is DNA? Long polymer of deoxyribonucleotides, right? So length, this point is very important. I hope you all pay attention here. Please pay attention here. Please pay attention here. The length of DNA is usually defined as number of nucleotides or a pair of nucleotide referred to as base pair. Just I said, adenine, it's a single base. It always joined with thymine. With how? With 2 hydrogen bond. Now I will call it as base pair. So you will see the principle of complementarity here, right, bache? So that's important to understand. Indeed, that's important to understand. So the length of DNA is defined as the number of nucleotides. The length of DNA is defined as the number of nucleotides present. Oh, sorry. Yes. So number of nucleotides present in it. Right, so this is also the characteristic of an organism. So in a bacteriophage known as this, what is this, 5 into 174 has 5386 nucleotides. This information is important. Bacteriophage lambda, it does have 48,502 base pairs. So in the paper they can confuse you. If they write 58402 bases, that's incorrect. They are talking about base pairs. And E. coli is having 4.6 into 10 raised to power 6 base pair. 4.6 into 10 raised to power 6 base pair. Correction here. And haploid content of human is having this 3.3 into 10 raised to power 9 base pair. And bache, in the diploid content, when the human cell is 2n, then it will be 6.6 into 10 raised to power 9 base pair. Is that clear? So it is such a long polymer. It is such a long polymer. So this information is important. So if you solve PYQs, you will get questions from this part. Understood? Chaliye, let's go to the next part. Structure of polynucleotide chain. Bache, all the things are discussed here, right, regarding bonding and everything. So I will provide you the notes in our official Telegram group, that is Unacademy NEET English, right? That is Unacademy NEET English. So I want you people to, right, I want you people to read it from the NCERT because we have already covered. So 5' end is having free phosphate. So this bond is phosphoester bond here. But this bond is phosphodiester. Okay, so look at the arrangement. Look at the arrangement. This is your polynucleotide chain. Understood? This is your polynucleotide chain. So in the NCERT, they have explained the bond forming and formation. Now, let's discuss about the Watson and Crick, right? Watson and Crick model of DNA replication. So Watson and Crick, they explained this model, right? So it is double helical model of DNA. So it is the BDNA that they explain. It's very commonly occurring and it is the right-handed DNA, right? It is the right-handed DNA. Okay, so very commonly occurring is the BDNA. The structure that we discuss is of BDNA, but it is not the only DNA present. A DNA, B, C, D, E, right? All of it is present. So there is one PYQ, there is one Z DNA. So from this Z, you can keep it in your mind, zigzag, right? So it is the left-handed DNA. Rest all are right-handed and this one is the left-handed DNA. So as I said again, it's a previous year question, right? It is again a previous year question. Understood? 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 This is a previous year question. So look at this 5' end phosphate base. They are joined with 3 hydrogen bond, right? So purine always form bond with pyrimidine. Jैसे C is forming triple bond with guanine, right? A is forming double bond with adenine. You need to keep in your mind. This is your DNA. So look at this 5' free phosphate group and look at this 3' 5' phosphodiester bond. Okay, okay. So DNA was an acidic substance. It was present in the nucleus, was first identified by Friedrich Mischer in 1869. So from each and every single line of NCERT, right, in the NEET examination, you'll see the question will be there. You people will see the question will be there. From each and every single line. Okay, from each and every single line, you will see the question in the paper. Chale. So Friedrich Mischer named it nuclein, and as I said, Altman will call it nucleic acid. Who's going to call it nucleic acid? Mr. Altman. Okay, bache. So due to technical limitation, such long polymer, right? It was very difficult to isolate such long polymer. So elucidation of structure of DNA remained elusive for a very long time. So only in 1953, right? So imagine something is there in nucleus. It was discovered in 1869, but the structure of DNA was explained in 1953 by James Watson and Francis Crick, and it is based on the X-ray diffraction. So let me tell you one thing here, bache. Let me tell you one thing here. I'm not going to discuss that story with you all, but actually it is not the mainly the Watson and Crick who explained it. Matlab, it was not just their work, right? So in the NCERT, we talk about Morris Wilkin and Rosalind Franklin. So actually she is the lady behind the discovery or behind the explanation of the structure of DNA, Rosalind Franklin. And she was an expert there in X-ray diffraction, also known as X-ray crystallography. Okay, so in the paper they can ask you this question: which technique is used to explain the structure of DNA? X-ray crystallography or X-ray diffraction it is. So she took the picture of that DNA, and without her knowledge, Morris Wilkin, right, because they were working in same lab, he shared it with Watson and Crick, and by using her picture they explained the pairing, the bonding, everything. And later on, when Rosalind Franklin, when she published her data, when she published her research, it was just considered as a kind of supporting experiment on whatever Watson and Crick has said, but actually it was her work. It was her work. She didn't get the credit for that, right? She's also referred to as the Dark Lady of DNA. Yeah, like she has that other name. So Rosalind Franklin is mainly responsible for that, and yes, she was betrayed, right? So no doubt in the paper, in the NCERT, we say that Morris Wilkin and Rosalind Franklin, their X-ray crystallography data, Watson and Crick used, but actually the story is different, right? So Watson and Crick, James Watson and Francis Crick, no doubt they explained the structure of DNA. They got the Nobel Prize for that as well, but it was not just their work. So they also, because Chargaff's discovery was also there, Chargaff's rule, right? On the basis of that, they understand that complementarity thing. Then they took this picture, they got to know about it. So here the scientists' names are given, right? And all of it is important. Okay? All of them are important. Is that clear? So let's discuss about the Chargaff rule. Let's discuss about the Chargaff rule. So what do you know about it? What do you know about it? Yes, what do you know about it? About the Chargaff rule? Yes. Yes, bache sign. What do you know about the Chargaff rule? Arvind Chargaff. Base pairing. Mention the points here. Mention the points here. Mention the points here, quick. Quickly mention the points here regarding the Chargaff's rule. Yup, the percentage of adenine and thymine should be same. That's it. Nothing else. That's it. Nothing else. Yes, please. That's it. Nothing else. Bache, obviously, Chargaff in 1950, he made the molecular estimates of different components of DNA. So when it comes to his observation, I'll tell you some important observation that are generalized as Chargaff rule. So molar amount of purines are equal to pyrimidine. Okay, bache? Okay, bache? Molar amount here, you can simply say the quantity of purines is always, equals to pyrimidine. Am I right here? It is always equals to pyrimidines. So it means purines, A plus G is equals to this T plus C. So can I write it like this? Can I write it like this? Please tell me. Can I write it like this? That yes, so if the quantity A and G is equals to this T plus C, so the ratio of purine to pyrimidine in all double-stranded DNA is 1, right? So on the basis of that, can I say that the ratio of purines and pyrimidines the ratio of all purines and pyrimidines in a double-stranded, be specific here, I'm talking about the double-stranded DNA because they show the complementary base pairing, we'll discuss about it. So ratio of purines and pyrimidine in a double-stranded DNA is equals to 1. Is equals to 1. Understood? This part clear? A and A plus G is equals to T plus C. Yes? Yes. Now the second point. Now the second point here, that the molar amount of, this is what he observed, adenine is always equals to thymine, and molar amount of guanine is always equals to cytosine. Is that clear? Is that clear? So if I am saying, chalo, I'll write it here only. Okay, so if I'm saying A, so basically I'm saying A is equals to T, C, and G is equals to C. That's what I'm saying. So third important point is that this A plus T over G plus C, this is species specific, right, bache? The ratio of A plus T over G plus C. Focus here, it is A plus G. I'm talking about the purine concentration here to the, right, the ratio of purine concentration to the pyrimidine concentration, that is equals to 1. Right, bache? That is equals to 1. But now I'm talking about on the things on the basis of the pairing here, that the ratio of A plus T and G plus C, the ratio of A plus T to G plus C is variable. It changes, but it is species specific. Okay, okay, it is variable but is constant for species. So in simple words, I can say that this ratio is species specific. This ratio is species specific. This ratio is species specific. Am I clear? This ratio is species specific. Understood? Sure? Pakka? So X-ray crystallography produced by and X-ray diffraction data produced by Morris Wilkin and Rosalind Franklin. So they proposed a very simple but famous double helix model for the structure of DNA, and bache, one of the hallmarks of their proposition was the base pairing between two strands. And this proposition was also based on the observation of Erwin Chargaff. So they got the Nobel Prize, but ultimately they got the contribution from different teachers. Okay, different not the teachers, different scientists. Okay, bache. Okay. Chaliye. So salient features of DNA. It is made up of two polynucleotide chain. Chalo. So you know the DNA structure. Let's move here very quickly. It's something like that. So 5' end, 3' end. Right, 5' end, 3' end. So it is anti-parallel. So it simply means, see, that's the case. That's the case. 5' to 3', 5' to 3'. Anti-parallel it is. Right? So arrow is indicating about the direction of synthesis. And we know that they have the pairing in between. So it's simple, bache, right? See, the nitrogenous bases are there in center. If there is adenine, you know that there will be thymine. If there is cytosine, there will be guanine, right? That's what we know. That is what we know. Yes or no? That is what we know. So now when you talk about this DNA here, the diameter is 2 nanometer or 20 angstroms. 2 nanometer or 20 angstroms, right? Matlab basically I'm talking about this. This diameter is 2 nanometer or 20 angstroms. Plus, let's say here you have one base and they are forming the base pair. This is your next one. So the distance between two is 0.34 nanometer or 3.4 angstrom. So basically when you talk about this now, let's say this part is considered as the groove here, minor groove. In the structure of DNA, that's how we consider it, minor groove. This part is the major groove, right? And definitely proteins are interacting here. So one minor groove and major groove, they form one turn of DNA helix. So in one turn of DNA helix, bache, you will see 10 base pairs, right? And that too specifically in bDNA, explained by Watson and Crick. Right, that in some DNAs it can be 11, it can be 11.5, it can be 12, but here it will be 10, right? 10 base pairs are there. Okay, so when you talk about this thing now, right, you will see one turn of helix, your one pitch of helix, it is, right, it is 3.4 nanometer or 34 angstrom. So that's what you have to remember. Like if I'm talking about the one turn of DNA helix, if I'm talking about one turn is having 10 base pairs, and I told you already one base pair is having a distance of 0.34 nanometer or 3.4 angstroms. Take it, bache? So if 10 is there, 10 multiply 0.34. Yes, so you will get 3.4 nanometer or 34 angstrom. So you should know about it. So let's say in the paper question can come from this part. They can even tell you that in one turn of DNA helix, instead of 10, they can tell you 12 base pairs are there. But the distance between two base pair is 0.34 nanometer. So you multiply it, you'll get the answer, right? So if you know about these things, if you understand these things, then if in the paper they change the statement, they alter the things, you'll be able to answer it, right? So let's discuss the structure here. So when you talk about the salient features of the DNA, we say that it is made up of two polynucleotide chains. Backbone is constituted by sugar phosphate and bases project inside. Are you clear with this particular line, bache? Are you clear here with this particular line? Yes. Are you clear here with this particular line? The sugar phosphate backbone and the bases project inside. Done? Done? So two chains have anti-parallel polarity. They are anti-parallel. One is in this direction, another is in this direction. Right, bache? So if one is in 5' to 3', then other has to be in 3' to 5' direction. So bases in two strands are paired through hydrogen bonding, and then they form the base pair. Adenine form 2 and cytosine and guanine form 3 hydrogen bonds with each other, right, bache? So two chains are, and yes, always remember, always a purine comes opposite to pyrimidine, and this generates approximately uniform distance between the two strands of helix. That's why it is important. This question clear? This statement clear? Do mark it in your NCERT. Do mark it in your NCERT that as a result always a purine comes opposite to the pyrimidine. So this generates approximately a uniform distance between the two strands of helix. Between the two strands of helix. Understood? Between the two strands of helix. Understood? Chaliye. So the two chains are coiled in a right-handed fashion, bache. So the pitch of helix is 3.4 nanometer, right? And a nanometer is 1 billionth of a meter. So if you have to write 1 nanometer, it is equals to, just we are saying that it is equals to 10 angstrom. So same way it is also equals to 10 raised to power minus 9 meter. Keep it in your mind. It is equals to 10 raised to power minus 9 meter. So that is the complete pitch of the helix. That is the complete pitch of the helix. Okay, what's that? That is the complete pitch of the helix. So there are roughly 10 base pairs in each turn. There are roughly 10 base pairs in each turn. Understood? So the distance between a base pair is approximately 0.34 nanometer, bache. So the plane of one base pair stacks over the another in a double helix. So this, in addition to H bonds, confers stability of helical structure. Important it is. So hydrogen bond is there for the stability, plus the pitch or the plane of one base pair stacks over the another, that is also providing the additional stability. So you look at the structure here. Please observe this diagram. Please observe this diagram. Okay. Please observe this diagram. So whatever I have discussed, whatever I have discussed regarding the DNA structure, regarding the sugar phosphate backbone, regarding the pairing, right? Regarding the pairing, all the points are given here. And the points that I marked as important, they all are previous year questions. Do not miss it. So 2 nanometer, major groove, minor groove, major groove, minor groove. So this part, this one major minor, this one major minor, they are forming one turn of helix. And in this one turn of helix, you have 10 base pairs in BDNA. You have 10 base pairs in BDNA. Clear, bache? So look at this 3' end OH of first sugar, 5' phosphate of next, 3' 5' phospho diester bond. Look at this. So this is your sugar phosphate backbone. Nitrogenous bases are there in center, which are connected together with the hydrogen bonding. Doubt? Yes, doubt. Yes, please, doubt. Done. Look at the structure, look at the bonding, look at the way they are present. So these diagrams are literally amazing to understand how things are there. Okay, how things are there. Done? Hello, done? The structure clear? So it's a triphosphate, right? So that is how it is. Chali. So next is the packaging of the double helix DNA, right? Let's discuss that. Now the another important part. So from this chapter, everything is important. Everything is important from this chapter. Okay, bache? Everything is important from this chapter. Okay, so the proposition of a double helical structure of DNA. For DNA and its simplicity in explaining the genetic implication became revolutionary. Obviously, that's why they got it the Nobel Prize now. So very soon, Francis Crick proposed the central dogma in molecular biology. So this central dogma in molecular biology just tells us about the flow of genetic information to DNA to RNA and RNA to protein. DNA to RNA and to RNA to protein. So DNA, its replication is there. It replicates like this. DNA will form mRNA. This is the transcription. DNA will form mRNA. This is the transcription. mRNA will form protein. This is the translation. This is the flow of information. DNA will form mRNA, that is transcription. And who gave it? Francis Crick. Who gave it? Francis Crick. So no, here you are not allowed to give credit to Watson. It's about the Francis Crick only. Again, previous year question it is. And I think this flow is the favorite question of your examiner. You can check PYQs. If I talk about last 15 years ke paper na, I think in minimum in 7 to 8 papers, this question will be repeated. This question will be repeated. Okay. So this is the flow of information. But other than that, bache, so this is the flow of information. Other than that, we also discuss central dogma reverse. Right? We also discuss central dogma reverse. What do we discuss? We also discuss central dogma reverse. Yes, bache, what do we discuss? We also discuss central dogma reverse, in which we see that RNA will form DNA, and that DNA is single-stranded, that is the complementary DNA, and then we'll further see that DNA, then RNA to DNA, and then again the same way the things will be carried forward. So it is the reverse transcription. It is possible with the help of enzyme, reverse transcriptase, and we also call it as teminism. This central dogma reverse, we also call it as teminism. We also call it as teminism. We also call it as teminism. Is that clear? Is that clear? Is that clear? So it is the central dogma reverse. It is the teminism. right? Reverse transcription it is RNA to DNA. Okay, teminism, reverse transcription is teminism. So this is the flow of information. This is the flow of information. So if you read NCERT, you are going to see this, right? And even in some state board's book, this point is given. Okay, DNA is good for. DNA is good for what? It is good for the storage of information. It is good for what? The storage of information. Information is stored in the form of, in the DNA. Information is stored in the DNA. But as I said, DNA will form mRNA and then it will form protein. So RNA is required here. Essential processes are around RNA, right? This is RNA splicing be here also RNA formation. So basically it tells us about the RNA word concept, right? RNA word concept. Ki RNA came first because RNA is also a nucleic acid, but RNA can also work like an enzyme, ribozyme, right? So some people believe that proteins, they came first. No, it's the RNA that came first. Clear, bache? So DNA, right, it is good for the storage of information, but when it comes to the transmission of information, right, DNA is good for storage of information, but when you talk about the transmission of information, right, information has to be transmitted, RNA is better. Am I right? Am I clear here? RNA is better. RNA is better. So for storage of DNA, this is for the storage of information, you have DNA, but when it is the transmission of information, RNA is better. Understood? Understood? RNA is better. Okay? Doubt? Any doubt? So for the transmission of information, RNA is better. Chale, next point, packaging of double helical DNA. Right, देखो, let's start with this diagram. Okay, let's start with this diagram. You talk about the DNA. DNA is not a very short, short molecule or something. No, it's a very long polymer. Right, it's a very long polymer. If I talk about the humans, right, our DNA is 2.2 meter long. It's a long polymer and we have to fit it in 10 raised to power minus 6m nucleus. That is so small. So if we want this DNA to be there in the nucleus, we have to pack it. We have to pack it. Let's say if you are having a bed sheet, right? You cannot expect that bed sheet to be directly to fit in the in your traveling bag or something. You have to fold it now. You have to fold it properly, right? And then you will put it in your traveling bag. Then you are going to put it in your traveling bag. Same is the case here. So you have the DNA which is 2.2 meter long, and in comparison to that, the size of the nucleus is very small. The size of the nucleus is very small. So we have to pack it so that it will get fit there, right? So it is regarding the eukaryote. The DNA packaging in the case of eukaryote. So ye dekho, double helical DNA, diameter is 2 nanometer. So you will see that how will it coil over the histone proteins. Then after coiling over the histone proteins, it is forming the nucleosomes. It is basically forming the chromatin. It will further coil to form solenoid, and it will further coil to form radial loop. And finally what you people are going to get, you people will get chromatid and the chromosome there, right? That chromosome that we see, right? The chromosome, this chromosome, right? When will you get it? You will get it after S phase, when DNA will get duplicated. So at the time of division, at the time of cell division, right, bache, the genetic material, the chromatin, it gets condensed. Then you will acquire this form, right? And even this is also your chromosome. It is not chromated. It is also chromosome having single arm. It is monad. Here the DNA amount is 2C, but this means the DNA amount is 4C. Okay, so that's how it is. So that is how it will get packed. Okay, and this packaging is important. This packaging is important, and this packaging is in the case of prokaryotes. This particular packaging is in the case of prokaryotes. Now we have to discuss it. So, bache, when you talk about the prokaryotes, take the example of a bacteria. It is having what? It is having the nucleoid there. Nucleoid is the genetic material there. There is no well-defined as per NCERT. Yes, bache, as per NCERT, there is no well-defined nucleus there. Am I right? As per NCERT, there is no. As per NCERT, there is no well-defined nucleus here. Am I right? As per NCERT, there is no well-defined nucleus there. Am I right? Am I right? There is no well-defined nucleus. Right, the genetic material is there in the cytoplasm, right, and we call it as nucleoid, and it is also the naked DNA. It is the naked DNA, not covered with, not present in the nucleus, and it is devoid, mane without, devoid of histone proteins. So in the case of prokaryotes, you won't see histone proteins. So this is the nucleoid, also known as genophore, also known as genophore, also known as prochromosome, right? So obviously here also the coiling is there. Even in the prokaryotes also the coiling is there. But you people will not see the histones there. In prokaryotes there are another positively charged proteins, right? You do not have the histones there. You have another positively charged proteins. You can say some polyamines actually are present there, right? Some polyamines are present there in the cell, right, in the prokaryotic cell, and that will help in the coiling. So you can see here when you talk about the DNA packaging, right, so firstly this information is also important. This information is also important. Please pay attention. So taken the distance between two consecutive base pairs, one after another, 1, 2, first one, second one, third one, like this. So we know that the distance is 0.34 nanometer or 0.34 into 10 raised to power minus 9 meter. Yes, this information is clear or not? Right, when you talk about the DNA, the distance between two consecutive base pairs, so we are saying it is 0.34 into 10 raised to power minus 9m. It is 0.34 into 10 raised to power minus 9m, yes or no? It is 0.34 into 10 raised to power minus 9m. So if the length of double helical DNA in a typical mammalian cell is calculated, how can you calculate it? Total number of base pair with the distance between consecutive base pairs. 6.6 into 10 raised to power 9 multiply 0.34 into 10 raised to power minus 9 meter. Right, that's how you can check the distance, and it is approximately 2.2 meter. Right. Right, so how can we check the distance? Total number of base pairs multiplied by the distance between the two base pairs, two consecutive base pairs. So length that is far greater than the dimension of a typical nucleus. That's why the packaging of double helical structure of DNA is required. Am I clear? Am I clear here? Yes, so this packaging is required. This packaging is required. Okay? Okay. Take. Chaliye. So in prokaryotes, just like your E. coli, they do not have a defined nucleus. DNA is scattered throughout the cell. Right, so it is obviously negatively charged, so it will held with some positively charged protein. They will form nucleoid, right? So here you will see large loops held by proteins, but as I said, you don't have histones here. You will have polyamines. You will have polyamines. What will you have here? Polyamines will be there. Excellent. Next part is regarding your eukaryotes. So when you talk about the eukaryotes, na bache, we divide it. In the NCERT, so in general it is explained na. So when you talk about the packaging there, you will talk about the first thing, nucleosome model. Second level of packaging mein solenoid model. Third mein radial loop model will be there. Right, so nucleosome model is there. Yes, nucleosome model, solenoid model, and after that what is there? Radial loop model, which is also known as dangler string. Right, so these are the different levels of packaging. So obviously question will come from mainly from this part. They talk about the histones here. So yes, the question can come. Right, the question can come. Okay, so see, DNA, as I said, being a negative entity. Okay, it will coil over positively charged proteins. It will coil over positively charged proteins, yes or no. So when you are talking about the positively charged proteins, we discuss about what? Histones. What do we discuss? Histones. So do we have histones there in prokaryotes? No, they are present in eukaryotes. So histones are positively charged protein. So bache, we know that protein is made up of amino acids. Many amino acids, they combine. Many amino acids, they combine, they join. Right? Many amino acids, they combine, they join with the help of peptide bonds. How do they join? They join with the help of peptide bonds. Right, bache? And they will give us the protein. So if histones, they are positively charged, means they will have what type of amino acid? Basic amino acids. What kind of amino acids do they have? Basic amino acids. Basic amino acids are the one that are positively charged. So basic amino acids are the one that are having 2 NH2 group and 1 COOH group. Are you getting my point? One acidic group is present there and 1 and 2 NH2 group. So obviously the net charge will be plus. The net charge will be plus. So overall, if you ask me about the basic amino acids, that will be whole. Whole means histidine, arginine, lysine. That will be whole. Histidine, arginine, lysine. That will be whole. Histidine, arginine, lysine. So in the histones, but you do not have histidine, it will be lysine and it will be arginine. So again, it is a previous year question. So lysine and arginine is present in the case of histones. So they are positively charged proteins. So we have the example here, of course. You have, I'm sorry. So you have H1, H2A, H2B, H3, H4. These are the 5 histone proteins that are playing role there in the packaging. H1, H2A, H2B, H3, H4. H1, H2A, H2B, H3, H4. Right, bache? So again, that is also previous year question. So now here in this case, your H2A, 2 subunits of H2A, 2 subunits of H2B, 2 subunits of H3, 2 subunits of H4, they form an octamer. So bache, octa mane 8. Octa mane 8. It's like you have 2 Kit Kat chocolates, 2 Dairy Milk, 2 5-star, 2 Temptation. ठीक है? Total 8 chocolates are there. Total 8 chocolates are there. ठीक है? Okay, it's like this. It's like this. So 2 H2A, 2 H2B, 2 H3, 2 H4. So it will be an octamer. Octamer. 8 subunits are there. So in the paper they can ask you that H1, whether H1 is the part of octamer, yes or no? No. So bache, that double helical, that coiled structure, that DNA will coil over this octamer. Right? This is your one octamer. Let's say this is your next. Like this, like this. So when the DNA coil over this octamer, then we use the word nucleosome for it. And here you people will see that your H1 act like a linker. It is the linker histone protein. It is attached to this linker DNA. So what's going to happen? You have the DNA will coil over the histone protein. So when DNA will coil over these histone proteins, which are forming an octamer here, you will use the word nucleosome. Right, bache? So obviously there will be so many nucleosomes present, and that's how they are going to join. So this is one nucleosome, this is another. So in between this DNA, which is available, you call it as linker DNA or connecting DNA. Linker DNA or connecting DNA. And with this linker DNA, linker histone protein, your H1 is also present here. Right, your linker histone protein H1 is present here. Right, bache? So this structure, it looks like the beads on a string. Right, it looks like what? It looks like beads on a string. It looks like beads on a string. How does it look? It looks like beads on string. How does it look? It looks like beads on string. Is that clear? Is that clear? Okay. Done? Sure. So under electron microscope, it appears like beads on string. So this is your nucleosome. When DNA is coiled over this octamer, this is your nucleosome. And now, and H1 is the linker histone protein. It is not the part of octamer. It is not the part of that octamer. It is associated with that linker DNA. So one thing that you have to remember here, bache, this DNA, because it is not very tightly packed. It is not very compactly packed. So this DNA is available, right? The genes present here are available for the mRNA formation, right? So we call it as transcriptionally active. What do we call it, people? Yes, what do we call it? We call it as transcriptionally active. We call it as transcriptionally active. What do we call it? We call it as transcriptionally active, and the name here is Euchromatin. The name here is Euchromatin. The name here is Euchromatin because it is transcriptionally active and it is loosely bound. And it is loosely bound, right? Lightly stained. And the one which is very tightly packed, the genes which are not available for the transcription, they are heterochromatin. So this part clear? This part clear? Yes. This part clear? If not, do let me know. If not, do let me know. Histones clear? Right, so they have the positive charge. Okay, okay. So if you talk about a typical nucleosome contains around 200 base pairs of DNA. Just imagine. 200 base pair of DNA, right, bache? So nucleosome constitute the repeating unit of a structure, right? And that is known as chromatin. Done, bache? That is known as chromatin. Is that clear? Is that clear? So this linker DNA part clear. So overall, in the case of nucleosome, like your DNA is 20 angstrom, right? 2 nanometer. So when you talk about this nucleosome, if you talk about the diameter here, so it will be around 100 angstrom. Yeah, sometimes it is also written as 11 nanometer in some books. So that this is the first level of packaging. Can you see here? Yes, right? So you also go for this thing, 11 nanometer, the diameter of this nucleosome, right? So nucleosome core is having 8 histone molecules. H1 is the linker one here. So each nucleosome contains 8 histone proteins around which the DNA wraps 1.65 times. Okay, and as I said, around 200 base pairs will be there on each nucleosome. Okay, bache? Okay. Yes, please tell me. Is it okay? So in mammalian cell, we can say approximately 33. In mammalian cell, you can say that approximately 33 million nucleosomes are present. Just imagine, because they are forming this chromatin, right? They are forming this chromatin. So approximately, right, because in mammalian cell, what do you have? 6.6 into 10 raised to power 9 base pairs. 33 million nucleosomes. 33 million nucleosomes. Right? So now, bache, see, this is the second level of packaging. This part is the second level of packaging. Now this nucleosome is further coiling. Matlab, it's like, it is like, see, it is like this. It is like this, right? This is your DNA, you are coiling it. You are coiling it. Okay, fine. We are coiling it. We are forming the nucleosome. Now, what are you doing? You are further coiling it. Just to show you, right? You are further coiling it. So same way, you have the right. You have the DNA nucleosome. It is forming the chromatin. It will further coil. It will form solenoid here. Look at the diameter, 30 nanometer. So nucleosome fold up to produce a 30 nanometer fiber. It's a solenoid wire. It's a solenoid fiber. So it is having 6 nucleosomes. Imagine 6 nucleosomes per turn. The way it is coiling to form a fiber or wire-like structure, 6 nucleosomes per turn. Then it will further coil to form radial loops. So this is the third level of packaging. And here you will see there will be some acidic proteins. Some negatively charged proteins will also be the part of the coiling here. Is that clear? Is that clear? Right. So each chromosome in the center, it has 1 or 2 interconnected scaffolds now. Right. So they will have non-histone chromosomal proteins. Okay. Okay. So when you talk about these loops, you will see non-histone chromosomal proteins are there. And let me tell you, they are acidic also. These are non-histone chromosomal proteins, and let me tell you, bache, they are acidic also. It doesn't matter that they need to be positively charged only. No, they are even negatively charged. Okay, they are even negatively charged. Understood? So each chromosome in the center has 1 or 2 interconnected scaffolds that are made up of non-histone chromosomal proteins. Right? So over this scaffold is basically the solenoid fiber. Right? And the lateral loops are there. So that's how it further coils. And look at this, how they are coiling. Right? How they are coiling. And finally, it will form that chromatid and ye dekho, chromosome. Look at this. So when you talk about this, its diameter is 700 nanometer. 1400 nanometers. So see, we started from 2 and that's what we have. Right? Beads on string. It is showing chromatin, the linker DNA in between. Right? The nucleosome. Look at the diameter. In some books 11 is given, in some books 10 nanometer is given. Then 30. This is a solenoid fiber. Further, the radial loops, and that's how you will get your metaphase chromosome. Understood? Any doubt? So what do you have here in the histones and all? Clear? Ye dekho, active genes, right? Because they are transcriptionally active, loosely folded, right? Bache, loosely, lightly stained. When it is very coiled, right? Some genes, they will not be available for the transcription. That will be heterochromatin. Okay. Okay. So in the interphase nucleus, it is in the resting stage, right? You know that the DNA is present in the DNA is not coiled. It is not compactly arranged. It is in the form of loose threads. That's how it is. We use the word chromatin, chroma, color. It will get stained with the basic dye. That's the point here. Understood? That's the point here. So let me show you this part from the NCERT. Then let's move to the experiments. Okay. Let's move to the experiment. So you will get a lunch break. Not now. Not now. Wait till 2. Okay. Wait till 2. So there is a set of positively charged basic proteins called histones. Any doubt? There is a set of positively charged basic proteins called histones. Any doubt? Yes, people, any doubt? So protein acquires charge depending upon the abundance of amino acid residue. So histones, they are rich in basic amino acid like lysine and arginine. So as I said, that's a previous year question. Previous year question it is. Right? So both the amino acid residues, they carry the positive charge. Why? Because they have 2 NH2, 1 COOH, okay, in their side chains. So histones are organized to form a unit of 8 molecule. And what do we call it? We call it as histone octamer. What do we call it? We call it as histone octamer. What do we call it? We call it as histone octamer. Understood? Understood? So, the negatively charged DNA is wrapped around the positively charged histone octamer, and it will form nucleosome. So a typical nucleosome contains 200 base pair of double helix, right? So nucleosome constitute the repeating unit of a structure in nucleus. We call it as chromatin, right? So they are thread-like stained bache. They are thread-like stained bodies. They are seen in the nucleus. Okay, so it looks like beads on the string under the electron microscope. And then bache, for the further packaging, non-histone chromosomal proteins are there. So even in the NCERT, see, they have clearly mentioned this thing, that in a typical nucleus, some regions of chromatin are loosely packed and are referred to as euchromatin, right bache? So, but the one which is densely packed and it is transcriptionally inactive, it is heterochromatin. These things are important. Okay. Okay, doubt. So euchromatin, transcriptionally active, transcriptionally inactive. DNA is loosely packed, actively present in prokaryote and eukaryotic genome, genetically active, present at inner side of nucleus, stained lighter, early replicate, they are not sticky, allow gene to form a protein. That's what you need to remember. Yes, doubt. Yes, people, doubt. Do you have any doubt? Do you have any doubt? Great. So next is the search for genetic material. Let's talk about that experiments and then let's move to the DNA replication. So if you will complete this chapter properly, you can ensure your 24 marks. Then let's move to the biotech unit, which is not very lengthy, right? Which is not very lengthy. We have to finish it ASAP, right? So here in this chapter, everything written is important. Everything given is important, okay? Okay. Done. Angie, so doubt? Ma'am, you are teaching very fast. Sheetal, I can't help it. It's a marathon session. If you want me to teach slow, yeah, if you want to go slow, you can watch other lectures of mine, the recorded one, right? Already available on this channel. Okay, any doubt? Hello? Nah, ma'am, we don't have doubt. है ना? We are super duper intelligent. We are super duper intelligent. We don't have doubts. Okay, chalo. So next is the experiment part that we need to understand. Okay, so when you talk about the experiments, you know it, right? That quest, that what exactly is the genetic material, it started very late, right? So when you talk about the experiment, the two experiments that you need to keep in your mind is Griffith's transformation experiment and Hershey and Chase transduction principle or Blender's experiment. These are the two most important one. Okay, so now let's talk about this transforming principle, and as I said, it is given by Griffith. So it's Griffith's experiment, Griffith's transforming principle. And this experiment is not telling us that what exactly is the genetic material. It is telling us that there is some something which can transform one bacteria into another. Let's take the example. So here when you talk about this particular experiment, right, what he did, he took the mice obviously, right, and he took a bacteria. Which bacteria? Streptococcus pneumoniae, also known as Diplococcus pneumoniae, right, or sometimes it is also referred to as pneumococcus. So this is a bacteria. What type of bacteria it is, bache? It's a coccus. It means it's a spherical bacteria. What type of bacteria it is? It's a spherical bacteria. What type of bacteria? Spherical bacteria. But when you talk about this particular bacteria, you have two strains here, right? What do you have here? You have two strains of this bacteria. What is the name of this bacteria? It is Streptococcus, yeah, Diplococcus or pneumococcus. So here in this particular bacteria, one strain is the RS strain and another strain is the R strain. So these are two different strains. S mane smooth means here in this particular bacteria, when you grow this bacteria, you will get smooth colonies. Very smooth colonies you are going to get. Why? Why? Because it is having the outer covering. That polysaccharide coat is there. Right? Like when you talk about the bacteria, you say na cell membrane, cell wall, glycocalyx or cell coat is there. So here in this S strain bacteria, what will you see? That it gives us the smooth colonies, right? And it is the virulent one. Virulent means it is the pathogenic one. It causes the pneumonia, right? It causes the pneumonia. It causes the pneumonia, and that pneumonia can even kill, right, bache? Right, bache? That pneumonia can also kill that particular mice. Okay, when it is the R strain, when we grow it, we get the rough. R stands for rough. So it is non-virulent, matlab it doesn't cause pneumonia. It is non-pathogenic. It is non-pathogenic, right, bache? So it is also referred to as R2 strain, S3 strain. So it is the virulent one and the non-virulent one. Yes, bache, doubt? One is the virulent, the one which causes the disease, and one is the non-virulent one. Any doubt? Any doubt? R2, S3. R2, S3. Smooth, virulent, pathogenic. Right, so it is having that polysaccharide coat that can cause pneumonia. The other one is not causing it. So obviously, bache, see, this is about that experiment. When that S strain, when it was injected into the mice die. Why? Due to pneumonia. Right, he conducted the experiment, Mr. Griffith, he conducted the experiment and that's what he got. Right, S strain, it was injected into the mice die. Now, when it is the R strain, when it was injected into the mice live. These two things clear? Why? Because it is the non-virulent one. It is the virulent one. It is the non-virulent one. It is the virulent one. So Griffith was able to kill bacteria by heating them. Now in the third experiment, what he did, he was like, yeah, I will take heat-killed S strain. Heat-killed S strain means I will give the heat. What is going to happen? That polysaccharide coat will be destroyed. It will be non-pathogenic. So when that heat-killed S strain, when it was injected into the mice live. These three things clear? These three things clear? These three things clear? Yes or no? Yes or no? These three things clear? Yes or no? Yes. So then what he did, he was like, he took heat-killed S strain and R strain, which was light, but both are non-virulent. Even the heat-killed S strain cannot cause pneumonia. Even the R strain cannot cause pneumonia. So when they both were injected into the mice die. So it was just like a question mark for Griffith, that how is it possible? Because both are non-virulent, then how mice can die? Then he realized that something from this S strain, something from this heat-killed S strain transformed the R strain in such a way that even R strain started forming that polysaccharide coat that caused the pneumonia there. Right? So he was not able to explain. He was not able to explain that it is the DNA which is the genetic material, but he basically explained it in the form of some transformation. Right? Just say, for an example, he concluded that R strain bacteria had somehow been transformed, right, by the heat-killed S strain. So some transforming principle transferred from heat-killed S strain, it had enabled the R strain to synthesize a smooth polysaccharide coat and to become virulent. So this must be due to the transfer of genetic material. However, the biochemical nature of genetic material was not defined from his experiment. Understood? 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 Now, the next thing is this: biochemical characterization of the transforming principle. Next is what? Next is the biochemical characterization of the transforming principle. So when you talk about it, here you are going to talk about Oswald Avery, Colin MacLeod and McLean McCarthy. Again, PYQ, right? So they work to determine the biochemical nature. But what exactly is that transforming principle? What exactly has transformed that R strain into the S strain, right, bache? So they purified biochemicals like proteins, DNA, RNA, right, from that heat-killed S cell, They purified proteins present in that heat-killed S cell, DNA and RNA, because they want to check that which has transformed that R strain into the S one. So they discovered that it was the DNA alone from S bacteria that can cause R bacteria to get transformed. So what was that experiment? Let's say this is the R strain here. Obviously, if you will add the protein, just asking you this thing, if you will add the protein from heat-killed S strain, if you will add the protein from heat-killed S strain bacteria, what do you think? Yes, what do you think? This R strain will get transformed or not? What do you think? This R strain will get transformed or not? Quick. This R strain will get transformed or not? Acha, protein from heat-killed S strain will transform R strain. So those who are saying yes, they really need to end this class, right? They really need to end this class. They should go outside, they should meet people, they should go and party, because they are not able to understand it. So it's a no. How can protein transfer R strain? We know it. If you add DNA, then DNA will transform it, right? Like when they were experimenting, they were not aware of it, but you people know it. You people know it. Strange, right? That's strange. So basically what he did, from the heat-killed S strain, he took protein, RNA and DNA. Now let's say, if this is a culture where you have R strain plus you have everything from that heat-killed S strain, you have DNA, RNA and protein from S strain, heat-killed S strain. So what do you think? And yes, now you have added the protease here. What do you think? Transformation will occur or not? These are my questions that you need to answer. This is here in this test tube you have R strain, you have DNA, RNA and protein from the heat-killed S strain. Now you have added the protease here. What do you think? The transformation will occur or not? What do you think? Transformation will occur or not? What do you think? The transformation will be there or not? Transformation will occur? Yes, ma'am. Why? Why? Because it is the protease. It will digest protein. It will eat that protein, and protein is not transforming. Now, if you add RNAs here, right? If you add RNAs here, what is RNAs? RNA digesting enzyme. So what do you think? Transformation will be there or not? Transformation will be there or not? Transformation will be there or not? Yes. Now let's say here you have R strain and from heat killed S strain again DNA, RNA from heat killed S strain, DNA, RNA and protein is present. Now you have added DNase here, DNA digesting enzyme. So you tell me transformation will occur or not? Transformation will occur or not? No transformation, because DNase will, right? No transformation. Why? Because DNase will digest the DNA and it is the DNA which is responsible, right bache? So that's how it is. Okay, so as per NCERT, you can mark it that they also discovered that protein digesting enzyme protease and RNA digesting enzyme RNases, they did not affect the transformation. So transforming substance was not protein or RNA. So digestion with the DNase inhibit the transformation. So it is suggesting that DNA caused the transformation. They concluded DNA is the hereditary material, but not all biologists were convinced. So they were right, but all biologists, they were not convinced. Okay, so then we have the unequivocal proof, right? the unequivocal proof that it is the DNA that is the genetic material, it came from the experiment of Alfred Hershey and Martha Chase, 1952. It is important. The discovery is important. The scientist's name is important. The year is important. Right? It is important. Right? So they worked on a virus that infect the bacteria, that is bacteriophages. So bacha, your bacteriophages are hyperparasites. Right? They are hyperparasite. Why? Because it is parasitic over, it's a parasite of parasite. It's a hyperparasite. Right, bache? It's a parasite of parasite. It's a hyperparasite. So what are you talking? You're talking about the experiment here, right? It is the Hershey and Chase experiment. You're talking about the bacteriophage here, and to be specific, it is T2 bacteriophage that was used. So what is the name of this experiment? It is also known as blending experiment and transduction principle या transduction experiment of Hershey and Chase. Okay, so transformation is when a cell is accepting the genetic material from the surrounding and it is making it the part of its genetic material. Transformation. Now the word is transduction. When do we use this word, bache? Transduction. When a virus is used to pass the genetic material from one cell to another. When a virus is the agent, when the virus is used to pass the genetic material from one bacterial cell to another. Is that clear? Is that clear? Is that clear? Clear? Right, so this is the bacteriophage, T2 bacteriophage it is. Overall, in the case of bacteriophage, let me tell you, they are double-stranded DNA viruses. So, virus, you know that, how does it work? It is going to use the outside the body, it is living or non-living? Outside the body, virus is living or non-living? Yes, people, outside the body, is it living or non-living? It is living or non-living? Outside the body, virus, non-living, right? So obviously when virus will get the living host, it will pass its genetic material here. It will use the host's machinery. What will it do? It will use the host's machinery to replicate its genetic material and to form multiple copies. To replicate its genetic material and to form multiple copies. So that is how it works. That is how it works, right? It is going to infect it and it will form multiple copies. Now, when you talk about this bacteria, especially this bacteriophage, I believe you know the structure. You know that this one is the outer protein coat and this one is the DNA present here. This one is the DNA present here. So for conducting this experiment, what they use? They use radioactive phosphorus and radioactive sulfur. So S35 and P32, these isotopes were used. Radioactive S35, P32. S35, S sulfur. Where do you see sulfur? You will see sulfur in protein. You will see sulfur in protein. You know na? Sulfur containing amino acids. Yes, the trick for sulfur containing amino acid is CMS, Chief Minister Sir. So it is cysteine, it is methionine. It is cysteine, it is methionine. They are sulfur containing amino acids. Cysteine and methionine, what are they? Sulfur containing amino acids. So in simple word, sulfur is the part of protein coat and P, this is the in the DNA. So two experiments were conducted parallelly. In one experiment, radioactive S35 and P32 was used. Is that clear? Is that clear? So this experiment is having 3 steps: infection, blending and centrifugation. Infection, blending and centrifugation. So when it is the infection, both type of labeled phages, they were allowed to infect these bacteria in separate experiments, right? Both type of labeled phages were allowed to infect. They were allowed to infect normally growing bacteria. Right? They were allowed to infect normally growing bacteria. Right? Labeled phages matlab ye dekho. S35 protein capsule is radioactive. P32 DNA is radioactive. Now there is the blending. Matlab we allowed to infect it. So it means like here right? When scientists, when they were conducting it, they were not clear with it. But we know na these experiments, yes or no? We know these experiments, yes or no? Yes or no? So obviously if a virus is allowed to infect the bacteria, it will pass its genetic material within this bacteria. It is going to pass its genetic material within this bacteria, right, bache? Right, bache? So you have what? You have the infection. This is the first step. Second is blending. So these bacterial cells, they were agitated in a blender so that they can break contact between virus and bacteria, right, bache? Right, bache? So these bacterial cells, they were agitated in a blender to break contact in between virus and bacteria. So basically in first step we allowed the infection, in the second step, right, in the first step we allowed the infection, in the second step we are going to break their contact, and in the third step we will do the centrifugation. So we will separate their components, right? So the virus particles were separated from bacteria by spinning them in centrifuge. So bacterial and viral components were separated. Yes, bache. These components were separated in a centrifuge, right? So what's going to happen? This is see, centrifugation, right? As per the way things will settle down, you know that bacteria and virus is even smaller than bacteria. So when there is the S35 is the case, right? So S35 is the case, means protein coat is radio labeled. Ultimately we know that when there is the centrifugation, the virus components, they will settle down at base, right? Virus component will settle down at base and bacterial component will be above, right? Bacterial component will be above, virus particle will settle down at base. So when S35 was used, so that radioactivity was there at the base. So radioactivity was there in the palate. So this part is the supernatant. No radioactivity was there. So no radioactivity in bacterial content indicates that it is not the protein that will pass to the next generation. That it is not the protein that will pass to the next generation. Now again, here you have the virus component. This is the palate here. And here you have the supernatant. Supernatant is telling us about the bacterial content, right? It is telling us about the bacterial content here, right, baches? This supernatant, it is telling us about the bacterial component here, right, bache? So when you talk about this supernatant, when we were using P32, right, when we use P32, yeah, Hershey and Chase, when they use P32, the radioactivity was present here. So it is indicating what? It is indicating that it is the DNA, because ultimately bacteriophage, the job of bacteriophage is to infect the bacteria, So obviously it is the genetic material that it will pass to the bacteria, and that is the DNA. That's why the bacterial component is getting it, right? So here you guys can see. So when it is S35, no radioactivity detected in cell, radioactivity detected in supernatant. One second. So radioactivity, my bad. So this is the cell component. Heavier thing will settle down at base. My bad. Cell component, the heavier thing will settle down at base, right? But so it is the viral component, because heavier thing will settle down at base. That's the point. My bad. Yes. So it is the heavier thing that will settle down at base. So heavier thing means bacterial cell, its component. So bacterial components are present here at the base. So in the first case, when it is S35, supernatant, having viral components, so radioactivity was there in supernatant, in the viral component. And when you people talk about this P32, yes, when you people talk about this P32, so base is showing us the bacterial cell, या bacterial cell component. So here you will see the radioactivity is present. Here you will see the radioactivity is present. Yes or no? Yes or no? So in first case, it is here in supernatant. but in second case it is here at base. Palate matlab bacteria is receiving it. Okay bache, bacteria is receiving it, that's it, right? So yes, even I just mention it incorrectly, but the point here is that radioactivity, who's going to, basically it is the DNA which will pass, right? It is the DNA that will pass. Okay, so DNA, if it will pass, you are performing the centrifugation, so the things which are heavier, they will settle down at base. That's the point. The things which are heavier, they will settle down at the base. That's the point. Okay, so this is about the Hershey-Chase experiment, and it proved that it is the DNA which passes to the next generation. That's what we need to remember. So the properties of genetic material and everything we will discuss after a break of 15 minutes. So now students, you will not get very long breaks. 15 minutes are more than sufficient, because we have to complete many chapters, right? We need to complete many chapters, and I started with molecular basis of inheritance, and I'll go for the biotic because these 3 chapters are itself very, very, very important. Okay, so yes, we have to speed up and we have to finish it ASAP. Okay, so break time till 2:15 p.m. Okay, done. Okay, so my food was not ready, so I took time. Chalo. So let's continue. Yes, let's continue. So let's talk about the properties of the genetic material, and I believe you understand, right? Why do we say that it is the DNA which is the genetic material? Why can't we say the proteins are genetic material, or fats are genetic material, or carbohydrates are genetic material? Simple, simple. If it is a genetic material, no doubt it should store information, plus it should pass the information, right? There should be a mechanism developed around it that information can be passed from one generation to another. Am I right? That information can be passed from one generation to another. So when we talk about the properties of the genetic material, this is what we have, that it should be able to generate its replica. It should be able to generate its replica. Right, bache? That it should be able to generate its replica. It should be stable chemically and structurally. It should be stable chemically and structurally. Right, bache? It should be stable chemically and structurally. Right, bache? So first thing is, it is able to generate its replica. It is able to generate its replica, number one. Number two is, it should be chemically and structurally stable. It should be chemically and structurally stable. Are you getting my point? Right, bache? So when I'm saying structurally and chemically stable, one thing that you need to keep in your mind, that whenever you talk about the genetic material, and obviously because DNA is the genetic material, DNA is fulfilling these conditions. So that DNA material, it should not be very reactive, that it is always reacting and changing its structure. No, but yes, it should allow the mutations, that too at a slower rate. Right, and that mutations are important for the variation, so that we can adapt in the changing environment. Is that clear? Is that clear? So it should be able to generate its replica. It should be stable chemically and structurally, and it should provide the scope for slow changes. Right, so when we are saying providing the scope for slow changes, so the word that we have is mutation. The word that we have is mutation, and it is required for the evolution. Right? How is it required? It is required for the evolution. Is that clear? Is that clear? Sure? Done, bache? Right? So it should be able to express itself in the form of Mendelian characters. So when you talk about the Mendel's, right? Yeah, Mendelian characters. You remember the concept of factors? Yes. Do you know the concept of factors? Right, bache? That they are paired, they are discrete units, they occur in pairs, right? The way they pass to the next generation. So it is regarding that. Is that clear? So these are the properties of the genetic material, right? These are the properties of the genetic material. Understood? Okay. Right. Now when you talk about the DNA, see, the genetic material should be stable enough to change with the different stages of, not to change with different stages of life. Of course, right, the stability should be there, then only the information will pass now. But slow changes are required, mutations are required, that too at slower pace, that too not at a very fast pace. Some mutations, they can, basically when you are defining mutations, you are saying sudden changes. And where that sudden changes will occur, that sudden changes will be there in the genetic material. That sudden changes will be there in the genetic material, yes or no? Yes or no? Right, that sudden changes will be there in the genetic material, yes or no? Yes or no? Right, bache? So obviously some mutations, like it's not like that mutations, they don't occur. That noticeable change and all the things, they also occur in the DNA. They also occur. But if they will be very quick, if they will be very fast, of course, right, that complete DNA and that integrity will not be maintained. So they are allowed, but at a slower rate. At a slower rate. So further 2-OH group. Further 2-OH group present at every nucleotide in RNA. Now, here we are talking about the DNA and RNA. See students, I told you about the RNA word concept. I told you that some scientists, they believe that proteins, they evolved first. Some scientists, they believe that it is the RNA that evolved first. Some scientists, they believe that it is the protein that evolved first. Some scientists, they believe that it is the RNA that evolved first. These are the two things that we have. Now, when you talk about the RNA, see, DNA will form mRNA, mRNA will form protein. So we can see RNA is helping in the transmission also, transmission of information also. Plus, bache, RNA is also helping in the protein synthesis. It is forming ribosome, the tRNAs. We have that examples. Am I right here? We have that, right, bache? We have that. examples, right? We have that examples, and we see that how the RNA is so involved in the basic metabolic processes of our lives, right? And it is important. So when you talk about the RNA word concept, so we see the importance of this RNA, and plus RNA is a nucleic acid. RNA is a nucleic acid, but RNA can also act as an enzyme. It can also act as an enzyme, and that is ribozyme. We call them as ribozyme. Again, bache, that's a PYQ. That's a PYQ. Right? That's a PYQ. Okay? Okay? Yes or no? Yes or no? This is something you need to keep in your mind. Right? Now, my point here is, that initially it was DNA there. Then from DNA, initially it was RNA there. From that RNA, DNA formed. From that RNA, right? Initially, DNA was there, and initially RNA was there, and from that RNA, DNA formed. Yes, from that RNA, the DNA formed. Yes or no? From that RNA, the DNA formed. Yes or no? Yes or no? Yes or no? Hannah, it is there. Definitely. So RNA is a single-stranded. RNA is a single-stranded molecule. Plus, when you talk about the RNA, which sugar is there in RNA? Ribose sugar. That ribose is having that 2-OH group. So they are saying that that 2-OH group, which is present in the RNA, which is present in the ribose sugar of RNA, it's a very reactive group. It makes RNA labile and easily degradable. Is that clear? That makes RNA, yes bache, that makes RNA labile and easily degradable. Is that clear? So what is the meaning of labile? Something that can be easily changed, that can be easily altered, right? Something that can be easily changed, something that can be easily altered, right? That is the meaning of labile. So RNA is catalytic also. That's why it is reactive. That is also the point. So DNA, chemically it is less reactive, structurally more stable, and that is why with the time from ribose sugar, that deoxyribose sugar came, right? And that deoxyribose sugar is important. That deoxyribose sugar is important, right? So therefore DNA chemically is less reactive and structurally more stable when compared to RNA. So therefore, among the two nucleic acids, the DNA is a better genetic material. In fact, the presence of thymine at the place of uracil also provide additional stability. So these two changes that we discussed, right, they are related to the stability part which is required, right? That is why with evolution, the better things came, right? Okay, done, bache. So both DNA and RNA are able to mutate. Right? If let's say, if you have a statement-based question, right? If you have a statement-based question, if you have SBQs, if you have statement-based question, if you have SBQs, statement-based question, so if they are saying ki DNA is able to mutate, yeah, RNA is able to mutate, so some students, they are like, no, DNA is a stable structure, DNA will not mutate. No, it's not like that. It's not like that. Even DNA can mutate, right, bache? Even it is the DNA that is able to mutate. Are you getting my point? So both DNA and RNA, they are able to mutate. Both DNA and RNA, they are able to mutate, they can mutate. So in fact, RNA, because it is unstable, here the mutations are comparatively at a very fast rate. Okay, and you know that we have the viruses where it is the RNA that is the genetic material, and we use the word retrovirus for that. And such viruses, their lifespan is short, right? Their lifespan is short, but they mutate at a faster rate, right? You can talk about the HIV, you can talk about the COVID-19, that coronavirus, it was also the RNA virus. So because these viruses, they mutate at a faster rate, students, right? They mutate at a faster rate, they evolve very fast, right? So that's why in their case it's difficult, because see, you are studying the RNA, you know, you are studying RNA virus, you know about its structure. Okay, so now you are also learning that how is it working, then you will try to make some vaccine or something regarding that, but immediately it will mutate, right? So that kind of things are there, right? That's why it becomes difficult to deal with such type of viruses. Okay, chaliye. So RNA word concept, what that concept is saying, it is saying very simple thing, that it was the first genetic material, and we have the evidences where we see that how RNA is important, right? That how this RNA is important for the metabolic processes. So metabolism, chemical reaction, translation, splicing, for all it is important. So it all revolves around the RNA. So RNA is the genetic material as well as the catalyst. Right, bache? Right, bache? So RNA being catalyst was reactive and hence unstable. So DNA has evolved from RNA with chemical modifications that makes DNA more stable. Okay, done. And DNA also having the, see, complementary strands, and it resists changes by evolving a process of repair. Yes, when we talk about the replication of DNA, we discuss about the DNA repair. So if base pairs are not attached properly, this DNA can change it, alter it, repair it. So mutations are there, but at a slower rate, not at a faster rate. Understood? Understood? Chaliye. Let's go for this. So see, RNA self-replicating by ribozymes. RNA forms some inorganic sources. RNA forms from inorganic sources. It self-replicates by ribozymes. RNA catalyzes protein synthesis. Membrane formation changes internal chemistry, allowing new functionality. So RNA codes both, what is this, DNA and protein. So protein catalyzes cellular activities and DNA becomes master template. So matlab, it is trying to explain it. Now we need to talk about the DNA replication. That is our next topic, right? We need to talk about the DNA replication. I believe if you're watching this lecture, right, you're not naive, right? You know about this chapter. Maybe not in detail, maybe you are here for the revision, and those who are just starting this chapter, I cannot I cannot accept this fact that students, they don't even know that we talk about the semi-conservative model of DNA replication. Of course we do, we discuss that, right? So it is the semi-conservative model of DNA replication which is the part of our syllabus. Who proposed it? Watson and Crick. I'm not saying that they have provided the experimental proofs. No, Watson and Crick, they proposed it. Watson and Crick, they proposed it. Yes or no? Watson and Crick, they proposed it. They proposed what? They proposed the semi-conservative model of DNA replication. They proposed the semi-conservative model of DNA replication. That is what we have. So you have the double-stranded DNA, right? You have the double-stranded DNA. So these DNA strands, slowly they open, they act like a template. So if you don't understand what is the meaning of template, you can take it like this, that here, you know that the complementarity is there. Actually, Watson and Crick proposed it. When they studied the structure of DNA, they were like, yes, complementarity is there, right? The two strands are complementary to each other. If adenine is present, complementary to it, thymine is there. If cytosine is present, complementary to it, guanine is there, right? If cytosine is present, complementary to it, guanine is there. Are you getting my point? Are you getting my point? These are the things that we see, right? These are the things that we see. Yes or no? Yes or no? So obviously, if on one strand A is there, T is there. So we can use this DNA. We can use this DNA, right? We can use this information, right? We can use this principle of complementarity. We can add the new bases. We can form the complementary strand and we can form the new DNA. That's the point. So semi-conservative model of DNA replication was proposed by Watson and Crick, but experimental evidence, especially in case of prokaryotes, was provided by Michelson and Stahl. So when you talk about the semi-conservative model, I'll explain all others as well. Simple. That's your DNA. That's your DNA. It will open. That's your DNA, right? It will open. It will open. Slowly it will open. So let's say if here adenine, thymine, cytosine, guanine, or things like that are there. And here obviously, so if you are opening it, then complementary to it, you can add new bases. If adenine is there, you know I have to add thymine. If thymine is there, you know I have to add adenine. If cytosine is there, guanine. Guanine is there, cytosine. If guanine is again there, cytosine. That's how you can form a new DNA strand. That's how you can form a new DNA strand. So this older one having information is acting like a template. This older one having information, it is acting like a template strand. So complementary to it, new strand will synthesize. New strand will synthesize. So this one is having the information to form the new strand. This one is acting like a template. To use it, use this information and accordingly as per the rule of complementarity at the basis. So this is the semi-conservative DNA replication, where you will see that daughter cells, they will have one, right? Daughter cells, they will have one parental strand, one newly synthesized strand. But it is not the only DNA replication. Conservative DNA replication is there, where this DNA strand opens, right, bache? Complementary to it, new strands, they get synthesized. So new strands, they join with each other, older strand, they join again, and that's how the flow is. So the information will be, it's not semi means half, it's conservative. That parental strand will be conserved as such. And from that parental strand, it will open, the daughter strands will synthesize, that daughter strands will reanneal, they will join, they will form this double-stranded DNA, and again, they will also follow the same method, conservative. And when it is dispersive, which is very rare, you will see a part of parental strand, a part of parental strand, and in between we see that new strands they form. Just say here, blue and yellow, that is dispersive. Okay, so in our syllabus in detail, we discuss what? We discuss the semi-conservative mode of DNA replication. What do we discuss? We discuss the semi-conservative mode of DNA replication. So the scheme suggested that the two strands, they would separate, they will act as a template for the synthesis of new complementary strand. They will act as a template for synthesis of new decomplementary strands, right? They will act as a template for the synthesis of new complementary strand. Is that clear? Is that clear? Is that clear? So after the completion of replication, each DNA molecule would have one parental and one newly synthesized strand. So it is the semi-conservative DNA replication. Any doubt here? Any doubt here? So who proposed it? Yes, who proposed it? Who proposed it? Watson and Crick. But when it comes to the experimental proof, Matthew Michelson and Franklin Stahel, they performed an experiment. And bache, in 1958, they performed an experiment on E. coli. But they were not the one. They were not only the one, right? We also talk about Tyler and colleagues. Tyler and colleagues. And they experimented in the case of fava beans, Vicia faba. Fava beans. What are they? These are the fava beans. So basically it's a plant. So it's a eukaryotic cell. It's a plant. So it's a eukaryotic cell. Right? It's a plant. So it is what? It is a eukaryotic cell. Tyler and colleagues. Vicia faba, fava beans, eukaryotic cell. So here what's my point? See. And yes, what they used? They used radioactive thymidine. Radioactive thymidine to prove the semi-conservative model of DNA replication in the case of Vicia faba. So once I was solving the PYQs, yes, even I do solve the questions, right, bache? I do solve the questions. So when I solved it, there was a question that who provided the experimental proof of semi-conservative nature of DNA replication in the case of eukaryotic cell. So just like you, I also missed that eukaryotic cell part, and I like Michelson and Stahel. And obviously it was incorrect. So if they are specifying eukaryotic cell, Tyler and colleagues. Plant cell, yeah, eukaryotic cell, Tyler and colleagues. Understood? Tyler and colleagues. Understood? Sure? Right? And in the case of bacteria, your world famous bacteria, E. coli, Michelson and Stahel. Okay? So what they used? They used heavy isotope of nitrogen, and that is N15. It is not radioactive, it is heavy. It's important. It's a PYQ. It is not radioactive, it is heavy. Heavy isotope of nitrogen. Not radioactive, heavy isotope of nitrogen it is. Very simple it is, students. Very simple it is. Dekho, you know that when you talk about the DNA, it is Escherichia coli. You know that when you talk about the DNA, when you talk about what? When you talk about the DNA, deoxyribonucleic acid. In the case of DNA, what do you have? This naturally occurring nitrogen is 14 and this isotope of nitrogen. This isotope of nitrogen is there, naturally occurring nitrogen. This is the isotope of nitrogen that is present there in the DNA. Now I'm taking this cell, right? I'm taking this E. coli. I'm growing it in a culture, right? I am growing it in a culture. Now here in this culture, obviously I'll provide it the food, right? I'll provide it the food. So here in this culture, the source of nitrogen will be NH4Cl, that is your ammonium chloride. That is your ammonium chloride. And I'm using 15N. 15NH4Cl means here when I'm talking about this ammonium chloride, yes, bache, when I'm talking about this ammonium chloride here, so what am I doing? What am I doing? Yes, bache, that's how you have to write. What am I doing? Yes, what am I doing? Basically, I'm providing the source of nitrogen, right? That is the ammonium chloride. So yes, about this chemical, there is a PYQ. So it is having 15N, the heavy isotope of nitrogen. So it is heavy, not radioactive, PYQ, right? So I'm growing it in a culture and I'm growing it for many generations. Why? Because I want this E. coli to have 15N to be the part of its DNA. Is that clear? Is that clear? Now, in the case of this bacteria, remember one thing, that here the generation time is 20 minutes. In the case of this bacteria, remember one thing, and what is that one thing? That generation time, right? What is that one thing? That generation time. Generation time is 20 minutes here, right? What is the one thing that you need to keep in your mind? That here the generation time is 20 minutes. Means in 20 minutes, one E. coli cell, it divides into two. Why? Because it is having bidirectional mode of DNA replication. Why? Because it is having bidirectional mode of DNA replication. Bidirectional mode of DNA replication. Is that clear? Is that clear? Is that clear? ठीक है, चलिए. So this part clear. So now this E. coli having heavy isotope of nitrogen, I am again shifting this E. coli, right? This E. coli is having 15 and nitrogen. I am again shifting it to a source where it is having what? Where it is having NH4Cl again, but here it is having the light isotope, the naturally occurring one of nitrogen. I will allow it to grow here. That's my point, right? I will allow it to grow here. That's my point. Is that clear? I will allow it to grow here. That's my point. Is it clear? Yes, please tell me. Is it clear? Done? Done? Understood? 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 Yes or no? Understood? So obviously I will shift it. Dekho, here we are going to use cesium chloride density gradients, centrifugation will be done. Dekho, I'll give you the context first. This is the diagram that we need to understand here, students. Yes, this is the diagram that we need to understand here, right? Dekho, firstly you have the 15N. 15N means heavy isotope of nitrogen. Right, you will have the E. coli having heavy isotope of nitrogen. Now what will you do? Right, we'll go for the centrifugation also. We will go for the centrifugation also initially, right? And that will be centrifugation in a cesium chloride density gradient. What will you use? I'm writing it here. It is in an NCERT. Centrifugation in in cesium chloride. Centrifugation in cesium chloride density gradient. Centrifugation in cesium chloride density gradient. That's how we will go. So we have to obtain the bands here. Matlab, let's say, just whenever we conduct the experiment, we go for some standard results. Standard results as in, just even if you are going for the lab test, right? Just we say, okay, this thyroid value, right? The value should be in range. So we know about that range before, right? Yes or no, students? Please tell me quickly. We know about that range. Done, बच्चे? Done. So basically, when you go for the test, even in the lab, when we go for it, we know the standard values, that the standard values should be in this and that range. On that basis, we say, right, your values are low or high. Same is the case here. Just say, let's say when you have the E. coli having 15 isotope DNA. So by centrifugation, when you will obtain the band, because it is heavy, it should be here. Let's say it should be here, right? You are doing the same thing with the lighter one also, because it is lighter one, its position will be here. So these are the things that you already know, right? These are the things that you will obtain first. Now what will you do, right? You are going to grow that E. coli having 15 isotope of nitrogen, right, for 20 minutes in a culture where you have NH4Cl with light isotope. Now, after 20 minutes, when you will take out that E. coli, right, and when you will do this centrifugation and cesium chloride density gradient, right, now obviously what you want to check, you want to get the bands. You have to check, right, after 20 minutes. Why after 20 minutes? Because this is the generation time of E. coli, right? It is the generation time of E. coli. After 20 minutes, you will get 2 daughter DNA molecules. What will you get? 2 daughter DNA molecules. Now, after 20 minutes, when they obtained the E. coli cells and when they obtained the band, it was a hybrid. It was not at, right? It was not at the level of 15N. It was not at the level of 14N. Right, it was not at the level of 15N, it was not at the level of 14N. Matlab they obtained something in between. So obviously they called it hybrid. Now in the same culture medium, they grew the E. coli for another 20 minutes, matlab in totality 40 minutes. So obviously they will get 1 cell, 2 daughter cells. 2 daughter cells will give you 4, because now the source of nitrogen is 14N only. So this thing will be there as such, because these two will open, they will act like the template, complementary to it, new strands will add it, right? So that is what we are going to get. So here there will be 2 hybrids, 2 DNA, daughter DNA having 14N, right? So on the basis of that, we will get the band. So on the basis of that, you can calculate also. Let's say I'm telling you after 80 minutes, right? Or means after next 20 minutes, what will you get? After next 20 minutes, what will be the condition? After next 20 minutes, what will be the condition? Done? Yes? Yes, please. So definitely it proves the semi-conservative mode of DNA replication, because parental strands were 15 and now the source of nitrogen is 14N. So one strand is parental, one is newly synthesized as per the source of nitrogen available. So hybrid is indicating that semi-conservative model of DNA replication is there, right? This is the experimental proof, and something very important, right? Do not miss it, do master it. Okay. Yes, any doubt? So very similar experiment involving use of, here it is radioactive thymidine to detect distribution of newly synthesized DNA in chromosome was performed on VCA faba beans by Taylor and colleagues in 1958. So these experiments prove that DNA in chromosome also replicate semi-conservatively. Any doubt? Any doubt? Yes. Any doubt? Very good, बच्चे. निहाल. चलिए. So next thing is the DNA replication that we need to study. So excited? Excited? Next thing is the DNA replication that we need to study. Next thing is what? It is the DNA replication that we are going to study. देखो, notes I have added. ठीक है? I am here to explain you this topic in the form of a story, be it transcription, translation, or even if it is the genetic code. Where is genetic code? It is there. Okay, so I am going to explain you these things in the form of a story. In the form of what? In the form of a story, I'll be explaining it to you. Okay, everything is given in the NCERT. You have to read it later. If there will be any doubt, yes, I'll take the special class on the platform, right? You can ask your doubts there. But the point here is that please listen to me very carefully. Right, bache? Right, bache? Right, bache? Please listen to me very carefully if you really want to master this particular chapter. And this is one of the most important chapters, right? It is one of the most important chapters. If we have completed molecular basis of inheritance, matlab you say कि मेरा 20% syllabus is done. 20% syllabus is done because principles of inheritance and variation is not that lengthy, right? It is not that lengthy. Okay, plus if you understand MBOI, biotechnology will be easy for you. Okay, because they are also related. So with the completion of this one chapter or the biotechnology part, you have completed the major portion. I always give this example. I don't remember the year actually. I think it is 2020. Yes, it is 2020 or maybe, not in 2020, 2021 or 2, somewhere 2021 or 2, right? You will see that 11 questions were there from the biotechnology. 11 questions means 44 marks. 44 marks. So biotechnology is also a very important unit. Okay, so what's the point that we have here? DNA replication. DNA is the genetic material, and we know that it's very important for a genetic material to replicate. Replication is very, very, very important thing here. Okay, replication is a very, very, very important thing here. Am I right? Am I right? Am I right? Yes or no? Replication is a very, very, very important thing here. Right? So keep it in your mind. When you talk about the DNA replication, I'll associate it with cell cycle. So you know that cell cycle is having interphase. Interphase is having G1, S or G2 phase. So it is the S phase in which DNA replication occurs. Right? In which DNA replication occurs. So if initial amount of DNA, if initial amount of DNA is 2C, then final will become 4C. Then final will become 4C. Right? If initial amount of DNA is 2C, then final will become 4C. That's the point we have. That is the point we have. So it is the interphase. What's that? It is the interphase. It is the S phase. Understood? Understood? So one more thing, again it is the favorite question of your NTA, that when you talk about the S phase, the centriole duplication occurs, right? The centriole duplication occurs there in the S phase. So this is the DNA replication. Now in your syllabus, the DNA replication that you study is of prokaryotes, and to be more precise, it is of E. coli bacteria. It is of E. coli bacteria. The DNA replication is of E. coli there in our syllabus. That is what I am saying. Right, bache? Right, bache? So it is of E. coli. It is of E. coli that we study in our syllabus. Right, bache? So if you talk about the DNA replication in prokaryotes and eukaryotes, is there any difference there? Yes, basic things are same. You have to open the DNA, it will act like template, semi-conservative nature, all that things are same. But enzymes can vary, right? Right. So yes, there are certain differences as well. There are certain differences as well. Right, bache? Right, bache? So prokaryotes, so in our syllabus, majorly we have the DNA replication of the prokaryotes and that too of E. coli. Okay? Now we have to replicate the DNA, and the mode of replication is semi-conservative mode, right? Semi-conservative mode of DNA replication is there. Let's understand about that semi-conservative mode. Okay, let's understand about that semi-conservative mode. Clear, bache? Clear? Chaliye, let's talk about it. Okay, so as I said, understand it like a story. Understand it like a story. Now, even if it is your prokaryote, or even if it is your eukaryote, one thing that you know, that prokaryotes mein you have circular DNA, eukaryotes mein you have linear DNA. Now when you talk about the DNA replication, bache, it will not just start from any point, right? It will not just start from any point, right? Bache, I'll quote one example here. Just allow me 1 minute. Yes, so I'm back because I have to give you one example here. So, DNA replication. So, DNA replication, is it going to start from any point? Of course not. Of course not. There are certain points from where the DNA replication will start, and we use the word replicon for that. We use the word origin of replication. So, origin of replication is a site. It is a site at which replication starts. It is a site at which replication starts. It is a site at which replication starts. So it is the origin of replication, right? What's that? It is the origin of replication, means it is a site at which replication starts. This first point clear? Now, prokaryotes, because they have circular DNA, they have one origin of replication. One origin of replication. So, origin of replication is the site at which the replication starts, and I am saying that prokaryotes, what do they have? They have one origin of replication, and we call it as ORIC site, right? So, there are certain proteins which are involved to recognize it. Okay, okay, so it's a circular DNA. It is having one site at which the replication will start. Okay. My bad. Okay, just let's say this is the site. So I will call it as ORIC. There are certain proteins which will recognize it, and then the enzyme will come for the replication. Now, in the case of eukaryotes, linear DNA is there. Linear DNA is there, yes or no? In the case of eukaryotes, what do we have, students? Linear DNA is there. Linear DNA is there. So when you talk about this, here you will see many origin of replications. There will be many sites. It is not the one place from where the replication will start. There are many sites. So, prokaryotes are what? They are mono, means single. Replicant, single, monoreplicant they are. Monoreplicant they are. Right? They have single origin of replication. They have single origin of replication. Is that clear? They have single origin of replication. Now when you talk about the eukaryotes, what these eukaryotes are? Poly, या फिर you can say multi-replicator. Poly or multi-replicant they are. Understood? Poly or multi-replicant they are. Understood? Understood? Okay, so let's say if it is the circular DNA, equali. So here the origin of replication is, so bidirectional DNA replication will occur. So it is the eukaryotic DNA, right? Here you will see many DNA replication, many origin of replication will be there. Monoreplicant and polyreplicant, thing clear? Monoreplicant and polyreplicant, thing clear? Done. Now DNA is a double-stranded molecule. DNA is what? DNA is a double-stranded molecule. Am I right here? DNA is a double-stranded molecule. What's that? Double-stranded molecule it is. Double-stranded molecule it is. ठीक है जी, 5' to 3', right? Like this. Now, if I want to copy the information given in it, I have to open it. I have to open it, and it's not a very easy task. They are connected together with the hydrogen bonding, and I want to open the DNA because I want, right? I want to use that information to synthesize a new strand. To synthesize a new strand, that is our requirement. That is our requirement. So at the origin of replication, an enzyme will come. Now we have to discuss about the enzymes. Now we have to discuss about the enzymes. So what is going to happen there? Yes, what is going to happen there? Right, you need the enzyme. The very first enzyme that we need to discuss is bache helicases. Right, what is the name of enzyme? It is helicases or unbindase. Helicase or unbindase. What's the name of enzyme? Helicase or unbindase. And it's not the only enzyme that we have. There are certain other proteins or other enzymes involved. So there are enzymes or proteins which will help the helicase to recognize the origin of replication, which like, so obviously in your syllabus, we have no need to discuss the things in that detail. Okay, we have no need to discuss the things in that detail. Right, bache? Right, bache? Okay, so helicase and unbindase is there. Helicase and unbindase is there. So helicase or unbindase, like name is suggesting that this is the enzyme that is going to open the DNA double helix. Right, that this is the enzyme that is going to do what, bache? That is going to open your DNA double helix. So first step is going to be the unzipping, where you have to break these hydrogen bonds. Is that clear? So helicase is the enzyme. Helicase is the enzyme, and magnesium acts as a cofactor here. Done, right? So I want to open this double helix. So this enzyme also need, helicase also need energy from ATP hydrolysis, right? Energy is required to open this DNA double helical structure, and what's that? It is the ATP hydrolysis that will provide the energy. It is the ATP hydrolysis that will provide what? That will provide the energy. Now the point is very simple. Look at this. Look at this. Just imagine. This is your DNA double helix. You are trying to open it. You are trying to open it. When we try to open the DNA, we cannot open it in one go like this. You cannot do this. Yes, ma'am, we are opening it in one go. No, not possible. Not possible at all. Not possible at all. Because you are breaking the hydrogen bonds here. Slowly, 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 this will be done. Very slowly. And when you are doing it, you will get what? You will get a replication fog. You will get a replication fog. Imagine it now. It is like this, right? It is like this. One strand, 5' to 3', 3' to 5', and we are trying to open it. How? With the help of a helicase. So we cannot do it in one go. It will go slow, right? It will go, the opening will be there slowly. So you will get a Y-shaped replication fork here. What will we get here, bache? Y-shaped replication fork here. Am I right? What are we going to get here? Y-shaped replication fork here. Is that clear? Is that clear? Is that clear? Done, bache? Done, bache? Done? Yes, this part clear. Now, try to understand one more thing here. See, again, I'll give you the example. You say, you are talking about, in your syllabus, you have the circular DNA, right? And basically we discuss about that. And even if I don't talk about this circular DNA thing, but obviously it's the part of your syllabus. We are talking about the E. coli. I'm showing you here the circular DNA, and I'm saying bidirectional DNA replication is there. It's not like that firstly your DNA will, your enzymes will copy this particular strand of DNA, right? And then it will reach here, and then it will copy this. No, in both the direction, the synthesis will occur. Right? In both the direction, the synthesis will occur. That's why in the case of E. coli, the generation time is 20 minutes. Otherwise, it can even be 40 minutes. Okay, 42 minutes. Okay, so in both the direction, DNA replication will occur. Just here, if let's say synthesis is going on, here also it will occur. So in both the direction, synthesis is going on. Okay, so imagine it like this. Here you have the origin of replication. Here you have the origin of replication. So this DNA replication is in both the direction. Helicase will come, will start unbinding it. Helicase will come, will start unbinding it. So you are opening your DNA double helix. You are opening your DNA double helix. Then you will get here a replication bubble. What will you get here? You will get here the replication bubble, because DNA replication is going on towards both the sides. Okay, what will you get? Replication bubble. Replication bubble. So if you look at that replication bubble, जैसे ये देखो, this part or this part, it is also a Y-shaped replication fork. It is also a replication fork. So when you are using the word replication bubble here, right? So replication bubble here, if I'm saying, I'm talking about the two replication forks here. What am I talking? I am talking about the two replication forks here, right bache? Right bache? Jैसे if you look at this part, see this, only this half, it is a Y-shaped replication fork. So when there is a replication bubble, it is this complete structure, because bidirectional DNA replication is there. So two replication forks will be there. Any doubt? Any doubt here? Any doubt here? Guys, what do you want? What do you want? What do you want? Do you want me to just run like this and to just, just to give you the feel ki haan ji, your syllabus has been done, or do you actually want me to cover the syllabus? Right? And obviously, if I talk about the detailed section, detailed lectures, in that detailed lectures, everything has been discussed in detail, obviously. That's why it is a detailed lecture, right? Because we emphasize the NCERT there also. We emphasize on everything. We also talk about the new questions that can come. Now it is a marathon. I'm supposed to complete a syllabus somewhere in 10 hours or in 12 hours, right? It's like this. It is like this. But ultimately, what's our motto? What's our point? We need to understand our syllabus. See, if I take instead of, because see, the way I'm explaining, it's not fast, it's slow and steady, but it is explaining everything. But if you want me to continue the same pace, because they see, then I will take three parts of class 12. I will not finish it in 2 goes, zoology, botany, no. I'll keep it class 12th biology, and I will take 3 parts of it. 3 parts of it. Is that okay for all of you? Is that okay for all of you? Because I really don't want to rush. Because 3 parts, right? I'm clear here. 3 parts of your, yes, 3 parts of your class 12, 3 parts of your class 11, total 6 lectures. Syllabus will be over. है ना? And I don't want my sessions to be like this. Yeah, ma'am is just, you know, shh, she's doing shh, 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 shh. She's just doing shh, 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 shh. है ना? She's just coming and she's like this over. This over. No, I really want to explain the things. Okay, so if you are investing your 10 hours with me or 11 hours with me, so they should be beneficial. They should be beneficial. Okay, if I have to stand for 10 hours, I should feel like it. I have done something nice. Whosoever is going to watch this lecture, this lecture is going to benefit that student for the NEET examination as well, for the CBSE board exams as well, right? Okay. Okay, so be sincere, right, and be consistent. So this part is clear, the Y-shaped replication fork thing. This part is clear, the Y-shaped replication fork thing. Let's continue then. Okay, let's continue then. This is what we have, Y-shaped replication fork. Okay, so helicase is the enzyme that we will use. Now, you're trying to open this DNA, right? You're trying to open this DNA. Okay, you are breaking the hydrogen bonds, but again, they are not in their stable condition. They will try to re-anneal. They will try to rejoin, and we do not want that. We do not want that, right? We do not want them to rejoin. We want them to stay separated. Why? Because we have to read that information. We have to add the bases complementary to it, right? We have to add the bases complementary to it. So now comes the role of SSBP. That is your single-stranded binding proteins. Now here, what is going to happen, bache? There will be the role of single-stranded binding proteins. So that single-stranded binding proteins that will join here with the these strands, right? And they will not allow, they will stop re-annealing of separated strands. Am I clear here? Re-annealing of separated strands here. Is that clear? Is that clear? Now, next thing that we need to discuss is topoisomerase. Next enzyme that we need to discuss here is topoisomerase. So it removes the supercoiling. It removes the supercoiling. Right? Now, what is the meaning of that? And it also uses the energy from ATP hydrolysis. Now, bache, if there is a helical structure, you can give it a try. You can even give it a try. Trust me. Acha. I do this a lot with these wires, na? And that's why my last vocom, that's not working. Chalo, koi baat nahi. Ye dekho. It is a helical structure. You are trying to open it, right? You are trying to open it. You are opening it. There comes a time when some coilings will form and they will not allow the smooth opening of that. Ye dekho, some coilings will form. They will not allow the opening of that, right? Because it's a helical structure. You're trying to open it. So some coilings will form. Right, so we have to remove that coiling, right? If let's say you're trying to open that helical structure, at times the coiling will come. So we need the enzymes that can cut the DNA strand, that can remove that coiling, and that can again join that strand, right? So for that we have the topoisomerase. Which enzymes do we have? Topoisomerase. So when you talk about the topoisomerase, there are 2 classes, class 1 topoisomerase, class 2, but you have topoisomerase 1, you have topoisomerase 2. So topoisomerase 1 will make a cut in the single strand of DNA, will rotate that coiling wala part and will then remove the coiling, and topoisomerase 2 will cut both the strands like this. Their main agenda is to remove the supercoiling. Keep it in your mind. They also use the energy from ATP hydrolysis. Now we also discuss the enzyme DNA gyrase. Do you remember that? DNA gyrase. DNA gyrase it is. Gyrase it is. It is also, it removes supercoiling, bache. It removes the supercoiling, right? It removes the supercoiling and it removes positive supercoiling and introduces negative supercoiling. So this DNA gyrase, it is basically a topoisomerase. It is actually nothing, but it is a topoisomerase in prokaryotes. So this DNA gyrase is a class 2, DNA gyrase is a type 2. It's not class 2, it's type 2. Okay. This DNA gyrase is a type 2 topoisomerase which was discovered there in the prokaryotic cell. So in prokaryotes, you will call that topoisomerase as DNA gyrase. Understood? You will call it as DNA gyrase. Understood? Now this is your replication fork. This is the direction of the replication fork. What is the meaning of direction of replication fork? Actually, your helicase is attached here. It is moving in this, it is moving towards this side, and that's how it is slowly opening the DNA helix. That's why I'm saying this is the replication of your DNA fork. Okay? So the role of these enzymes clear? The role of these enzymes clear? The role of these enzymes clear? Okay, so look at the template strand. Look at the template strand. Yeah. So look at the template strand here, 5' to 3'. And here it will be 5' to 3'. So when I look at the template strand here, this is my template strand. What is the polarity of this template strand? See, your replication fork is moving in this direction. Your application fork is moving in this direction. In this direction, your helicase is moving and opening it. So if I talk about the polarity of this template strand, what is the polarity of this template strand? Please tell me. And please tell me about the polarity of this particular template strand as well. Please tell me the polarity of both the template strands here. Yes. Yes, yes, yes. Yes, please. You have the template strands here, right? You have the template strands here, right? This is also a template strand. This is also a template strand. So this template strand is having polarity 3' to 5'. This template strand is having polarity 5' to 3'. Am I right? It is having polarity 5' to 3'. So if it is having the polarity 3' to 5', so synthesis will always be, synthesis always occur in 5' to 3' direction. Synthesis always occur in 5' to 3' direction, right? So if it is 3' to 5', this direction will be the synthesis. This is the direction of synthesis. Then this is the direction of synthesis. This is the direction of synthesis. Now 5' to 3'. Here, if I talk about the synthesis of DNA strand, don't you think this direction, 5' to 3'? That's how the DNA will get synthesized. Is it making sense to you? Yes or no? Is it making sense to you? Yes or no? Yes or no? Yes or no? है ना? So if this is the template strand having polarity 3' to 5', complementary to it when there is a synthesis of new strand, this strand synthesis is in the direction of replication fork. Means your application fork is moving in this direction, your strand is also getting synthesized in this direction. Am I clear here? Am I clear here? Am I clear here? If it is a yes, some fire emojis in the chat section. Now here the direction of synthesis is opposite to that replication fork, away from the replication fork. So again, that's a previous year question. Again, that's a previous year question, that if the template strand is having polarity 3' to 5', complementary to it, the strand will synthesize in 5' to 3' direction. So what is the direction of that synthesis? It is towards the replication fork. And when the template strand is having polarity 5' to 3', so the synthesis will be in the opposite direction. That means away from the replication fork, right? That means away from the replication fork, right? That means away from the replication fork. Is that clear? Is that clear? Is that clear? Is that clear? Okay, chaliye. Now we have to synthesize the strand, right? So now when it comes to the synthesis of the strand, obviously enzymes are involved there. Of course, enzymes are involved there. Let's talk about it. So in prokaryotes, when you talk about that enzymes, right, what are you going to discuss? You will discuss about DNA polymerase, है ना? And when you talk about the DNA polymerase, or it is that main replicase enzyme. So when you talk about the DNA polymerase, right, I will use the short form of DNA polymerase. I'll call it DNAP. Okay, I'm going to use the short form of DNA polymerase, that is DNAP. So we will have DNA polymerase 1, we will have DNA polymerase 2, and we will have DNA polymerase 3. These are the three enzymes that we discuss. These are the three enzymes that we discuss in the case of prokaryotes. So when it is the DNA polymerase 1, we also call it as Kornberg enzyme. We also call it as Kornberg enzyme. This is the scientist who discovered it. And when you talk about this, it is the main replication enzyme. It is actually the replicase DNA polymerase 3. It is actually the main replicating enzymes. It is actually the main replicating enzymes. Are you getting my point? Right? So it is the DNA polymerase that is required for the synthesis of strand, but it will polymerize these nucleotides, right? And that too in 5' to 3' direction. So you have DNA polymerase 1, 2 and 3 here, right? So it is the 3 that is the main replicating enzyme. Now, other two are also playing the role. Just DNA 2 is having a very major role in DNA repair, right, bache? So they are involved in DNA repair, right? Proofreading mein. They say this one is also, so basically these enzymes are having their role in proofreading and DNA repair. So DNA polymerase 2 is very important for the DNA repair. So now I'll come to this part also. Dekho, we are going to discuss two things. One is exonuclease activity here. Right, what are we going to discuss here, bache? We will be discussing the exonuclease activity and polymerase activity. We will be discussing exonuclease activity and polymerase activity. What are we going to discuss here, kids? What are we going to discuss here, kids? We will talk about the exonuclease activity and we will talk about the polymerase activity here, right? Exonuclease activity and polymerase activity. These are the two words that we have. Okay? Exonuclease activity or polymerase activity. Exonuclease activity and polymerase activity. Exonuclease activity and polymerase activity. Try to define it. Try to define it. Yes, please. Yes. Right. So exo means ends, nuclease that will digest nucleic acid. But when I'm saying exonuclease activity, just for an example, let's say this is your DNA. This is your DNA. So it will start acting on this DNA from the ends, right? At the ends it will act basically, and exonuclease activity means it will remove the bases from the ends. Polymerase activity, to polymerize, it will add the nucleotides, and it will form this chain. it is going to form this chain. That is the meaning of exonuclease activity. ठीक है बच्चे, that is the meaning of exonuclease activity. Is that clear? Sure. That is the meaning of exonuclease activity. Sure. Okay. So now when you talk about these enzymes, see, all of them are having polymerase activity. All can polymerize the DNA strand. These three, out of these three, all can polymerize the DNA strand. All can add bases the 5' to 3' direction. But the point is, why do we say it is the DNA polymerase 3 which is the main replicating enzyme? Because, bache, see, it is the fidelity is the word here, but let's to simplify it, because we just do not want to add the bases. We have to add the bases quickly, plus accuracy is important. Matlab here we need to focus on speed and accuracy. We need to focus on speed and accuracy. We need to focus on speed and accuracy. Are you getting my point? Are you getting my point? We need to focus on what? We need to focus on speed as well as accuracy. Speed as well as accuracy. These are the two things that we need to focus. These are the two things that we need to focus. Right, speed as well as accuracy. So for this, DNA polymerase 3 is the perfect enzyme because it is fast and accurate as well. It is fast and accurate as well. That's the point. That's the point. Okay, right. So now when I'm talking about the exonuclease activity, it is also important. Just say, let's say, if you have an enzyme that is having 5' to 3' exonuclease activity, what is the meaning of this particular line? That this enzyme is having 5' to 3' exonuclease activity. It means this enzyme will move from 5' end to 3' end, and it will remove the bases. If they are not appropriately attached, bache, it will remove the bases. That is the meaning here. That is the meaning here. Right, bache? So out of all these three enzymes, all are having 5' to 3' polymerase activity. All are having 5' to 3' exonuclease activity. But when you talk about the DNA polymerase 1, it is having one unique property. Right? It is having one unique activity. And that unique activity is 3' to 5'. One second. Yes. One second. Yes, so I am talking about the exonuclease activity. So I told you about 5' to 3' exonuclease activity. So 5' to 3' exonuclease activity means 5' to 3' exonuclease. Here I need a correction. Listen to me carefully. Here I need a correction. Listen to me carefully. So 5' to 3' exonuclease activity is the word, means that enzyme will move from 5' towards the 3' end. Right? It will remove the bases if they are not correctly attached. That's the point. Now the another word is 3' to 5' exonuclease activity. 3' to 5' exonuclease activity. What is the word here? 3' to 5'. It means this particular enzyme will move from 3' to 5' and it will remove the bases if they are not properly attached. It will remove the bases if they are not properly attached. That's the point. Now, when it comes to 3' to 5' exonuclease activity, bache, this is a common activity. All the three enzymes are having this. All the three enzymes are having this. Is that clear? Is that clear? Is that clear? All the three enzymes are having it. You talk about any of the enzyme here. All the three enzymes, yes, bache, all the three enzymes are having this activity. Okay, they have to move from 3' to 5'. Right? They have to move from 3' to 5' and they need to remove the bases if they are not appropriately attached. Just say this enzyme is also having this activity, this enzyme is also having this activity, this enzyme is also having this activity, and plus 5, because synthesis is always in 5' to 3' direction, and all three are having 5' to 3' polymerase activity. But the point is we go for the DNA polymerase 3 because it is fast and accurate. But the most important thing here is a unique property. Initially I just mixed it. So there is a unique activity in DNA polymerase 1. It's a previous year question. Please pay attention. It's a unique activity of DNA polymerase 1. There is one unique activity of DNA polymerase 1, that is 5' to 3 dash exonuclease activity. 5 dash to 3 dash exonuclease activity. It's a unique property here. It's a unique property here. And this property is important for primer removal, and then because it is having the polymerase activity also, so it will help in primer removal and gap filling. Primer removal and gap filling. This is something I will explain. This is something I will explain, but this is important, right? Only in DNA polymerase 1 you will see 5 dash to 3 dash exonuclease activity. Try to understand it in this way. Try to understand it in this way. देखो, जैसे you are synthesizing a strand in this direction, 5 dash to 3 dash, right? जैसे let's say you are arranging something from A point to B point. Now you have to verify it. You are moving back from B to A. You are moving and you are also checking कि whatever I need to place, am I placing it correctly or not? Am I placing it correctly or not? ठीक है? मतलब it's like कि you are, let's say you have to arrange some books. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Now you have to verify that whatever you have arranged, is it fine or not? So you'll move like this 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. That's how the things are going to be. That's how the things are going to be. That's how the things are going to be. That's how the things will move. Right, bache? Right, bache? That's how the things will move. So same is the case here. The strand will get synthesized in 5' to 3' direction. So one enzyme will move from 3' to 5' direction, and it will check if there will be any incorrect basis, it will remove. Now when I'm saying 5 dash to 3 dash exonuclease activity, that is unique, and I will explain its role. Okay, so I told you one thing very clearly, that it is the DNA polymerase 3, which is the main replicating enzyme. Am I correct here? I told you clearly that it is the main replicating enzyme. Which one is the main replicating enzyme, guys? Which one is the main replicating enzyme? Yes, which one is the main replicating enzyme? Your DNA polymerase 3. But again, it is having a drawback. It is having the limitation. Jaise for an example, let's say there is a head chef. Right, there is a head chef. That head chef is like, okay, I have to cook something. So head chef is also having the assistants. So head chef is like, okay, you chop this thing, you bring me this, you bring me that, then I will arrange everything. Same is the case here with the DNA polymerase 3. Same is the case here with the DNA polymerase 3. What this DNA polymerase 3 is going to do? This DNA polymerase 3 is not that easy enzymes, right? It is a high maintenance enzyme. This enzyme is like, oh please, I cannot just come and I cannot just start the synthesis of the DNA. I cannot just start the synthesis of the DNA. I need 3-OH group to start it. It needs, for its activity, it needs 3-OH group, right? Just for an example, some students, yeah, let's say if you talk about any type of movement, it's not like that. Just let's say you are also like, array, whatever our government is doing, nah, it's not appropriate. We have to fight back. We have to stand. But you are also feeling the same. But you cannot take that stand, right? You're like, theek hai yaar, we should do this, we should do that. Now let's say one day I have decided that I am that revolutionary person. I'll start fighting. You will also join. Okay. Right? You have started this fight. We are also there to support you. It happens that some people, they cannot initiate the things by their own, but if you have initiated it, they can continue. If you have initiated it, they can continue. Same as the point here. Literally same as the point here. You have the DNA polymerase 3. This DNA polymerase 3 is having the polymerase activity. It is the main enzyme here. It is the important enzyme here. Right, bache? Right. It is the important enzyme here. But the point here is that it cannot start the synthesis by its own. It is having the 3-OH group. It needs a 3-OH group. It needs a free hydroxyl group to start it. Right. So just to fulfill this thing, we have primase. Which is also known as DNA dependent RNA polymerase, which is also known as DNA dependent RNA polymerase, which is also known as DNA dependent DNA dependent RNA polymerase. DNA dependent means it will use DNA as a template, and complementary to it, it will form primers, RNA primers. So what are that primers? That primers are 15 to 20, right, short nucleotides that will be added here, added here, added here, added here. Now let's say if they are added, primase has formed it, right? Even if they are the RNA primers, it will have free 3-OH group. Definitely it will have free 3-OH group. And now your DNA polymerase will be like, wow, primer is there. Wow, this is the place from where I have to start. Wow, I have the free 3-OH group. Chaliye, let's continue. Let's continue. Let's continue. Right, let's continue. So that's how the synthesis will go on. That's how the synthesis will go on. But this synthesis, right, that is how the synthesis will be. Now you got it, the role of DNA polymerase 3, the role of primase here. But the point here is, this DNA replication is kind of bidirectional. No? It is not like that, ki firstly polymerase will be like, shoo, let's go, let's synthesize the DNA. Then let's come back, then let's synthesize the DNA. No, it's a big no. It's a big no. It's a big no. What is it? It is a big no. Yes, bache. It is a big no. Yes or no? Yes or no? Beta, it is, Latika, it is DNA dependent RNA polymerase because it is a RNA primer. Okay? 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 So, what is this? What is this? Primase is required. So primase will form RNA primer. Your DNA polymerase 3 will be like, I'm happy, chalo, I got the 3-OH group, I'll add the further bases. But it will not come back, right? It will strand, it will synthesize the other strand also parallelly. Now imagine, this is your DNA polymerase. Imagine, this is your DNA polymerase. It has to synthesize the strand. One strand is in this direction, obviously, but other strand has to be in the opposite direction. Other strand has to be in the opposite direction. Because, see na, synthesis always is in 5' to 3' direction. So here, the synthesis has to be like this. Has to be like this, 5' to 3'. Hai na? So now, imagine how difficult it is for DNA polymerase to synthesize. DNA polymerase is moving like this. DNA polymerase, your helicase, they move in the direction of replication fork. Your helicase is opening DNA like this, your DNA polymerase 3 is also moving like this. Now, it's easy for this DNA polymerase 3 to synthesize the strand in this direction, right? Because this is the direction of replication fork also. So it is easy for it to move like this, like this. Okay, it is easy for it to move like this. It's literally easy for it to do this. Now it has to synthesize the another strand parallelly, right? One strand, it can do it if it has to synthesize both the strands in same direction. Now it's like, it has to synthesize one strand in this direction and other strand is in this direction. Don't you think it's a difficult task? Don't you think it is a difficult task? So the structure of DNA polymerase is in such a way that here though very smoothly it is going, continuously it is synthesizing the strand. But for other strand having polarity 5' to 3', its structure is in such a way that it will coil that DNA. Then it will take a part of it, then it will synthesize, right? Then it will synthesize. So towards this side, the synthesis will be discontinuous. The synthesis will be discontinuous. Like here, the first strand will synthesize, the next strand, like here, the discontinuous synthesis is there. What is there? The discontinuous synthesis is there again and again, because DNA polymerase is like, yeah, I am effectively, if I have to synthesize both the strand in this direction, I am okay. But now its problem is that for one strand it has to go like this, and for other strand it has to be. It's difficult for this enzyme. It's difficult for this enzyme. So it will coil this DNA in such a way that it will get this stretch. So here, multiple times, the connection in between this template strand and DNA polymerase 3 breaks again and again. Here, it is the requirement of primer to form, right? Again and again, there is the requirement of primer. Again and again, primase will come, primer will form. So So the another famous question here is that when we talk about the template strand having polarity 3' to 5', right, complementary to it, now you know the reason also, leading strand forms, right, complementary to it, leading strand forms. So leading strand means it is synthesizing continuously, right, continuous strand, leading strand, continuous strand. Now template strand is having polarity 5' to 3', so complementary to it, lagging strand will form. Lagging strand means it is a discontinuous strand. Its synthesis is discontinuous. It is not continuously synthesized. So here the question will come from the polarity, that if the template strand is having polarity 5' to 3', complementary to it, leading strand will form or lagging. It is the lagging strand that will form. It is the lagging strand that will form. It is the lagging strand that will form. Right? So note down. Please note down. Yes, please. Yes, please. Understood? Right. So number 1, number 2, number 3, like this is how it will form. Okay. So it is the leading strand, it is the lagging strand. So because here there are fragments, so scientist who discovered it was Okazaki. So that's why they are also known as Okazaki fragments. Right. That's why it is also known as Okazaki fragment. That is why it is also known as Okazaki fragment. Okazaki fragments. Understood? Now see, here you have these primers now. You have these primers now. These primers in between DNA. Don't you think it's important to remove these primers? Don't you think it's important to remove these primers? है ना? It is important. It is important. So for primer removal, which enzyme do you have? So for primer removal and even the gap filling, because these gaps need to be filled as well. So for primer removal, you know that 5' to 3' exonuclease activity is required, right? For primer removal, 5' to 3' exonuclease activity is required, which is there in your DNA polymerase 1. So DNA polymerase 1 with its 5' to 3' exonuclease activity, it will remove the primers here. It will remove the primers here. So don't you think this gap needs to be filled? This gap present in between needs to be filled, right? So for that gap filling, even your DNA polymerase 1, it is having that 5' to 3' DNA polymerase activity na. It is having that 5' to 3' DNA polymerase activity na. So that DNA polymerase activity is going to play the role here, right? So here the primer will be removed, the DNA bases will be added here, the primer will be removed, and then the DNA bases will be added here, bache. They will be added here, and finally here the DNA ligase, which is also the molecular glue, it joins sugar phosphate backbone here. It joins sugar phosphate backbone here. It joins sugar phosphate backbone here. Understood? Understood? It joins sugar phosphate backbone here. That's how it works. Clear? That's how it works. That's how it works. Clear? This is all about the DNA replication. So look at the enzymes here. Look at the diagrams here. Single-stranded binding proteins are attached. Helicase is moving. This is the direction of replication for DNA polymerase 3 will also move like this. But here for the synthesis of, here for the synthesis of this strand, it has to form the strand in opposite direction. So that is why here the lagging strand will form. And topoisomerase, it removes the supercoiling. So DNA gyrase is the first topoisomerase discovered in prokaryotes. It is also type 2 topoisomerase. So wo dekho, DNA polymerase 3, how the strand is getting synthesized. And look at the direction of helicase. So helicase is moving towards the direction, helicase is opening the DNA helix like this. So see the leading strand, same direction. But lagging strand is in opposite direction. Understood? Understood? Hanji, understood? Yes, please. Yes, please. Okay, so look at the role of the enzymes here. Recognition of origin of replication, DNA protein, but again not required for you. Unbinding of DNA helicase, stabilizing single-stranded binding protein. In both the cases, primase is required. Now this point is very important. Here you have the DNA polymerase 3 as main enzyme, but in the case of eukaryotes, you have different types, right? You do not have in eukaryotes, bache, you do not have DNA polymerase 1, 2 and 3. I told you na they are different. So DNA, when you talk about the eukaryotes, do you talk about DNA polymerase alpha, beta, delta, epsilon, like this? These are the different types of polymerases which are there in eukaryotes. So in eukaryotes, you do not have DNA polymerase 1, 2 or 3, right? See, you have DNA polymerase delta for this leading strand, DNA polymerase alpha for this lagging strand. So in eukaryotes, that's how it works. Okay? So removal of RNA primer, obviously DNA polymerase 1, 5' to 3' exonuclease activity, right? And then replacement of RNA with DNA, again DNA polymerase 1. Joining of Okazaki fragments, DNA ligase. And ligase also need NAD, nicotine amide adenine dinucleotide. So removal of that supercoils, DNA gyrase or DNA topoisomerase 2. That's the point. Understood? Hanji, clear? Clear? So this is all about the enzymes. So notes will definitely be shared. Okay. So here it is written about their exonuclease activity also. So you people are going to get proper notes. Don't worry about it. So ye dekho, leading strand, lagging strand. Yes. Observe the diagrams. If there is any doubt, please ask. Please feel free to ask. Done, bache? Okay. Okay, so again, the enzymes, and this is the diagram from NCERT. You know na? This diagram is from NCERT. Yes. So one strand will synthesize continuously, and here the template strand is having polarity 3' to 5'. Discontinuous synthesis, template strand is having polarity 5' to 3'. Keep it in your mind and see the role of enzymes here. Role of enzymes here, very important it is. So you can use or you can make such type of, yes bache, you can make such type of flow charts, right, for your quick revision. Understood? For your quick revision. So see, this is the replication bubble. We are focused on one side, the leading strand synthesis, and that's how your lagging strand will form. Number 1 at the end, 2, 3, right? This question was there in CSIR net once, a very basic question. So look at the way it is getting synthesized. Done. So next part that we need to understand is transcription. Any doubt here? Next part that we have to understand is transcription. Any doubt here? Sorry. Any doubt here? So do complete it, do revise it. Bache, it is that DNA dependent RNA polymerase, your primase. Right? And if you want to use DNA dependent DNA polymerase, what you can use it for your DNA polymerases? You can use it for your DNA polymerases. Understood? Understood? Yes? Understood? So revise it. Take 5 minutes and revise it. Yes, take 5 minutes and revise it. So 5 minute ka break you can save, though I am here, right? So revise it quickly. Do it. Do it, do it, do it. No question is there related to the topic, है ना? Bache, I have already explained the NCERT, right? Whatever is written in NCERT, it has all been done. Revision sorted? Topic is easy, right? Topic is easy, right? Chaliye. Next is that transcription, because we know now we have to make the, yes, now we have to do what? Now we have to make the mRNA from that DNA. Right? Now we have to do what? We have to make the mRNA from the DNA. Now we need to understand something. This is the process of copying the genetic information from one strand of DNA into RNA. Now, in the definition itself, they have specified one strand of DNA. This is important. Right? Please pay attention here. This is important. We have to do what? We have to copy the information. What we have to do? We have to copy the genetic information, and that too from one strand of DNA into the RNA, and this is what transcription is. So both the strands of DNA will not be copied. It is the one strand of DNA that will be copied into the RNA. So obviously for the synthesis of this RNA, principle of complementarity will help here. How is it possible? See, it's very simple. In RNA, at place of T, we have U. Rest everything is same. It's simple. Dekho, if in the DNA you have A, T, G, C, T, A, C, G. So, if from this strand you have to form the mRNA, so complementary to A, it should be U. At the place of thymine, now you have to put uracil. At the place of thymine, so complementary to thymine, adenine. Complementary to guanine, cytosine. Complementary to cytosine, guanine. Complementary to thymine, adenine. Complementary to adenine, uracil. Complementary to cytosine. That's how it is. So, your mRNA will have this base. Right? This will be the sequence on the mRNA. So rule of, right, that principle of complementarity is also functional here. The principle of complementarity is also functional here. Understood? Understood? It is also functional here. Right, bache? Except the adenosine complements now with uracil instead of thymine. This part clear? This part clear? Right, bache? Right. So in the DNA replication, the entire DNA will be duplicated, but here it will not be like that. Only a segment of DNA and only the strand, and only one of the strand is copied into the RNA. So this necessitates defining the boundaries that would demarcate the region and the strand of DNA that would be transcribed. So when we talk about it, right? So let's discuss it first. Okay, let's discuss it first. Dekho bache, it's very simple, right? I'll give you the example here. Okay, please pay attention. This is C. This is your one DNA strand. This is your another DNA strand. One DNA strand is having polarity 3' to 5', and another is having polarity 5' to 3'. That's how it is. That's how it is. Yes, please. That's how it is. One is 3' to 5', another is 5' to 3'. That's how it is. Just imagine. Let's say these are the bases that are present on your DNA. These are the bases that are present on your DNA. Please look at it. If here it is adenine, what will be there? What will be there on another strand? What will be there on another strand? Write down the bases in the chat section quick. A complementary to A, T, G, C, A, T, T, C, C, G, T, A or G. That's what we have. I'm talking about a stretch of DNA. Now I have to form the mRNA, right? mRNA, M stands for messenger RNA. M stands for messenger RNA. Yes or no? M stands for what? M stands for messenger RNA. And let me tell you, this messenger RNA is the least stable RNA. Right? Here itself, let me make it clear, because we have to discuss tRNA and rRNA also. So what am I talking? I'm talking about the mRNA. What am I talking? I'm talking about the mRNA. M stands for messenger and it is the least stable RNA. It is the least stable RNA. What's that? It is the least stable RNA, and it forms 1 to 5% of total cells RNA, right? 1 to 5% only. And you know that when we copy the information from DNA, the mRNA forms. Now, let's say I have to make the mRNA. Synthesis will always be in 5' to D' direction. Let's say I'm giving you this. You have to form mRNA complementary to this strand as well as this strand. Tell me what will be there on the mRNA. Right, this is the DNA strand that will be used as a template. You have to form the mRNA. This is the DNA strand that will be used as the template. You have to form the mRNA. Then please tell me, please write down the number of bases. Answer? Very good. So here it will be UGCA, UA or G. Am I right? Am I right? Now look at this part. T complementary to A, C, G, U, A, G, C, A, U or C. That's what we have. That is what we have. Now when you look at it, right, if let's say both the strands of DNA, if they will be involved, if both the strands of DNA, they will be involved in this RNA formation. So people, you will see that, see, this mRNA and this mRNA, they are complementary to each other. A and U, C and G, G and C, U and A and U, they are complementary to each other. If both the strands of DNA will start making the mRNA, then that mRNA will also be complementary to each other. You will get double-stranded RNA. Then don't you think the translation process will be affected? Because in translation, we have to decode this mRNA and we have to add the amino acid so that we can form the protein. Isn't it? So that we can form the protein. Isn't it? Isn't it? So both the strands of RNA here, right, they will be complementary to each other. They will form the double-stranded RNA. Again, not possible. Again, not possible. Right? Again, not possible. Right, bache? Right, bache? Right, bache? Done. Moreover, you know that when we read a mRNA, Actually, your ribosomes will read mRNA. They will read it in 5' to 3' direction. Then they'll take the codons, right? These three nitrogenous bases, they form the codon. We discuss it in genetic code. These three nitrogenous bases, they form a single codon. That's why we have triplet base codon. That's why we have triplet base codon, and it will give us one amino acid. It will give us what? It will give us one amino acid, right? It will give us what? It will give us one amino acid, right, bache? So if both the strands will be copied, we will get the information for different sequence of amino acid, different proteins will be formed there. Again, that is of no use for us. So that is why only one strand of DNA, be copied, and that strand of DNA will be your template strand. Right, it will be your template strand. So which strand will be your template strand? Having polarity 3' to 5'. This strand is your template strand, right? So complementary to it, this other strand is what? It is known as coding strand. It is known as coding strand. What do we call it, people? We call it as coding strand. So what's the word that you have? You have template strand, right? Template strand is also known as antisense strand. It is also known as non-coding strand. It is also known as minus strand. And on the other hand, if it is known as the coding strand, right, it is also known as non-template strand. It is also known as non-template strand. It is also known as sense strand or positive strand. It is. Right, bache? Right, bache? That's how it is. So which strand is the template strand? Again, a PYQ in transcription. Which strand of DNA is the template strand? Having polarity 3' to 5'. So this one is having polarity 3' to 5'. It is acting like a template. Or obviously it is 3' to 5'. Complementary to it, the mRNA will be 5' to 3'. And one more thing that you need to focus here, right, that why the other strand is known as coding strand. If you don't understand the concept, maybe in paper you will be like, Yare, they are asking about the strand which will be copied, right? So maybe this coding strand is getting copied, but actually it is not the case. It is not the case. So this is the template strand, this is the coding strand. It is very simple, bache. Look at this part, UGC, AUU, CCG, UAG. Here, if you compare it with the coding strand, they have same sequence, except at the place of thymine in the mRNA, uracil is there. Am I correct here? Am I correct here? Done? Yes, so whatever is the sequence on mRNA, that will be same like coding strand, only at the place of thymine there will be the uracil, right? mRNA will have uracil and the DNA will have thymine. That's why it is the coding strand. So this is the important thing. Now, when it comes to the transcription, right? I know we have to speed up, but I think these two topics and even the translation one, it takes time. Bagi HGP completely theoretic, right? That genetic code is easy and simple, and even that DNA fingerprinting and operon also, it's not going to take much time. And for the operon and genetic code, the separate videos are there on this channel, right? You can understand it from there. Okay, bache? Okay, chali. So now, the transcription, is it going to start from any part? Of course not. Of course not. Right. So, transcription will start from and transcription unit. there will be a transcription unit. So now when you talk about that transcription unit, it will contain a promoter. There is a region, we are calling it as promoter. Ultimately, it is on the DNA only. There is a region that is also known as terminator, right? There is a region that is also known as terminator. Promoter is there, terminator is there. And the part here is what? The part here is structural gene. The part here is what? It is the structural gene. And here this structural gene is having this as template strand, and this one is the coding strand, right? So, bache, promoter to promote. It will promote a transcription, terminator, it will stop transcription. So this is the template transcription unit having promoter, having terminator, in between structural gene is there. In between structural gene is there. Now, what is the next part? Dekho, in the paper when it comes to the question, they can ask you about the position. Dekho, understand one thing. Let's say it is a 0. Yeah, you can say it is a start point. But if it is a start point, in the NCERT also words like upstream and downstream, it is given. Words like upstream and downstream, it is given. So this is the start point. If you are talking about this direction, it means upstream. If you are talking about this direction, it means downstream. Right, this is the upstream part here, this is the downstream part here. This is the upstream part here, this is the downstream part here. Right, this is the upstream part here, this is the downstream part here. Right, bache? So upstream is not in the context of 5- means it is the bigger one, it is the upstream. That is incorrect. That is incorrect. This is a reference point, start point. If we are moving right towards this side upstream, towards this side downstream. In the case of transcription also, let's say when you talk about mRNA synthesis, somewhere here there will be the start point. So can I say that upstream to start point, promoter is there? Upstream to start point, promoter is there. Upstream, bache, who told you it is related to left side or right side? I'm simply saying you keep this start point thing in your mind, and that's how you consider it. Maybe your left and my left, it can be different, na? It depends what are we facing. So if this is the start point, so the promoter is present upstream to the start point or downstream to the start point. Upstream to the start point, yeah, downstream to the start point. The promoter is present upstream or downstream. Will you please answer? Will you please answer? Is it present upstream or downstream? Of course, it is present upstream to the promoter if this is the start point. Right? Promoter that will promote the transcription. That's the point. Now, bache, we have to see, even in NCERT, this line is given that because it is the coding strand that is not used as a template strand. So whenever we talk about the reference point, now we generally take coding strand. Let's say if I'm taking the reference of coding strand, I'm specifying this with the reference of coding strand. Promoter is present at 5- or is it present at 3- end? Tell me. So I'm back with my teacup. So you tell me. No, it's not a break. It's not a break. Chill, chill. I'm here only. Answer it to me. It is at, you are taking the reference of coding strand, it is at 5' end. So let's say if I take the reference of template strand, your promoter is at 3' end then. So keep it in your mind. Keep it in your mind while answering the question. Read it carefully that what are they mentioning. Are they taking the reference of template strand or are they taking the reference of coding strand? So if they are taking the reference of coding strand, it will be promoter will be at 5' end and terminator is at, and the terminator is at, and the terminator is at, chaliye. Exactly. So now let's talk about the gene. What is gene? Gene is a unit of inheritance. What is gene? It's a unit of inheritance. It's a unit of inheritance. So if I have to define a gene, can I define it? Can I define it in terms of, DNA segment only? No. Because when you talk about the DNA segment, right bache, when you talk about the DNA segment or DNA sequence, right, the DNA sequence, matlab sometimes when we define the gene is the unit of inheritance, we know it. So we cannot only say that gene is that part of DNA which is giving us the protein, DNA sequence which is giving us the protein. Matlab gene is what? Unit of inheritance. So if we want to define it in terms of DNA sequence only, it will be difficult because DNA sequence is also coding for tRNA, also coding for rRNA, plus DNA sequence can be used, it can have origin of replication from where the replication can start. It can be there in the mutation thing also. It may be involved in that mutation part, in replication part, in crossing over part. Different things are there. Different things are there. Right? Guys, I don't know whether you know it or not. Right? Initially, a theory was a hypothesis was there. One gene, one protein hypothesis. What was there? One gene, one protein hypothesis. What was there? One gene, one protein hypothesis. That one gene, it gives one protein. But actually, it is not true. It is not true. Right? I'll come to this context later. So, one gene, one polypeptide. Actually, it is the widely accepted one. It is the widely accepted one. It is the widely accepted one. See, one gene gives one polypeptide. Is that clear? One gene gives one polypeptide. Is that clear? Is that clear? So gene is the unit of inheritance. Gene is the unit of inheritance. Yes. Mr. Yush Tuber, there are other lectures also in which in detail I have discussed everything. So you can watch that also if you are so concerned about your syllabus. Okay? So please focus here. So what is there? One gene, one polypeptide. One gene will give you one polypeptide because sometimes a protein is made up of more than one polypeptide. That's why this is the thing that we believe. Now, I am saying that gene is what? Gene is a unit of inheritance. That's what I'm saying. Gene is a unit of inheritance. And we cannot define it in terms of DNA sequence because it can code for tRNA, RNA. Right, bache? Right, bache? Right? Right? But when it comes to the, it can code for RNA, it can code for tRNA. So when it comes to the sequence of DNA, which is giving us the protein, जैसे we cannot say gene is something which is, gene is the one which is giving us the mRNA only, या protein only, right? या protein only. It can also give you rRNA. Are you getting my point or not? Are you getting my point or not? Basically, what am I trying to say? I'm trying to say a very simple thing. If I'm talking about the sequence of DNA, which is giving me what? Which is giving me the information to make protein, which is giving me the information to make polypeptide, I will use the word cistron for it. What is the word that I will use for it? Cistron. In simple terms, if I have to define it, what will I say? I'll use the word cistron here. Are you getting my point? Which word am I going to use here? Which word am I going to use here? I will use the word cistron here. Understood? Understood? Yes, please. Yes, please. Cistron is what? Cistron is what? Right? So it's a part of DNA that is going to code for a specific polypeptide. Right, bache? It is coding for a specific polypeptide. This is the cistron. So you cannot simply say that gene is something, one gene, one protein concept. Yeah, gene is giving us the sequence of DNA is giving us the protein only. No, they are involved in further other things also. But when I'm using the word cistron, it is that part of DNA which is giving me the information for the polypeptide synthesis. Yeah, it is telling me about that polypeptide. Now here you have two words. One is monocistronic and another one is polycistronic. One is monocistronic, another one is polycistronic. But when it is the monocistronic, single cistron poly, देखो, polycistronic, P for poly, P for prokaryotes. And monocistronic, it is there in the case of eukaryotes. It is there in the case of eukaryotes. What do we have here? We have two words. What do we have here? We have two words. What do we have here? We have two words. I'm writing it here. Yes, I am writing it here. It's simple. Monocistronic and polyreplicant. These two things are there in a eukaryotic cell. These two things are there in a eukaryotic cell. Then polycistronic and monoreplicant, single replicant, but they are polycistronic. Where will you read it? You will read it in prokaryotes. Any doubt here? You will read it in prokaryotes. Any doubt here? Yes, please tell me. Yes, please tell me. Any doubt here? Yes? Sure? Sure? So monocistronic and polycistronic is the thing. So basically monocistronic mRNA, mono means single. So it will give me the information to synthesize a single polypeptide. But if it is a polycistronic, it will give me the polycistronic mRNA will give me the information to synthesize many polypeptide chains. Is that clear? That you will see in the case of prokaryotes, right? That we will also discuss when we will discuss about the operon concept. Is it clear? Because this part is also important, people. This part is also important. Is it clear? Is it clear? Are you sure? Are you sure? Okay, so let's discuss this process of transcription. I told you about the structural gene also, and even in the even let's mark the NCERT also. So here in the NCERT, you guys can see both the strands are not copied. It is explained, right? It is already explained. You can even read the NCERT, right? You will see that everything from the NCERT is covered. So transcription unit is having these three things, and this is the diagram. Promoter, this is the transcription start side. So it is somewhere there in promoter, like upstream promoter is present, right? This is the terminator. So 5' so template strand having polarity 3' to 5'. Template strand having polarity 3' to 5' is the template strand, and 5' to 3' is the coding strand. Any doubt here? Yes, any doubt here? Please have a look. Yes, do you have any doubt? Do you have any doubt here? Great. So you have the transcription unit. So here also, if you read it from NCERT, right? I'm covering the NCERT part as well, students. It's not like that ki NCERT will not be covered. You can just see this, right, bache, that there is a convention in defining the two strands, since because they have the opposite polarity. So a DNA dependent, again here this RNA polymerase is also DNA dependent polymerase, RNA polymerase. So that will help in the catalysis of 5' to 3', catalysis of mRNA in 5' to 3' direction. Okay, bache? Okay. Yes. Chalo, so this part is also clear. So, okay, so see, monocystronic mostly in eukaryotes. So if it is mostly written, you cannot say that always. Mostly in eukaryotes, or polycystronic is mostly in bacteria or prokaryotes. Is that clear? So DNA, gene is defined as the functional unit of inheritance. The DNA sequence is coding for tRNA or RNA. It can also define a gene. We can also define gene like that. However, by defining a cistron, when I'm using the word cistron, so we are saying that it is a segment of DNA coding for a polypeptide only. Is that clear? Is that clear? And structural gene can be monocystronic or polycystronic. Monocystronic means if it has given us the mRNA, that is giving the information for only one polypeptide. If it has given us the mRNA, that is giving us the information for the synthesis of, for polycystronic, right? mRNA means it will give us the information for the synthesis of many polypeptides. So that is the point here. So now we have to discuss this transcription thing in detail, right? We have to discuss this transcription thing in detail. So are you ready for that? Yes. Are you ready for that? Right? So obviously there are certain differences in the prokaryotes and even in the eukaryotes here, right? There are certain differences in the prokaryotes as well as in the eukaryotes here. Okay, so is that okay, or do you need a break? Right, again the break will be for 10 minutes only, not more than 10 minutes. You will get a 20 minutes ka break for the dinner only. So do you need a break or is it fine? Chaliye, no worries, it's 4:35. This time I will be punctual. I will be punctual and I mean it. Okay, I mean it. This time I'll be punctual. Hello. Okay, so guys, let's continue this transcription thing, and that too in the case of prokaryotes first. See, it's very simple. Just pay attention, okay? So you are talking about a prokaryotic cell, and this prokaryotic cell is having nucleoid. So basically when it comes to the site of transcription in the case of prokaryotes, here I am just being specific. As I said, I am just being specific here, right? What am I doing, guys? I am just being specific here. I am saying prokaryotes. Site will be nucleus. Why? Because they don't have the, the site will be cytoplasm. Why? Because they don't have the nucleus. Okay, so what is the site that they do have? It is the cytoplasm. It is the cytoplasm. Is that clear? It is the cytoplasm. Is that clear? Is that clear? Okay, so now when we talk about this transcription, obviously it is the transcription unit which is required. Yes, bache, it is the transcription unit that is required. Yes or no? Yes or no? So one thing that you need to understand is that be it transcription, translation, the way, how are we going to study it? It's simple. There are 3 steps. Overall, there are 3 steps. Number 1 is your initiation. Number 2 is your elongation. And number 3 is your termination. Number 3 is your termination. These are the 3 steps here. That's to start something, and then the elongation of that mRNA, and then its termination. These are the 3 things that we have. And if I talk about the enzyme used there, if I talk about the enzyme used there, yes or no? If I talk about the enzyme used there, right, then what am I going to say, bache? It is RNA polymerase. It is RNA polymerase. What is the name of the enzyme that will be used here? It is RNA polymerase. Is that clear? Is that clear? Is that clear? Chaliye. So now this is your DNA. If you want to copy the information, ultimately you have to open the DNA. That's a fact. Ultimately, we have to open the DNA. It's a fact, right? You cannot form your mRNA. You cannot just form your mRNA when the DNA is just helically bound. Of course not. Of course not. Right, bache? So obviously, we need to open the DNA helix. We need to open the DNA helix, yes or no? We need to open the DNA helix, yes or no? So which enzyme is going to do the same? Which enzyme is going to do the same? Yes, students, which enzyme will help in the opening of DNA helix during transcription? Exactly, exactly. So it will be the RNA polymerase. So RNA polymerase is required here, and it is also helpful in opening the DNA helix. Right, it is also helpful in opening the DNA helix. So in NEET 2020, this question was there, right? And even because many students, they made mistake there. Why? Because they don't read the complete statement. The enzyme used for opening of DNA double helix during transcription. So if I'm saying during transcription, if I'm specifying this transcription thing, then I'm talking about the RNA polymerase. I am talking about the RNA polymerase, right? What am I talking? I am talking about the RNA polymerase. So now you people know it very well. Yes, you people know it very well. Very well you know that there is a start point. There is a start point from where the mRNA synthesis will occur. But upstream to that start point, in promoter region, the RNA polymerase has to bind. Is that clear? Is that clear? Is that clear? Yes, bache. Is that clear? There is a promoter site now. RNA polymerase is there for the opening of DNA helix and the elongation. So this RNA polymerase, it has to bind promoter site. It has to do what? It has to bind promoter site. Is that clear? Is that clear? Now here in this promoter region, upstream to that start point in promoter region, you will see I'm not specifying the number of bases here. Just say 10 base pairs upstream. So when it is the upstream, you also use minus 10 nitrogenous bases. So it means it is specifying, this minus sign is specifying the direction. But let's not discuss that in that detail. Simple it is that you are talking about the start point, and here you have the promoter site. If RNA polymerase has to complete this process, it has to bind to the promoter site. Right, bache? It has to bind to the promoter site. This is the number one thing. And as I said that upstream to the start point in promoter region, you see a Tata box. A Tata box is present there. What is present there, bache? A Tata box is present there, right? This Tata box, and this Tata box is having the bases TATAAT, right? What does it contain? It contains TATAAT, right? 6 base pairs. So basically you can say upstream to the start point, I'm not specifying the number of bases here, you will get the Tata box. You will get the Tata box. And P for prokaryotes, P for Pribnow. It is known as Pribnow box in prokaryotes. It is known as Pribnow box in prokaryotes. Is that clear? Is that clear? Is that clear? And even in eukaryotes also, that Tata box is known as Goldberg-Hugeness box. It is known as Goldberg-Hugeness box. It is known as Goldberg-Hugeness box. Is that clear? Is that clear? So in eukaryotes also, in the promoter region, we have the Tata box, even in prokaryotes. So prokaryotes mein that is Pribnow box. So dekho, it is basically 6 base pairs are there. TATAT. Obviously it is a perfect site to unbind the DNA helix. Why? Because the hydrogen bonding between T and A is what? 2 hydrogen bonds are there. If it will be GC rich, so G and C, they are connected with 3 hydrogen bonds, then it will take more and more energy to break it. So this is what we have in the promoter region. This is what we have in the promoter region, and here your RNA polymerase will start unbinding the DNA helix. Now in prokaryotes, right? Now the next thing is that in prokaryotes you have, yes bache, now the next thing is that in prokaryotes we have what? In prokaryotes sigma factor is also present. There are certain factors that will help in sigma factor, right? That sigma factor, S for sigma, S for start, right? That sigma factor helps RNA polymerase to bind to promoter region. So it will help that RNA polymerase to bind the promoter region. S sigma factor. It will bring that RNA polymerase to the start point. Sorry, it will bring that RNA polymerase to the promoter region. Right. There the RNA polymerase will come to the promoter region, and it will help this RNA polymerase. It will help this RNA polymerase to get attached to the promoter site. So once this RNA polymerase will start mRNA synthesis, and it's not like that in one go mRNA will start forming. Sometimes some bases form, they remove, then proper synthesis is there. So basically what's going to happen once the RNA polymerase, it starts synthesizing the mRNA, then the sigma factor will leave. So sigma factor, it helps in initiation, yeah, in binding of, as I said, binding of RNA polymerase to promoter. Once RNA polymerase starts mRNA synthesis, it leaves, means this factor will leave. Is that clear? This factor will leave. Is that clear? Is that clear? Yes, please. Look at the diagram here. Look at the diagram here. So what is happening? Sigma factor will help RNA polymerase to bind to the promoter region, and that too there in the Tata box, so that unbinding can be easy. So RNA polymerase is going to unbind the DNA helix and during transcription, plus it will also form the mRNA. So polymerization of mRNA will also be done by this, synthesis of mRNA, and even the opening of DNA double helix. So it is also known as DNA dependent RNA polymerase. It is also known as DNA dependent RNA polymerase. It is also known as DNA dependent RNA polymerase. Understood? 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 So once sigma factor starts synthesis, the RNA polymerase starts synthesis, sigma factor leaves. That will be the elongation. As per the basis, as per the sequence on DNA, the basis will be added. Now, what is going to happen? Now then there comes the termination. Means when this mRNA approaches the terminator region. Sometimes we talk about a factor, rho factor. So rho factor will stop or it will terminate. It will stop or it will terminate a transcription, right? And sometimes it can be rho independent as well. It's not mandatory that always this factor has to be there. Sometimes it can be rho independent, and then some hairpin like structure will form and mRNA will leave. Right? So it is in the case of prokaryotes only that you have one RNA polymerase. It is in the case of prokaryotes only that you have one RNA polymerase. It is in the case of prokaryotes only sigma and rho factor is there. These factors are absent in eukaryotes. These factors are absent in eukaryotes. Doubt? Any doubt? This part clear? This part clear? Sure? This part is clear? Sure? Sure? Pakka? Done? So, now when you talk about the eukaryotes, right? In the case of eukaryotes, what is the story? First of all, no sigma and rho factor there. No sigma and rho factor are there. Plus some other factors are present, but yes, we do not discuss about them. But some other factors are present, but we do not discuss about them. We do not discuss about them, right? So, and in eukaryotes, the site of transcription is very important point. Site of transcription is nucleus. It is not there in the cytoplasm. It is in nucleus. This part clear? It is in nucleus. This part clear? And in eukaryotes, you will talk about 3 RNA polymerases. So do not confuse it with prokaryotes. Here you will see 3 RNA polymerases are there, right, bache? And it is the RNA polymerase 2 that forms immature mRNA, also known as precursor mRNA, also known as hnRNA. What is the meaning of hnRNA? One second. It is precursor of mature RNA, and it is the heterogeneous nuclear RNA. So it is your RNA polymerase 2 that will form the RNA in eukaryotes, and that too immature RNA. It is not fully mature RNA at the time of synthesis. It is immature. It is immature. So it will be done by your RNA polymerase 2. And here, bache, remember RNA polymerase 1, it synthesizes your 28S ribosomal RNA, your 5.8S ribosomal RNA, and other than that, RNA polymerase 3 means tRNA, snRNA, and your 5S ribosomal RNA. Right, bache? So SN means small nuclear RNA it is. Small nuclear RNA it is. Thick, small nuclear RNA it is. right? So this part is again very important. This part is again very important. Understood? This part is again very important. Understood? Sure? Sure? Any doubt here? Any doubt here? So, in the case of eukaryotes, right, bache, RNA processing is also the thing. But before that, I'll tell you the concept of split genes, right? Before that, I'll tell you the concept of split genes. Now, when you look at the NCERT part, here they talk about the 3 types of RNA. I already told you about the mRNA, and ribosomal RNA will form the ribosome. And let me tell you, bache, your rRNA synthesis is in nucleolus. It is in nucleolus. So nucleolus is known as the ribosome factory. Okay, so because your 5s ribosomal RNA, your RNA polymerase 3 is synthesizing it, so it will be there in the nucleoplasm. It is not there in nucleolus, it will be there in the nucleoplasm. Is that clear? Right? So all that things are important. So here see, they are saying all 3 RNAs are needed to be synthesized to get a protein in a cell. So mRNA provides the template, tRNA brings the amino acid and reads the genetic code, and ribosomal RNA is plays structural and catalytic role. Okay? So there is a single DNA dependent RNA polymerase that catalyzes transcription of all 3 types of RNA in bacteria, but you know that in eukaryotes you have 3 different types, right? Right? Okay. So RNA polymerase binds to the promoter, initiate transcription. It uses nucleoside triphosphates, right? And these are the, okay, yes, I have missed one point. Yes, I have missed one point in DNA replication as well as in transcription. So while discussing the DNA replication, I told you the bases will be added here, right? The nitrogenous bases will be added here. Yes or no? The nitrogenous bases will be added here. Yes or no? Yes or no? Sorry, nitrogenous bases, instead of that you can say nucleotides. So bache, when we synthesize the DNA or the RNA, just if it is the DNA strand formation, you need D-GTP, D-CTP, D-TTP, D-ATP. So what are they? Just D means deoxyribose guanosine triphosphate, है ना, cited, so it is cited in triphosphate. So in the chain here, what do you add? When you look at when we have studied the structure of the nucleic acid, we said na, monophosphate is added here. This is what we have said. The monophosphate is added here. This is what we have said. The monophosphate is there in the chain, in the polynucleotide chain. But when it comes to the substrate, our substrate is triphosphates. They are not the monophosphates, they are the triphosphates, because they have a dual purpose. They have what? They have a dual purpose. One, they are acting like substrate. Number 2, they provide energy for polymerization. Right? They provide energy for polymerization. What is the meaning of that? Because they are the substrate, we have to attach it in that chain. Now when you are attaching it, because they have 2 extra phosphates, they will get released and they will get removed. So when they remove, the energy is produced. That energy is used for the polymerization of this particular chain. Okay, so even in the RNA, when you have this ATPs, CTPs, GTPs, UTPs, they are required, so they are also having the dual function, right? They will also act like a substrate and they will also provide energy for the polymerization. Important it is, right? They will also provide energy for the polymerization. Important it is. Understood? Understood? Same is the case here in the case of RNA. And that's what given here. That only a short stretch of RNA remains bound to the enzyme. Once the polymerase reaches the terminator region, the nascent RNA will fall off and the RNA polymerases, this is the termination. But here, this is the line that I'm marking. That RNA polymerase binds to the promoter, initiate transcription. It causes these nucleoside triphosphates at substrate and polymerize in a template dependent fashion. Right? Okay. Okay. Done. So bacteria may, you know that, because transcription, mRNA doesn't require any processing. This mRNA will be there in bacteria, mRNA will be there in the cytoplasm. So bache, parallelly in bacteria, ribosomes will attach to this mRNA and protein synthesis will occur. You remember polysomes or polyribosomes, polysomes or polyribosomes, they are present there in cytoplasm. They are present there in cytoplasm. Yes or no? Right? They are present there in cytoplasm. So in bacteria, since the mRNA, that doesn't require any processing to become active, so transcription and translation, it takes place in the same compartment. Right? So it is simultaneous. But in the eukaryotes, the story is different. So in prokaryotes, transcription and translation can be coupled. It can be coupled. It can be coupled. So in eukaryotes, one thing is 3 types of RNA polymerase. Right, bache? So RNA polymerase 1, you know its role. It transcribes 28S. Oh, I missed 18S. Nobody even pointed it out. Ki, madam, you didn't write 18S. You know na 18S ribosomal RNA that will form the 40S subunit of the ribosome. Nobody pointed it. Ki, madam, you missed it. Na, theek hai. Everything is fine. So RNA polymerase 3 is responsible for transcription of tRNA, 5S ribosomal RNA and small nuclear RNA, and RNA polymerase 2 will form hnRNA. Important. Important. Done. Done. Chale. So now, bache, I'll give you one concept. This word is even given in your book, and that is of split genes, right? I'll give you one concept here, and that concept is of what? That concept is of split genes. That concept is of what? That concept is of split genes, right? Split genes are the one having coding part, also known as functional part, and non-coding part. Non-functional part. So, split genes, what do they have? They are having the coding part and they are having the non-coding part. They are having the functional part, they are having the non-functional part. Right, bache? Right, bache? Right. So these are the split genes. They have coding part or non-coding part. Means if this is the segment of the gene, some part of it is having the information for protein synthesis, but some part is not having that. So obviously, if this is the split gene, the mRNA that it will form, the mRNA that such gene will form also contain coding as well as non-coding part. Right? So that type of mRNA is your hnRNA, heterogeneous nuclear RNA. hnRNA, heterogeneous nuclear RNA. So here, this coding part, right? Because this gene itself is having some segmented region, some non-segment, some part having coded in some functional part, some non-functional part. So coding part is known as exon. It is having the information for the protein synthesis. Non-coding part is intron. it is having the information for the synthesis for, it doesn't have any information. It doesn't have the information for synthesis of protein. Just for an example, this is your mRNA. For an example, this is your mRNA. This is your mRNA. See this. See, here in this mRNA, students, let's say this white colored part is exon, the coding part. In between you have intron. In between you have intron. So this is your hnRNA. And in mostly in eukaryotes, bache, we have the split genes. They have some evolutionary significance. Let's not talk about it right now, but this is what we have, right? So this is hnRNA, or you can say this is immature RNA, or you can say this is the precursor of RNA. So in eukaryotes you have this, right? Mostly eukaryotic genes are split genes, right? Mostly eukaryotic genes are split genes. Okay, so the concept of split genes clear? Yes, the concept of split genes clear. So now from this part, right, what you have to do, right, you have to remove the introns. You have to remove the introns, and then you have to join the exons. Then you will get functional mRNA. Then you will get functional mRNA. Or we can call it as mature RNA. We can call it as mature RNA. So this is the functional RNA. This is the mature RNA. So when you say removal of introns, so for removal of introns, there is a word that is known as RNA splicing, and there will be further processing of it. So that is how from immature RNA we have to make the mature RNA that will give us the protein. Is that clear? That will give us because it will be the functional mRNA having information for the synthesis of protein. I hope it is clear, right? I hope it is clear. So basically in eukaryotes, right? See, so in eukaryotes, this is the nucleus part. This is the part where your mRNA will form. So in nucleus, what will be done? RNA processing. So this is the concept of eukaryotes only, right? So RNA processing mein, firstly, there will be the RNA splicing. So RNA splicing means removal of introns. RNA splicing means removal of introns. Okay, bache. And then there is the further processing will include capping and tailing. So this is what we discuss under RNA processing. Is that clear? Is that clear? Is that clear? Sure? Sure? So it's simple. Your mRNA will form here, right? You have to send it to the nucleus. You have to send it to the cytoplasm. Why? Because it is the cytoplasm where translation occurs, right? Be it a prokaryotic cell or eukaryotic cell. Translation will always be there in the cytoplasm, right, bache? Translation will always be there in the cytoplasm, right? Translation will always be there in the cytoplasm. So this is the nucleus that we have, right? So in which hnRNA will form. So obviously it has to undergo processing so that we can get a mature mRNA. Now if we want to send our mRNA to the cytoplasm, but if we have to protect it, right? In the cytoplasm there are the nucleases. There are the nucleases in the cytoplasm. We have to protect our mRNA from that nucleases. So it's simple, right? To this mRNA at 5' end, because you have two ends, 5' or 3'. At 5' end, there will be a capping. Capping will be done at 5' end. What is this capping? Here, an unusual base, that is 7-methyl guanosine. It's 7-methyl guanosine. Triphosphate will be added. triphosphate will be added. 7-methyl guanosine triphosphate will be added here, right? That is basically the capping, and this cap during translation, it helps recognizing the smaller subunit of ribosome. 7-methyl guanosine triphosphate will be added here with the help of enzyme guanyl transferase. With the help of enzyme guanyl transferase. Is that clear? With the help of enzyme guanyl transferase. Am I clear here? Right, bache? Right, bache? So see. Yes, any doubt here? Please tell me. Any doubt here? Right? Any doubt here? So firstly, it will be done. Then there is the tailing. 3' end will have tailing. Now, what is this tailing part? At 3' end, you know, many 200, 300 residues will be added. Many residues will be added, right? Many adenylic acid, yeah, right? Many adenylic acids will be added. So you can say polyadenylate residues. Polyadenylate residues will be added in template independent manner. How will it be added? It will be added in template independent manner. Right, bache? It will be added in template independent manner. Is that clear? Is that clear? So many residues of adenylic acid, yeah, we can call it as polyadenylated residue. They will be added how? In a template independent manner. So what is the meaning of template independent manner? See, you are forming the RNA, right, bache? You are forming the RNA from the DNA. It is on the basis of principle of complementarity. If DNA is having A, your RNA will have uracil. If your DNA is having guanine, your RNA will have cytosine. Complementarity is there. Complementarity is there, yes or no? Complementarity is there, yes or no? Yes or no? Definitely. But here you are not looking for any base. Yeah, you are not looking for any template. In a template independent manner, all the things will be added, and that too with the help of PAP enzyme, polyadenylate polymerase. So near about 200 to 300 residues of adenylic acids will be added here, and this capping and tailing protects this mRNA. It protects this mRNA from the nucleases, from the enzymes which are there in the cytoplasm which can destroy it. Now, when it comes to the, yes, removal of, now we have to discuss removal of introns. So for removal of introns, what is the word that we use? We call it as splicing. We call it as splicing. And who's going to help here? Spliceosome. And they will only be there in the eukaryotes, but you are not going to see them in prokaryotes. That's a PYQ. That's a PYQ. Clear? Clear? So one more thing I would like to mention. As I said, mostly eukaryotic genes are split genes, but there are some eukaryotic genes which are non-split. Right, bache? Which are non-split. Genes for histone and interferon. Right? Genes that are giving us histones and even interferons. It is playing role in that immune response. They are non-split genes. What are they, bache? They are non-split genes. What are they? They are non-split genes. Understood? 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 So for removal of introns, what is required? Spliceosome. We are calling it as splicing. So what is the spliceosome? Bache, it is a RNA protein complex that will remove the intron from that pre-mRNA. Right? This is the spliceosome. This is the spliceosome. So what does it contain? It contains snRNPs. What is it? It is small nuclear ribonuclear protein complex. It is small nuclear ribonuclear protein complex. What is it, bache? It is small nuclear ribonuclear protein complex. What is it, bache? Yes, yes, yes. Small nuclear ribonucleoprotein complex. They are snRNPs. What are they? They are snRNPs. So, or you can call them as SNRPs. That's how they are pronounced, SNRPs. There is one thing known as SNPs, that is different, single nucleotide polymorphism. They are SNRPs. SNRPs. How do we pronounce it? SNRPs. Small nuclear ribonucleoprotein complex. They'll join, they'll form spliceosome that will remove the introns in a lariat fashion, right? And then exons will be joined with the help of ligase enzyme and with the help of RNA ligase. ये देखो. So these small nuclear ribonucleoproteins, see, they'll form a complex. The way they are folding it, the way they are removing it, just have a look, right? So there are around 5 snRNPs, right? So they are required for the removal there, okay? So it can, so see, the snRP, they bind to the intron, causes it to fold into bring the 5' and 3' end of the intron closer. So it forms a loop, and ends of the exon also move closer together to eventually join together. Okay, bache. So intron detaches and splice sites connected to make a mature mRNA. So introns were previously thought to be a junk afterwards, but most are used in other processes. Done, bache. Spliceosome clear. Look at the way they are joining. Look at the way they are removing it. Okay, so RNA processing will be there in the case of eukaryotes. It will be there in the case of eukaryotes. Take. Okay. So before going forward, so see, this is the spliceosome, right? 1, 2, 3, 4, 5, U1, U2, U6, U5, U4. This is the spliceosome that we have. So it will be there in the prokaryotic cell. So look at this part of the diagram, right? Look at this part of the diagram. See this. Introns has to be removed. These are the spliceosomes. So 7-methylguanosine triphosphate and poly A tail is there. Polyadenylation. Done? Okay, so that's it, that's all. Next is the genetic code, but before genetic code, you already know about the mRNA. So let me tell you about the ribosomal RNA and then let's proceed. So tRNA ka structure we will study just before translation. So this is the ribosomal RNA, rRNA. So ribosomal RNA along with proteins, bache, they form ribosome. Like in the case of eukaryotes, if you ask me specifically in eukaryotes, and that is again a PYQ, 2023 ka PYQ. In eukaryotes, how many proteins are there? Near about 80. Jaise I think, it is a question. It was a question there in NEET 2023. How many proteins, ribosomal proteins, are forming the ribosome? So in eukaryotes, they are around 80. Okay, bache? Okay. So when you talk about the rRNA, obviously ribosomal RNA, and it has to form what? It has to form the ribosome. So its quantity is maximum. Do you know that? So it is generally making 80% of the cell's total RNA. Total RNA. Do you know that? Yes. Right. So look at this part. Ye dekho. So you know that in prokaryotes, smaller subunit is 30S. Larger subunit is, or I'll draw it in my way. Larger subunit is 50S. Smaller subunit is 30S. In prokaryotes, right? And not just in prokaryotes, my dear students, but even in mitochondria and chloroplast. and they will only join at the time of protein synthesis with the help of magnesium ion. So this is the larger subunit and this is the smaller subunit. So what is S here? What is S here? Class 11 concept. It is Swedberg unit. It is Swedberg unit and it is a sedimentation coefficient. Right, as per the way things they settle down, no? So it is a sedimentation coefficient as well. So do remember it, students. Do remember it. Your 30S subunit, right? No need to plus it. No need to add it. 50S is having 23S ribosomal RNA, 5S ribosomal RNA. Here you have 16S ribosomal RNA. So you have 34 plus 21, near about 55 proteins, or you can say somewhere 60 proteins are there. Right, near about 60 proteins are there. In 80S, look at this. Right, 18S ribosomal RNA is making the 40S subunit, and when it is the 60S, that is larger one, that's what we have. Kindly write it. Hanji, please write it. Yes, please write it. Done? Okay, chalo, let's go. So tRNA, we will just discuss it before translation. Now let's talk about the genetic code. What do you know about it? What do you know about it? Be quick. What do you know about it? Let's finish this chapter till 6:30. Then let's have a break. And after the break, what will we do? Biotechnology. So biotechnology is not very lengthy. We will directly start with the tools and the steps. We will not complicate it. Okay, we are not going to complicate it then. Anji, tell me. Tell me, what do you know about the genetic code? Yes, people, what do you know about the genetic code? Anything? Coding for amino acid, you answer it. You answer it. Let's have 1 or 2 minutes ka washroom break. Okay? Let's have 1 or 2 minute ka washroom break, and I need to get my water bottle, of course. I stay alone, so I have to go. I have to fill it, and then I have to come. Okay? So just allow me 1 minute. Okay, I'm back. Okay, so bache, genetic code. Term was given by George Gemo. Okay, and discovered by Nirenberg, Mathai Khorana, Dr. Hargovind Khorana, right? Right? Yes, bache, Dr. Hargovind Khorana. So what are we talking? We are talking about the genetic code, right, bache? So first of all, yes. So discovery part clear. Now, see, it's very simple. When we talk about the central dogma of information, that's how the things are. You have DNA. DNA is forming mRNA. mRNA is forming protein. That's what we have. This is the central dogma of information. DNA is forming mRNA. mRNA is forming protein. Yes. Yes, ma'am. That is what you have. That is what we have. Right. So now, bache, here the point is very simple. Point is very simple. DNA is giving us a mRNA. Both are nucleic acid. Complementarity is there. Now, mRNA is giving you the protein. Protein is again a polymer, but it is a polymer of amino acids. It is a polymer of amino acid. How a polymer of nucleotides can give us the polymer of amino acid? Right? Because that is a question that was fascinating the scientists a lot. That how is it possible that the polymer of nucleotides can give us the polymer of amino acid? So that relationship actually is explained with the help of your genetic code. So genetic code explains that relationship. Right, bache? Right, bache? Okay, so this part is written here that process of translation require transfer of genetic information from a polymer of nucleotide to synthesize a polymer of amino acid. From a polymer to nucleotide to synthesize a polymer of amino acid. So neither does any complementarity exist between nucleotide and amino acid, nor could any be drawn theoretically. So we cannot say that any complementarity is there, right? Yeah, any correlation is there, right? So there are ample evidences that suggested this notion that if we change the nucleic acid, it directly changes the protein, right? So basically some relation is there, and scientists, they were trying their best to decode it. They even tried their best to decode it. So if determining the biochemical nature of genetic material and the structure of DNA was very exciting, the proposition and deciphering of genetic code were most challenging. It was not that easy task. So in a very true sense, it required involvement of scientists from several disciplines: physicists, organic chemists, biochemists, and geneticists. Right, bache? So George Gemo, he gave the term genetic code. He was a physicist who argued that since there are only 4 bases, if they have to code for 20 amino acids, the code should constitute a combination of bases. Right? Right? So even in the chapter biomolecules, I requested you people to remember, right? To remember the name of all these 20 amino acids if you know, right? So these 20 amino acids, I think you should cram their names, and you can go for the alphabetical order. It works for me. So we have alanine, arginine, asparagine, aspartic acid, alanine, arginine, asparagine, aspartic acid, cysteine, glutamine, glutamic acid, glycine, histidine, isoleucine, leucine, lysine, methionine, proline, phenylalanine, serine, threonine, tryptophan, tyrosine, and valine. So tell me, I missed something or not? So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, and 19, 20. It's done. So these are the 20 amino acids. They are the ones that are involved in the protein formation. So one is your selenocysteine also and pyrolysin. It is your 21st and 22nd amino acid. So in NCERT this is not mentioned, but yes, again as I said, you people should know about it. So what is the name here? It is selenocysteine and pyrolysin. What's that? It is selenocysteine and pyrolysin. What's that? It is selenocysteine and pyrolysin. Understood? Yes, bache. Understood? Your 21st and 22nd amino acid. Right? Your 21st and 22nd amino acid. Selenocysteine and pyrolysin. So you should know about it. Right? These are the 20 amino acids. Now we have 20 amino acids that are going to form what? That are going to form the proteins. So now when you talk about the bases, you have UCAG. 4 bases are there. So let's say if I consider that each base is giving me each amino acid, then I will get only 4. If let's say I'm taking the combination of 2, that this position can be filled with any of the amino acid here, 4 into 4. 16. So basically if I take the combination of 2 like UC, yeah, CU, right, it can be CA, it can be CU, it can be AC, it can be AU, right. If I take the combination of 2, then I will get the 16 amino acid only. So now if I take the combination of 3, it was clear. George Gamow was clear that if we want from these 4 nitrogenous bases, right, from these 4 nitrogenous bases, if we want the amino acids to form, definitely they should be in combination. Individually they cannot do it. They cannot do it, right? So they should be in the combination. So it is 4 into 4 into 4, that will be 64. It makes sense. It makes sense. How? It makes sense, right, bache? Right, so combination of 3, that is 4 into 4 into 4, because any base can come here, any base can come here, any out of that 4, any base can come. So it will be 64 codons, and that are generating many more codons than required, right? Many more codons than required. So it's simple. Here we have a very simple mathematics. Please mark it, right? You have 64 codons. Out of that, let's subtract 20. We will get 44 codons. Out of these 44 codons, bache, 3 codons are the stop codons, let me tell you. 3 codons are the stop codons. They are the nonsense codons. They stop the protein synthesis. So it will be 41 codons. So basically we have extra 41 also. So this is suggesting us that one amino acid can be coded by more than one codon, right? Codon. So now, can I say that if this codon is triplet? Triplet means 3 nitrogenous bases are making one codon. 3 nitrogenous bases are making one codon. Of course, we can, right? Of course, we can. So there are some other discoveries that are mentioned here along with the scientist name. Yes, you have to remember it. They say providing proof that codon was a triplet was a very daunting task. So chemical method developed by Hergovind Khorana. So he was instrumental in synthesizing RNA molecule with defined combination of bases, homo and copolymers. Just let's say if they synthesize the RNA, let's say if it is having same bases, U, so on. It will give phenylalanine, phenylalanine, phenylalanine. So like that complete polypeptide chain will have phenylalanine. Now let's say if it is doing like this. Oh. So if it is like this, let's say, I'm just giving you example, Cu, 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 right? So again, CuC, UCu, CuC, UCu, CuC, UCu. So you will have 2, right? Same amino acids like this. Yes. So Hergovind Khorana. Who? Hergovind Khorana. Yes, bache. Who? Har Govind Khurana. Yes, bache. So he was instrumental in synthesizing RNA molecules with defined combination of bases. With what? With defined combination of bases. Is that clear? Is that clear? The next discovery, Marshall Nirenberg. He formed a cell-free system. Matlab, it's not mandatory that always the protein synthesis will be in a cell. So in a cell-free system, how the protein synthesis can be done. So obviously if we can synthesize the protein without a cell, so it can help us to code. It can help us to decipher that code. If we can understand that which bases, right, which combination of bases is forming which amino acid. That's the meaning here. Then Severo-Ochoa enzyme, that is polynucleotide phosphorylase. Polynucleotide, many nucleotides, polynucleotide phosphorylase. So it was helpful in polymerizing RNA with defined sequence in a template independent manner. So basically this enzyme helped the scientists to form the RNA in template independent manner. They are not dependent on the DNA or any other template only. They are like, okay, with the help of enzyme, let's synthesize the RNA. Finally, a checkerboard for genetic code was prepared. And that checkerboard was also simple. How many bases do we have? How many bases do we have? How many bases do we have? Four na? Four. है ना? Four. So the trick here is you can arrest gangster. What's the trick here? You can arrest gangster. You can arrest gangster. You can arrest gangster. So this, see, whenever we read the mRNA, it will be there in 5' to 3' direction. Whenever we read the mRNA, it will be there in 5' to 3' direction. So you can arrest gangster. Second base is also, you can arrest gangster. This is the third base. You can arrest gangster. That's how you have to write. Are you getting my point? That is how you have to write. That is how you have to write. So this is the 5' end. This is the 3' end. Okay. Right, that's how we have to write. So this is the first codon, this is the second codon, that one is the last codon. Right, so I'll give you the example. Just say, let's say, here I'm writing, first codon is A, second codon is C, AC 4 times, then you can arrest gangster. It's very easy to make this checkerboard. Matlab, now for us it is easy, but it was very difficult to decipher it. Then C, first codon C, second is A. So first two are CA, CA, CA. Then you can arrest gangster, UCAG. Come here, it is AG, 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 AG. You can arrest gangster. It's simple. It's simple. Yes or no? Yes or no? So UC, 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 UCAG, CC, UCAG. So don't you think it's easy to make this checkerboard? Here its first base is GC, GC, GC. Do practice it. You can arrest gangster. So for this particular column, the trick is Sir Pro Thar Ala means serine, proline, threonine, alanine. Serine, proline, threonine, alanine. Yes, bache. Serine, proline, yes, threonine, alanine. Is that clear? Is that clear? Right, so you can see serine is 4 here. Others are also there, proline, threonine, alanine. So these are the 3-letter code. Single letter code is also there. Single alphabet code is also single letter code is also there, but it is the 3-letter code. Just say ALA, alanine, right? ARG, asparagine. ASN, ARG, arginine. ASN, asparagine. Like this. So arginine, ARGININE, it is also denoted by capital R. So single letter code is also there. Right, these 3-letter code is also there. Right, so yes, there is a video here on Unacademy NEET English. Right, there is a video here on Unacademy NEET English only. right? You guys can put it genetic code, tricks for genetic code by Ambika Mam. You can search it. Simple. You can search it. So that's my suggestion. Do check that video for the genetic code, right? Because here in that particular video, even the properties and everything, right? Right, everything is explained there. So जैसे ये देखो UUC. Look at this. First two bases are same. There is a third base. This is actually बच्चे the vowel base, right? I'll explain it. Two are same, third is different. Two are same, third is different. First two same, third is different. Can you see that? So this is your genetic code, right? So do check that video, please. It's a request. So one thing that you need to remember here, AUG, it codes for bache methionine. It codes for methionine, and this methionine is the start codon or the initiator codon. It is the one that synthesizes that starts the protein synthesis, right? In prokaryotes, we call it as formyl methionine. In prokaryotes, we call it as formyl methionine. So it is AUG. It is AUG, and very rarely GUG can be the initiator codon. Very rare it is, and sometimes this GUG also code for methionine. So it's a rare thing, right? But mostly AUG is the start codon. Then bache UGG codes for TRP, that is your tryptophan. So tryptophan is coded by only one codon. There is only one codon for tryptophan. There is only one codon for methionine. You should know about it. You should know about it. जैसे ये देखो, I'll give you the example. For leucine, you have 6 codons, right? Right, बच्चे? For leucine, you have 6 codon. For serine, 4 here, 2 here, 6 codons are there, right? 6 codons are there. Can you see that? Isoleucine, 3 codons. Methionine single. Valine, 4. Yes, बच्चे? Valine, 4. Right? Doubt? Yes, बच्चे? Any doubt? Yes, please. Any doubt? Okay. Okay. So 3 are stop codon. You should know about stop codons. So how can you remember it? It's UUU, AG, AAG, AG, AGA, AGA. So it's UUU, AG, AGA. It's UUU, AG, AGA. It's UUU, AG, AGA. Is that clear? What's that? It's UUU, AG, AGA. Understood? Understood? Right? So, opal, ochre, amber. So these are the stop codons. They'll stop the translation. They are also known as the nonsense codon. Ochre, amber, opal. Right, bache? Right, bache? So they are the nonsense codon or the stop codon. They stop the protein synthesis. Understood? Right? So genetic code ki properties, they are also very important. They are also very important. Right? This is genetic code is triplet. 61 codons code for amino acid. 3 are the stop codons. So some amino acids are coded by more than one codon. This is the degeneracy. So I will explain these two properties because I have seen students struggling here. Right? See, one property is non-ambiguous. Ambiguous nature of genetic code. Non-ambiguous nature of genetic code, and one is what? Degeneracy of genetic code. One is what? Degeneracy of genetic code. Is that clear? Degeneracy of genetic code. Is that clear? Is that clear? So non-ambiguous. See, ambiguous means doubtful, but genetic code is non-ambiguous, or you can say unambiguous. So sometimes while reading the question, we just focus on the word ambiguous. We do not read what are they asking actually, non-ambiguous or unambiguous. Okay? Okay. So what is the meaning of this non-ambiguous nature? It's simple, bache. If you are talking about triple U, it in all the possible conditions, it will code for phenylalanine only. It is not going to code for anything else. Okay, it is not going to code for anything else, right? So it is Non-ambiguous, like if you will talk about the genetic code, be it the bacteria, or it is a virus, or it is a unicellular eukaryotic organism, or it is a multicellular eukaryotic organism, you people will see that UUU is giving you phenylalanine only, right? Some exceptions are there. Some exceptions are there, but otherwise it is like this. Okay, otherwise it is like this. Right, bache? Right, bache? Clear? Clear, 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 clear. So it is the non-ambiguous nature. What is the meaning of degeneracy? The meaning of degeneracy is very simple, right? The meaning of degeneracy is very simple, right? Here, degeneracy means one amino acid can be coded by more than one codon. For an example, UUU, UUC, both are giving you phenylalanine. Both are giving you phenylalanine. Simple. Now, UC. So, all four are giving you serine. All four are giving you serine. There are two other codons for serine as well, like your AGU, AGC. So these six are giving you serine. So it is the methionine and it is the tryptophan that are having only one codon. Rest all are having more than one. This is the degeneracy. And bache, it is said that this degeneracy is due to the third base. So this third base is also known as vowel base, right? And during translation, I'll tell you about it in detail. So this part clear? This part clear? This part clear? Bache, NEET Avengers Reloaded batch is a kind of entry point for your NEET Avengers fast track batch, right? So please, Shahrukh, do not spam here in the chat. Okay? Okay, so this portion is clear. This portion is clear, everybody here in the class. This portion is clear. Okay, so these two properties, they are the confusing one for students sometimes. So remember, so codon is always read in contiguous fashion, like it will be first three codons, then next three, then next three. No overlapping, no punctuation, no comma. So it is commaless. It's commaless. Okay? And then the code is nearly universal. From bacteria to human, UUU will give you phenylalanine, right? Some exception to this rule have been found in mitochondrial codons and some protozoans. So AUG is having dual function. Told you these are the stop codons. Done and dusted. Chali. Next, mutations and genetic code. देखो बच्चे, mutations and genetic code. See, we have the example of sickle cell anemia here. What is this sickle cell anemia? Bache, it is the result of point mutation. It is the result of point mutation. Point mutation means there is a change in single base pair. There is a change in single base pair. Just for an example, mutation part in detail we will discuss in principles of inheritance and variation. Just for an example, right? You know about the hemoglobin. Hemoglobin is the respiratory pigment. Hemoglobin is the respiratory pigment. It's a quaternary protein. It's a quaternary protein. It is having 2 alpha and 2 beta chains. It is having 2 alpha and 2 beta chains. Right, bache? Now, in hemoglobin, in beta chain at 6th position, it's a very, very, very important topic. Right? In beta chain at 6th position. Right? What is going to happen, bache? Glutamic acid is replaced by valine. It is replaced by valine. Are you getting my point? So in beta chain at 6th position. In beta chain, at 6th position, glutamic acid is replaced by valine. So the trick is good valine. The trick is good valine. Good GD, glutamic acid, valine. So glutamic acid is replaced by valine. Why? Because GAG will become GUG. This is my way to remember. GAG will become GUG. GAG will become GUG. Means there is a change of single base pair only. That's why it is the point mutation. At A, U will come. So this is giving us the glutamic acid. This will give us the valine. So when there is a normal hemoglobin gene, so GAC, CTC, that is in the case of DNA. So yes, question can come from this part also. So on mRNA, it has to be GAG. So if it is GAG means your CTC is the template strand. Keep it in your mind. Your CTC is the template strand because information on mRNA and coding strand is same, right? Is same, right? So what is there in that HBA peptide? First valine, histidine, leucine, threonine, proline, glutamic acid. Yes, you should remember this sequence. Question can come from this part. But when there is a mutation, and that to be of one, just one single base pair, so GAG, here it was GAG, here it will become GUG because there is a change in this gene here, and at the place of glutamic acid you will see valine. So RBC will become sickle shaped. Obviously the oxygen carrying capacity will also get affected. There is only one blessing in disguise, that the person having sickle cell anemia will not get affected with the malarial parasite. That's it, right? Will not get affected with the malarial parasite. That's it. Okay, so let's discuss the structure of tRNA and then let's go to the translation. tRNA translation operand, 3 major concept related topics, and HGP is completely theoretical, right? I'll mark the important things there, and DNA fingerprinting is also not difficult. We just need to read about the steps there. Okay, so up to this part all clear, guys? Up to this part all clear? So we have to finish it ASAP so that we can finish what? Yes, we have to finish it ASAP so that we can do what? So today, even if we target molecular basis of inheritance and both the units of biotechnology along with reproductive health, so still we have prepared for 80 marks. You can say ki haan ji ma'am, 80 marks are done and dusted. Nahin? Yes? Sure? Yes? Chalo. So next is the tRNA, right? Next is the tRNA, the adapter molecule. Next is what? Next is the tRNA. Dekho, I started my lecture knowingly. I started my marathon with the most important chapter. I know this chapter is lengthy. I know this chapter is important as well. So we cannot just skip it. We cannot just focus on the important things here only, because here ultimately everything is important. These are the mechanisms that you need to understand, and these are the mechanisms that you guys cannot avoid. Okay? Keep it in your mind. First thing. Second thing is that even in biotechnology, we will move quickly. Right, the chapters are not that lengthy. We can move quickly, right? We can just finish it. We can just finish it quickly. So if we will finish it today, in the next marathon, we can target principles of inheritance, evolution, ecology together. Okay. Then we can go for the third part: human reproduction, reproductive health, sexual reproduction in flowering plants, microbes in human welfare together. Okay, right? Human health and disease together. So in 3 classes, in 3 marathons, we will finish the syllabus, and that too in a proper way, right? So then I can say ki yes, if you watch my lectures, your class 12th is completed. Let's say if 180 marks are from class 12th, just giving you one example, yes, even just by watching these lectures and making notes and revising them again and again, you can ensure your 150 to 60 marks. And for other 20 marks, you need to read the chapters from the NCERT. Okay, you need to read the chapters from the NCERT. So now what we have to start? We have to start the tRNA. Okay, so just allow me 1 minute. students, I'll make one thing very clear here, right, bache? I will make one thing very clear here, right? If you are comfortable in that, very good. If you are not, then it is all up to you. Like some students are saying only one chapter, only one subject, only one. So what else do you expect? What else do you expect? Do you think that in 15 minutes it is possible to complete all the topics here? No. Do you think that MBOI is a chapter which we can ignore? Do you think it is a chapter where we can just give 2 and 2.5 hours and it will be done? It can only be done when students are already prepared. When they know, yes ma'am, we know this chapter, we know everything, you just start from the concepts only. You do not explain what genetic code is. We know what genetic code is. Leave it ma'am. You directly come to translation. Okay? So the point here is that you are also expecting a detailed lecture. You are also expecting to score 600 plus marks. So I cannot differentiate that the students that are my viewers right now, do they know this chapter or not? So accordingly we have to study. Okay, accordingly we have to study. And as I said, we need 3 lectures, 3 marathons to complete the class 12th. And that should be in such a way that students are able to grab it. Right? I'm not here for any formality, right? I'm not here for any formality that yes, I have completed the syllabus. Okay, this is done, leave it. No, I'm here to explain. Okay? Right? I'm here to explain. Okay? So it's all up to you. If you are not comfortable, you can watch the recorded lectures that are already there on this channel, right? You can go for it. You can watch them in detail. And there are some quick revision sessions also. Go for it. Okay, go for it, right? Done. So, Mr. Pandit, leave the class. That is something that I'm going to suggest. But I am not just going to be like, okay, like most of the teachers, they are doing this thing. 1 topic, 2 topics, 3 topics, this is it, this is it, this is it. In this topic, this is what you have, this is what you have. That thing can only work when you have completed that chapter twice or thrice. Okay. Right? That cannot work. That cannot help. Right? You are just giving yourself a feeling that yes, I have completed the syllabus. But trust me not. I took a lot of time. But after this, when you will read NCERT, there will be no line that you will not be able to decode. And that is the agenda here. From this chapter, 6 to 7 questions can come. Means 24 marks minimum. 24 marks minimum. We cannot. Right? I cannot compromise that. I'll be very clear about it. Okay, so now come here and revise. Now the point here is tRNA, the adapter molecule, and it is making near about 10 to 15% of the total RNA of the cell. Right, 10 to 15% of the total RNA of cell. Are you getting my point? Are you getting my point? And trust me, I was prepared for it. I was prepared for it. Right? I was prepared for it. Ki I will take 3 marathons, and in that 3 marathons, I'll be completing the topic. It's biology. It is not physics. It is not chemistry. And especially where I can just put the formulas or I can solve the questions. Right? I cannot say that, okay, you might have done this derivation. Let's directly come to the formula. Let's solve the questions. Not possible here. It's biology. It's a theoretical subject. We need to focus on the theory. Because a lot of theory is there, then yes, we have to study it as well. We have to give a lot of time here, right? Right? If you will check any good marathon or any good one shot, you will always find that this lecture will be covered in 2 parts and a lot of things are discussed here in this chapter. Okay? So it's totally up to you guys. What do you seek? As per that, just go for it. Done? So tRNA is the adapter molecule. tRNA is the adapter molecule, 10 to 15% of the total RNA of cell. That's what we have. And it is also known as, bache, yes, other than adapter, soluble RNA. Why do we call it as soluble RNA? Yes, so tRNA was also known as soluble RNA before the genetic code was postulated. Right, bache? Before the genetic code was postulated. Right, bache? Is that clear? So let's discuss the tRNA. One advice. So in the NCERT, they have given you this serine and tyrosine. Right? They have given you this serine and tyrosine. Right? So you should know their codons. You should know their codons. Is that clear? Is that clear? Right? Yes, bache? Okay. Done, bache? Chaliye. So now let's move to the next part. Let's move to the next part, tRNA. Here in the case of tRNA, I will directly mark the kind of questions that can come. Are you clear with this diagram? Do you know or do you understand this diagram? Do you know? Yes, bache, do you know about this diagram? Yes or no? Yes? There, done? Yes, bache. Tell me. Tell me. Yes. Do you know about it? See, you are talking about the tRNA, right, bache? So the one famous question is about its structure, 2D and 3D. So if it's a two-dimensional structure, it's a clover leaf-like structure. If it is a 3D structure, then it will be inverted L. That's a 1 PYQ. It is inverted L-shaped structure. What's that? It is inverted L-shaped structure. Clover leaf, inverted L shape, number one. Second point here is, bache, see, when you look at the tRNA structure, it is having the arms. When we are saying arms, see, these bases, they are complementary to each other. They are forming the hydrogen bonds, of course, right? They are forming the hydrogen bond, of course. And when you are talking about this part, this is loop. Here, you will see that this bond, this base is not forming base pairs here. This is the loop part, this is the arm part. This is the first thing. Now 5' end and 3' end is there. 5' end and 3' end is there. So when you talk about the 3' end, always when you read anything, when you read any RNA, you will go in 5' to 3' direction. You will go in 5' to 3' direction. Yes, bache. Yes, bache. Yes or no? Yes or no? You will go for the 5' to 3' direction. Yes or no? Yes? Please tell me. Done? So it is your CCA. So always at 3' end, we know that at 5' end phosphate is there, at 3' end OH is there. At 5' end you will have guanine. At 5' end mostly you will have guanine, right, bache? So when you see this 3' end, always it will be CCA. Some people call it ACC, but why am I specifying CCA? Because we read it in 5' to 3' direction, so it will be CCA. This part clear? This part clear? So this arm here is what? It is the amino acid attachment site at 3' end with COOH group. Like when you talk about the amino acid, that's what you have. That's what you have, bache. This is the general structure of an amino acid. Yes or no? This is the general structure of an amino acid. Yes or no? Tell me. This is the general structure of an amino acid. Yes or no? Please tell me. Yes, bache, please tell me. Is it the general structure of amino acid? Yes or no? है ना? Yes, ma'am, it is. So basically this amino acid with its carboxylic group, here it will attach to this OH. Amino acyl bond will be there. Keep it in your mind. Amino acid will attach here to this OH, right? With the help of its carboxylic. With the help of its carboxylic group, number one. Amino acid acceptor arm. Just here, this is the anticodone arm and this is the anticodone loop. But it will attach to mRNA. It will attach to mRNA. It will attach to mRNA. Then you have DHU loop or DHU arm. DHU, it is your dihydrouridine. It is your dihydrouridine. What's that? It is your dihydrouridine. It is your dihydrouridine. Keep it in your mind. Then you have T-Cyc loop or T-Cyc arm. This sign, it means pseudouridine. They are unusual bases. This is pseudouridine. This point clear? All of them are PYQs. All of them are PYQs. Right, bache? All of them are PYQs. Is that clear? Is that clear? Is that clear? Yes? Done, bache? Done? Okay. So, now, what you need to remember here. See, this is my way to remember. If it works for you, go for it. It is the D loop. D for down. D for down. When we feel down, we need charging. We say, na, yaar, I need to charge up myself. Right? We need the charging. So, D for down, who's going to charge here? Enzyme is going to charge here. So, this DHU arm, DHU loop, it joins with the enzyme. It attaches itself with the enzymes. And remaining this T psi C, it will attach itself to the ribosome. So that's why it acts like a adapter. This side will join with the mRNA. This side T, ribosome, D loop, enzyme, and here amino acid will attach. This part clear? This part clear regarding the tRNA? Now, what you need to remember here, we have 20 amino acids. Yes, we have 20 amino acids. How many amino acids do we have? 20. So, the translation is there in the cytoplasm, be it's a prokaryotic cell or it is a eukaryotic cell, the translation will be there in the cytoplasm. This is the site of translation. Are you getting my point? This is the site of translation. Are you getting my point? So if you have 20 amino acids now, so there will be 20 specific tRNAs for that. There will be 20 specific tRNAs for that. If there are 20 amino acids, there will be 20 specific tRNAs, and mRNA is having the codon. As per that codon, yes bache, as per that codon, tRNA will bring the amino acid, right, and further it will help in the addition of that amino acid so that protein can form. This part clear? Now the point here is, ma'am, no doubt we have 20 amino acid, but we have even more than one codon. Then in that case, what's the scenario? Guys, in that case, you need to focus on the Wobble hypothesis. Right, bache? So, even while explaining you the genetic code, I made it very clear that first two bases, right? I made it very clear that first two bases, yes, that first two bases, they show the, yes, bache, Watson and Crick pairing. Like when there is the mRNA and tRNA, when they will attach to each other, first two bases, they will show what, bache? They will show Watson and Crick base pairing. Right? What will they do? What will they do? They will show Watson and Crick base pairing. Right? They will show Watson and Crick base pairing. Is that clear? Is that clear? Is that clear? Watson and Crick base pairing. Third base will be the unusual one. Right? Third base will be the different one. Right, bache? Third base will be the unusual one. Third base will be the different one. Right, bache? Right, bache? Okay. 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 So on that basis, you will say that tRNAs will be 20. That third base will show the vowel base pairing, and then, right, it is manageable. But 20 amino acids are there. Then, now, when we are talking about the tRNA, obviously we are moving towards the translation here. Obviously, we are moving towards the translation here. Right? Where are we going? We are going towards the translation here. Where are we going? We are going towards the translation here. Now, the point here is, students, like, see, for translation, ribosome, mRNA, tRNA, amino acids, they all are required. Some other factors are also required. Now, what am I explaining? I'm explaining a very simple structure here. You have 20 amino acids, you have 20 specific tRNAs. Their third base will show the Bobble base pairing, right? So that is not the usual Francis and Crick base pairing, right? So even if there are different codons, but the amino acid is same, the tRNA will manage. Then we also talk about the enzymes here, the specific enzymes here, and that enzyme is amino acid tRNA synthetase. Amino acid tRNA synthetase. That's the enzyme that will be used here, right? They are also 20 in number one. Now, when you talk about the ribosome, you know it very well that only at the protein synthesis, only at the time of protein synthesis, the larger and smaller subunit, they join. the larger and smaller subunit they join. So here what you need to remember, that when you talk about the ribosome, the larger subunit is having 3 sites. It is E, it is P, it is A. It is just like our homework. Yes, we should know that. So it is EPA. What do we have here? It is EPA. What do we have here? It is EPA. Is that clear? Is that clear? What do we have here? It is EPA. It is EPA. So E is the exit site, P is the peptidal site, A is the aminoacyl site, A is the aminoacyl site. These are the 3 sites. These are the 3 sites. And moreover, in larger subunit, here you will see a kind of groove. Not the kind of, actually a groove from where the protein chain or polypeptide chain will come out. This part clear regarding ribosome? Any doubt here? Any doubt here? That here you have EPA, there is a groove through which the polypeptide chain will come out. That's what we have. Amino acid tRNA synthesis, ribosome. This is our requirement, right? This is what? This is our requirement. Look at the structure of tRNA here for quick revision. So this arm here is the variable arm. So right now, so its function is not clear, but it helps to categorize tRNA into 2 categories, class 1 and class 2. Class 1 and class 2. Is that clear? So this is the CCA part, this is the anticodon, right? So ye dekho, look at this. Just see, this is your tRNA, 5' to 3'. Let's say this is your mRNA. You know that starter codon is what? The start codon is what? Yes, Bhavya, the start codon is, please tell me, the start codon is? The start codon is? Yes, please, the start codon is? AUG. So if mRNA is having AUG, what will you have on tRNA? You will have UAC. So if 5' end of mRNA is having A, this is at 3' end and this is at 5' end of tRNA. So be clear about it. Be clear about it. Okay? Be clear about it. Understood? Right? Arms clear, structure clear. Now we are going to talk about the translation. So look at it. Okay? Translation is a very simple topic, I believe. Literally, translation is a very simple topic. Right? It is a very simple topic. How many of you find it simple? Quickly tell me. How many of you think that it's a simple topic? Or how many of you think, nahi yaar, ma'am, it's a difficult one? Nahi, ma'am. It's not simple. It is the difficult one. It is the difficult one. How many of you think so? Moderate. Arey, write it now. Moderate, difficult, easy. What's that? It's a simple topic. I agree. It is genuinely. It's very simple. You have the mRNA. 5' to 3' end, and that mRNA is having the codons. That mRNA is having the codons. Are you getting my point? That mRNA is having what? That mRNA is having the codons. Isn't it simple? Isn't it simple? It's simple, ma'am. Now what's going to happen? See, for the protein synthesis, and again, it's a previous year question, that's why I'm addressing it. The very first thing that is required is the activation of amino acid. The very first thing. What is the meaning of amino acid? Be it the prokaryotic translation or it is the eukaryotic translation, the start codon is methionine. In prokaryotes, it is formyl methionine. In prokaryotes, it is pragya, formyl methionine. So you are talking about the activation of amino acid. You are talking about the activation of amino acid. So what is going to happen? Your amino acid plus ATP. The enzyme is aminoacyl tRNA synthetase. What is the name of enzyme? Amino acyl tRNA synthetase. So what is going to happen, bache? Amino acid AMP enzyme complex will form, right? And these two pyrophosphates, they will release. Understood? They will release. This is the first step. Second step is the charging of tRNA. Charging of tRNA, or you can also call it as loading of tRNA. Charging of tRNA, and you can also call it as loading of tRNA. Is that clear? Charging of tRNA, or you can also call it as loading of tRNA. So it's very simple. It's very simple here. What is the meaning of this? This is the specific amino acid that is charged. We have to put it on its specific tRNA. Definitely this enzyme is going to help. So this enzyme, amino acid enzyme complex, amino acid AMP enzyme complex, what's going to happen? This is the tRNA, and we want our amino acid to attach at its 3' end. That's what we want. So how is it this possible? This enzyme will play the role. This enzyme will play the role. It's like, for an example, Sai and Shizuka, they have a fight, okay? And I am the person, I'm like, Sai, you should talk to Shizuka, okay? Shizuka, you should talk to Sai. So I'm telling you this thing, I'm convincing you people to come and to talk. To come and to talk. So I will be just like an enzyme, right? I'm bringing you people closer. I'm telling you, okay, now discuss your issues. Sort it out. That's the thing here. You have the enzyme. You have the enzyme, right? With the help of ATP, that enzyme is attached to that amino acid. Now it has to, right? It has to charge the tRNA, load the tRNA. It has to put that amino acid to the tRNA. So that enzyme will be like, it will bind to this tRNA site, tRNA's D loop, and then it will bring that charged amino acid to this 3' end here. That's how the amino tRNA will be charged. Okay, so you said you know this topic, so let's be quick. Okay, so this amino acid AMP enzyme complex, this amino acid AMP enzyme complex plus tRNA, what will you get? tRNA amino acid, and the enzyme will be free and your AMP will be free. Hanji, doubt? Your doubt? Any doubt? This is the charged DNA. Now you know that when it is the translation or if it is the transcription, 3 steps are common. First, initiation. So for that initiation, what has to be done? This is the mRNA, right? This is the mRNA. It has to bind with the subunit. Which subunit? Smaller subunit. So if it is 30S, means I'm talking about the prokaryotes here. If it is 30S, it means I'm talking about the prokaryotes here. Yes or no? Yes or no? So what is going to happen here? That you have the smaller subunit, 30S. Right? You have the smaller subunit, 30S. Yes or no? You have the smaller subunit here. This is the 30S. It has to bind with the mRNA. How will it get attached to the mRNA? Bache, there is a sequence that will help. Dekho, 30S subunit is ultimately having 16S ribosomal RNA. Yes or no? Yes or no? It is ultimately having 16S ribosomal RNA, right? Yes or no? Yes or no? So, bache, this mRNA of prokaryotes, they have a sequence. They have a sequence of some base pairs. So, it is known as Shine-Dalgarno sequence. Shine-Dalgarno sequence. Right? It is the sequence of certain bases. We call it as FD sequence or Shine-Dalgarno sequence. So, complementary to its sequence is present on 16S ribosomal RNA. So, 16S ribosomal RNA is having anti-Shine-Dalgarno sequence. So, they will show the complementarity here. That is how the smaller subunit will bind with the mRNA. Is it clear? Shrek, Sai? Yes, guys. Is it clear? Is it clear? So, mRNA will bind with the smaller subunit. It is the 30S here. 30S subunit is having 16S ribosomal RNA. So, mRNA is having a sequence of bases. We call it as Shine-Dalgarno sequence. And anti-Shine-Dalgarno sequence is there on the RNA part. So, they will show the complementarity. They will bind. right? Now what's going to happen? Your charged tRNA will come. Your charged tRNA will come. So because it is the initiation, so it is definitely having formyl methionine without any doubt, because I am talking about the prokaryotes. So it will be formyl methionine. So it's simple. This thing will happen first. This thing will happen first. Anybody having any doubt? Anybody having any doubt? This thing is happening first. Yes or no? Yes or no? So how is it possible? Magnesium ion is required. Some initiation factors. Just say IF3 is required. So when you talk about the prokaryotic initiation, right, you will talk about certain proteins. IF1, IF2, IF3. Along with that, GTP molecules are required. Magnesium ion is required. So this IF1, IF2, IF3 is nothing. They are the proteins. They are the proteins. Understood? They are the proteins. So for binding of this, for the formation of this complex, magnesium ion and initiation factor 3 is required. Any doubt? Initiation factor 3 is required. Is there any doubt? Now that charged tRNA will come, right? Now that charged tRNA will come. And that charged tRNA, this is the first one, bache. This is the first one. So it is for formyl methionine. It is for formyl methionine. Am I clear here? It is for formyl methionine, right? So firstly ribosome and mRNA bound, and then comes the tRNA. So for this step, GTP, initiation factor 2, initiation factor 3 is required. These are the requirements. So for this step, GTP is required. GTP is required. Understood? Understood? So obviously, if it is AUG, here you will see UAC. UAC. That's how it will be. Now, bache, larger subunit will bind here. Now, right, finally at the end, larger subunit will bind. Clear, bache? Clear, bache? Clear, bache? Yes or no? Now, larger subunit will bind. So, when larger subunit binds, all initiation factors leave. Okay, they will leave. So these are the prokaryotes. That is why IF, initiation factor 1, factor 2, factor 3. If it will be for the eukaryotes, it will be EIF. E means eukaryotic initiation factor. Okay, so now you have the complete ribosome, 70S. So as I said, 3 sites are there. So bache, your first tRNA always comes at P site. Understood? When there is the initiation, your first tRNA always comes at P site, PYQ it is. Any doubt here? Any doubt here? Any doubt here? Hanji, any doubt here? है ना? Done? So first tRNA, your next tRNA, जैसे let's say here if you have UUU, so whatever is there on the mRNA, as per that the next charged tRNA will come. So next tRNA always comes at A site. It always comes at A site. Now understand this thing, right? Understand this thing. This is your first tRNA. You know that at 3' end amino acid is attached. This is your next tRNA, right? At 3' end amino acid is attached. Now they need to link, right? See, in very simple way I'm showing you. This is the exit side. This is the peptidal side. This is the A side. These sites are there on the larger ribosome. Now they will show what? Bond formation, peptide bond formation. Actually, when the second amino acid comes, I'm writing it as steps. Number 1, number 2. When the second tRNA comes, all of a sudden something happened. and because of that, the COH bond with this 3' end, it becomes weak. It becomes weak, right? This bond here becomes weak. So basically what is happening here, right? When this next tRNA with next amino acid come, this amino acid now, because the bonding gets weaker, so this amino acid gets separated from this tRNA and it will attach here. It will attach itself to the tRNA present on the A side. So P side pe, the tRNA which is present at the P side, that will be having nothing now, that is having no amino acid, and the tRNA present at A side is having 2 amino acid. It has formed dipeptide. This is elongation. It has formed dipeptide. And how is it possible? Obviously the bond formation. Just see. ये देखिए, so there will be the bond formation. So water will go out, dehydration will be there, water will go out, dehydration will be there, and you know that you will see CONH bond here. What are you going to see here, students? You will see CONH bond here. So which bond it is? It is peptide bond. And who's going to form it? Actually in the larger subunit of the ribosome, that 23S ribosomal RNA is there. In the larger subunit of ribosome, 23S ribosomal RNA is there, right bache? 23S ribosomal RNA is there. So it is, this RNA is actually like what? It is the ribozyme, right? Here it is act like an enzyme. This RNA act like an enzyme here, right? So this is also known as peptidal transferase. This is also known as peptidal transferase. What do we call it? We call it as peptidal transferase. What do we call it? We call it as peptidal transferase. So 23S ribosomal RNA is the ribozyme that will catalyze the bond formation here. Now this part is clear. This part is clear. This part is clear. Now what's going to happen? There will be the translocation. Translocation of ribosome. In a very simple language, in a very plain language, I'm explaining it. Your ribosome will translocate, or you can say that your ribosome will slide over mRNA. Your ribosome will slide over mRNA in 5' to 3' direction, right? In 5' to 3' direction. Your ribosome will slide over mRNA. Your ribosome will slide over mRNA in 5' to 3' direction. And which enzyme is used there? Translocase enzyme. Translocase enzyme is playing the role there. So it's not the mRNA that moves. It's not the tRNA that moves. Your ribosome will translocate. Ribosome will move. Your ribosome will translocate. Your ribosome will move. This part clear? Your ribosome will translocate. Your ribosome will move. This part clear? This part clear? Right? And for this sliding, GTP is required. For this sliding, GTP is required, and certain protein elongation factors are required here. Elongation factors are required here. Is that clear? Elongation factors are required here. Is that clear? Is that clear? Right? And that elongation factors in prokaryotes are 2TUSG, means EFTU, EFTS, EFG. What's that? EFTU, EFTS, EFG. Okay, that's what we have here. Okay, that is what we have here. That is what we have here. Understood? So translocation of ribosome will occur. Now see, what is going to happen due to this translocation? It's simple. It's simple. Your ribosome has translocated. Now that empty tRNA will come to, that tRNA without any amino acid will come to E side. The tRNA having dipeptide will come to P side, and next tRNA will come to A side. will come to A side. So that's how the translocation is helping. Are you getting my point? Now from E side, this tRNA will leave. Again in the same way, bond formation will come. Again, this ribosome will translocate. Again, this will come to E side, this will leave. So this process will continue till they find the termination codon, या stop codon or the nonsense codon. So which codons are that? Okay, you, ag, aga. Right, so this is your ochre, amber, opal. Bas. That's it. Okay, that's it. Done, bache. Right, so the linkage between last tRNA and polypeptide chain is broken. And how? With the help of release factors here, RF1, RF2, RF3, and again for this termination, GTP is required. Hydrolysis of GTP is required. Okay, bache. Okay, bache. Okay, so this is all about the translation. Trust me, it is simple. It is simple. So ye dekho. EPA, then there is a groove here from where this polypeptide chain will come out. Right, polypeptide chain will come out. And let me tell you, in eukaryotes, the 5' cap, it helps in binding of mRNA to smaller subunit of ribosome. Understood? To smaller subunit of ribosome. Understood? To smaller subunit of ribosome. Understood? Understood? ये देखो. So see this. So at least if you cannot make that complete checkerboard, if you cannot remember it, and in that, see, I have posted the individual videos on our channel, right? In that individual videos, right, the genetic code with trick is explained. So with the help of trick, you can remember all the amino acids written there. So do check that video, okay? So that's it. This is all about the translation. You can just look at the diagram here. The start codon is coming. It is the start codon is coming. The first one is coming at which side? It is coming at your P side. The second one is at A side. Okay. The second one is at A side. So these wonderful diagrams are also attached. So look at this summary. In the elongation, next is at this point. They will form the polypeptide, like ribosome will translocate. The one which is at A will come to P, and through this groove actually the amino acid comes, the protein chain comes out, right? So release factor at the ends, they help in the tata bio of this tRNA and this polypeptide chain, RF1, RF2, RF3. Okay. Okay, and further these proteins will be processed. Further these proteins will be processed. Done and dusted? Pakka? Sure? Sure? Are you sure? Done. So this is all about the translation. Okay, bache, this is all about the translation. So the next thing is the regulation of gene expression. Okay, regulation of gene expression. See, we will discuss regulation of gene expression. In the case of HGP, I will just highlight the PYQs, and DNA fingerprinting, obviously once you will understand the genetic engineering and all that part, it will be clear. So after this, we are directly going to start the biotechnology. Okay, bache? So 2 chapters are there in biotechnology, and 3 to 4 hours are sufficient to complete it, because that is not lengthy. If molecular basis of inheritance and processes are clear, in the genetic engineering, you don't have anything difficult. You need to understand what restriction endonucleases are doing, how are they cutting, and that complete steps which are given thoroughly in the NCERT. Done? Chalo. So regulation of gene expression. How a gene is going to express itself? Gene makes the protein. Gene make the protein. How a gene is going to express itself? Gene make the protein. Gene, mRNA, mRNA to protein. That is what we have. Now, bache, in the case of eukaryotes, at the transcription level, we can control it. At processing level, we can regulate that gene expression. Even when we transport the mRNA from nucleus to the cytoplasm, we can do it. And even at the translational level, we can do it. right? But in the case of prokaryotes, you don't have all that things, right? Transcription, translation is going on all together, so you cannot control it there. Done, bache? Done, bache? Yes. Is it clear? Sure. Is it clear? Is it clear? Sure. Very good. So in the case of prokaryotes, what do you understand? You talk, in the case of prokaryotes, what do you discuss? You talk about the operants. So the concept of operand is not there in the case of, it is not there in the case of prokaryotes. It is only there, in the case of eukaryotes, it is only there in the case of prokaryotes. Is that clear? So what is, how do you define operand? How do you define operand? So before starting this part, I'll tell you about the genes. Just say we have two types of genes. One is constitutive genes, and another category is of non-constitutive genes. One is of constitutive genes and another category is of non-constitutive genes, right? One category is of constitutive gene, another is of non-constitutive. Constitutive genes are your housekeeping genes. They are your housekeeping genes, and these are your luxury genes, right, bache? So when it comes to the constitutive gene, we always, cell always need their expression. Cell always need their expression. Cell always need these genes to express. Just say for an example, your genes for glycolysis, right? Glycolysis is the glucose breakdown into the pyruvic acid. The breakdown of glucose into the pyruvic acid. This is what we need. This is what we need. The breakdown of glucose into the pyruvic acid, right? And that's the first step of respiration. We need them. Non-constitutive luxury genes, they will only express when required. Only express when required. Only express when required. Is that clear? Chaliye, I'll give you the example now. Firstly, I'll ask you one thing, that how do you define operand? How do you define operand? What is operand according to you? How do you define it? Quick. हाँ जी, क्विक. वाह डीजे टिल्लू, बहुत बढ़िया. Quick. What is operand? Define operand. Define operand. Yes, please. Define operand. Now no response, है ना? Now no response. But when we need to spam, we can, है ना बच्चे? So it's a part of DNA, right? It's a part of DNA. Operand is a DNA segment having single regulated unit, right? It is the part of DNA. It is a part of DNA segment having single regulated unit and having more than one structural genes, one operator gene, one promoter gene, and one regulator gene. Is that clear? Is that clear? That's what we have here in operin. Is that clear? That is what we have here in operin. So it is the part of DNA, right, which acts like a single regulated unit, right, bache, and having more than one structural genes, one operator, one promoter, and a regulator one, right? Right? Okay. 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 Good. So now in the case of operin, see, remember in 1961, right, two scientists, they elucidated the operin in the case of E. coli. So when you talk about the operin, there are two words. So let's come to this part directly. So ye dekho, it is given here, the elucidation of lac operin. Lac stands for lactose. What is a lactose? It's a sugar. It's a disaccharide. It is made up of galactose and glucose. Remember, it is made up of galactose and glucose, right? So two scientists, Francis Jacob and a biochemist, Jake Monod. They were the one who elucidated the first operin. Scientist's name is important. It is again a previous year question. Right, scientist's name is important. It is again a previous year question. Clear, bache? Clear, bache? So what is the point here? They were the first to elucidate a transcriptionally regulated system. They were the first to elucidate that. They explained it in the case of E. coli. Right, they were the one who explained it. Right, bache? Right, bache? And they called it as operin. They called it as operin. So when you talk about the operin, so in 1961 they did so. Okay, in 1961 they did so, Jacob and Monod. Okay, so when we talk about the operins, we have two words. So anybody in the class who can explain it? One is inducible operin, one is inducible operin, and the another part is what? Another part is the repressible operin. So anybody in the class who can explain is inducible and repressible. But inducible and repressible. Inducible means to induce. Inducible means to induce, to start something. So here, by default, genes are not expressing. By default, genes are what? Their expression is off. So we have to switch it on. We have to switch it on. What we have to do? We have to switch it on. So by default it is off. We have to switch it on. And in repressible operin, by default they are on, but we have to switch it off. Inducible, repressible. Inducible, repressible. Now there is a very simple story. We talk about the bacteria, that is E. coli. But E. coli normally feeds on glucose, right? But in the case of E. coli is having the enzymes that can even digest lactose. But you tell me, if E. coli, it feeds on glucose, if it can digest glucose, and if there is the availability of glucose, so do you think that enzymes for the lactose formation should be active? Do you think the enzymes for the lactose formation, lactose hydrolysis should be active? Yes? Yes? Do you think it is required? Of course not. Of course not. Of course not. Now, in the system, in a culture, E. coli is growing. Now here, the food available is lactose. So obviously E. coli has to switch that on. E. coli has to start the expression of lac operin, right? So that it can digest the lactose and so that it can survive. Okay? Okay, that's the first point. Now, what's the next point here? Next point here is, see, everybody, Pipo ZYA. Repeat it. Pipo ZYA. Pipo ZYA. Pipo ZYA. I'll repeat the most common mistake here. P-I-P-O. Then you have ZYA here. Now, what's the meaning of that? Dekho, I here means regulator gene. I here means regulator gene. P means promoter. O means operator. ZYA, they are the structural genes here. They are the structural genes here. And this Z, this gene, it gives enzyme beta galactosidase, bache, and it breaks the lactose into glucose and galactose. Y gives the permease. Again, that's a PYQ. And A gives trans-acetylase. A gives trans-acetylase. Right, bache? So again, important it is. PYQs. Now understand, the sequence is this. Pipo, ZY. Pipo, ZY. Pipo, ZY. By default, what is happening? Normally, what is happening? This is the I here. It will form mRNA. That mRNA is forming the protein. This protein here is repressor. And this protein, bache, this protein is attached to the operator here. This protein is attached to the operator here. These lines are not given in NCERT, but I am explaining it because then only you will understand the topic in a better way. Promoter and operator, they are overlapping. Promoter and operator, they are overlapping. Promoter and operator, they are overlapping. What are they doing? What are they doing? They are overlapping, number one. Secondly, product of repressor, product of I gene, regulator gene, binds with operator. Matlab, whatever that I gene is going to form, it will bind with operator. Number three, your RNA polymerase, it binds with promoter. Doubts? Doubts? Any doubt? Doubt? Hanji, doubt? So promoter and operator, they are overlapping. Product of I gene binds with operator. It always interact with operator, and this is the one that is going to decide the switching on and switching off of the gene. Then RNA polymerase bind with promoter. Now, see, in your syllabus, you people study the negative regulation. What do you study? In your syllabus, you people study the negative regulation of lag operon or negative regulation of inducible operon or negative control of inducible operon. So this is the part in your syllabus, but we have the positive regulation also. So why do we call it as negative regulation? If regulatory genes, if it forms repressor, then it is the regulator, negative regulation. If regulatory gene, it forms the activator, then it is the positive regulation. If regulatory gene, it is forming the repressor, then it is the negative regulation. If regulatory genes, it form the activator, it is the positive regulation. Understood? Understood? So this repressor is attached here by default, and that's why what is happening? No transcription, no translation. So if I want to activate this, right, it is the negative regulation of inducible operon, but positive also exists, but that is not the part of our syllabus. Why negative? Because regulatory gene is forming a repressor. So if we want to switch it on, what we have to do? We should not allow the repressor to bind with the operator, because repressor is attached to the operator. RNA polymerase cannot bind with promoter. It cannot do the protein, it cannot do this transcription and translation. Now here in this case, what's going to happen? It's very simple, right? So if we want to switch it on, we need an inducer. So inducer here is lactose. Inducer here itself is lactose. And to be more specific, it is allolactose. That is the inducer here. It is the allolactose. That is the inducer here. So this particular inducer, this will bind with repressor. It will make it inactive. So if it is inactivated, it will not bind with the operator. Your RNA polymerase binds with promoter, and the polycistronic mRNA will be ready, and you will get these three enzymes. You will get these three enzymes. So lactose is the inducer here. That's the PYQ. Now, the point here is, what is the function of beta galactosidase? Bache, it acts on lactose and it breaks that lactose into galactose plus glucose. What is the role of permease? This permease allow lactose to enter within E. coli. Because if permease is not there, if imagine After Z, if there will be the stop codon here, then obviously the operon will not get active. Why? It will not induce, the expression will not be there, because it is the permease that is required to allow the lactose to come inside the E. coli to act, then it will act like the inducer. And A, the function of A is not very clear, but it helps in transferring the acetyl group. So it is somewhere helping detoxification or something. Okay, bache? Okay, bache? So that is how it is. Now, I don't know whether you have this question or not. Ki ma'am, if after the, right, in presence of inducer, if Y is going to give its expression, right, then how lactose can be the inducer? How lactose can enter in the cell? It's simple here. Always remember, some expression of permease is always there within E. coli. Some amount of permease will always be there in E. coli, which will allow the lactose to enter, and to act like an inducer and to start this transcription and translation, and further all the things will occur. Is that clear? Is that clear? So this is all about the lac operon. I told you negative regulation inducer. So that are the kind of questions you will get. Next is Human Genome Project, the most ambitious project for the sequencing of the human genome. Very important question here is the vectors used. BAC, YAC. Bacterial artificial chromosome, yeast artificial chromosome. Second question is the techniques. Express sequence tag and sequence annotations. Very important. So the project started in 1990, right? And with, right, because we have to sequence the human genome, and imagine we have so many cells. Each cell is having 46 chromosomes, right? And when I'm using the word genome, I'm talking about the haploid content. So even if you consider 3.3 into 10 raised to power 9 base pairs, so imagine in each cell we have to see, right? We have to talk about all the bases present in each cell, the expression we have to consider. Okay, so with this Human Genome Project, one science that emerged to keep all that data, that is bioinformatics. Again, a PYQ. Right? And it was very expensive project. So now when you talk about the goals here, to identify all the approximately 20,000 to 25,000 genes in human DNA, to determine the sequence of 3 billion chemical base pairs that make up human DNA, store the information, improve tools for data analysis, transfer that technology to other sectors like industry, and address all the ethical, legal and social issues that may arise from the project. Okay. Now here, when it comes to the methodology, these are the general things that you should need. That project was completed in 2023, but still, bache, right, the chromosome 1, its genes and all that sequencing was done in 2006. And other non-human model organisms, because that is also PYQ, they are bacteria, yeast, C. elegans, which is a free living non-pathogenic nematode, Drosophila, that is your fruit fly, plants, etc. They have also been sequenced, right? So this is again the PYQ. So one question for sure will come from HGP, right? So methodologies may EST, express sequence tags, matlab to identify all the genes that are expressed as RNA. So in this approach, we will only sequence the genes that has formed the RNAs. We are not talking about the entire genome there, right? Gene has expressed, matlab gene has formed the mRNA and then protein. Then comes the sequence annotation. So when you talk about the sequence annotation, the complete genome will be sequenced. The complete genome will be sequenced, including coding as well as non-coding sequences. This is important, right? So basically that Frederick Sanger sequencing techniques, they were used, right bache? And here you guys can see the fragments were sequenced using automated DNA sequencers, and they worked on principle of method developed by Frederick Sanger. So these sequences were then arranged based on some overlapping regions present in them. And it's not possible, bache, let me tell you, it's not possible to take out that entire polymer of DNA and to check it. Obviously, we have to break the DNA into the fragments first. Then the vectors will be used so that we can clone that particular fragment. And after that, they will be sequenced. And when they were sequenced, overlapping regions will be there, which will show the common origin of that gene. Accordingly, they'll be arranged. Accordingly, they'll be given to the particular chromosome. So it might seem easy, but it is not. Right, bache? Okay. Okay. So the which Vectors were used. That is also PYQ. The techniques PYQ, Frederick Sanger. Right, this point is important. So required generation of overlapping fragments for sequencing. So alignment of these sequences was humanly not possible. So specialized computer-based programs were developed. So these sequences were subsequently annotated and were assigned to each chromosome. That's it. Okay, ji? So sequence of chromosome 1 was completed in May 2006 and we have total 24 chromosomes now. X and Y separately, they were also sequenced. Okay, bache? They were also sequenced. So after all that, what we got to know? Yes, so many things. Jaise ki ma'am, for an example, the number of genes, the total number of genes that are present. Am I right? The total number of genes that are present. And we got to know that less than. Right, if I talk about the features, total number of bases, total number of genes, it was clear. So human genome contains what? Human genome contains, yes please, 3167.4 or 64.7. Quickly tell me, it is given in NCERT. 316.4 or 3164.7 या फिर 3167.4. What should be there? Yes बच्चे, what should be there? What should be there? Be quick. अरे यार, million. The word is million, not billion. Nucleotide bases are there. Right बच्चे? So human genome contain 3164.7 million nucleotide bases. Number one, it's not billion, it's million. And average gene is having 3000 bases. Right, bache? So average gene is having 3000 bases. And largest human gene is, bache? Human gene is dystrophin gene. You remember? Dystrophin gene is the largest human gene. So from this part, see, this is something you need to read from NCERT by yourself. I'm not going to discuss it because this is completely theoretical, and yes, you have to remember it. Right, bache? Yes, you have to remember it. It is important. Done, bache? It is important. Done? Done, bache? Okay, so the most important information that I'm going to tell you here is that because of this we got to know that less than 2% of the genome is providing us the functional information, or providing us the information for protein synthesis. And from this HGP only, we got to know that 99.9% we are same and there is a difference of 0.1%. With this HGP, with this sequencing, we got to know that there are near about 1.4 million locations in our genome where there is a change in only single base pair. Right, you remember SNPs, single nucleotide polymorphism, right bache? It is single nucleotide polymorphism. And it is pronounced as SNPs, right? So SNPs is different, SNPs is different, SNP is different, SNP is different. Clear bache? Sure? Yes please. Then let's go, let's discuss the biotechnology, Didi. So yes, up to this part, is it clear or not? Up to this part, is it clear or not? Sure? So do you solve the questions? Yes, bache, do you solve the questions? Let me ask something. Just a minute. So, do you have any doubt here? Firstly, tell me this. Do you have any doubt here? Done. So these points should be clear. Single nucleotide polymorphism, and with this you know that DNA fingerprinting. Okay. Okay, bache. So here you talk about the repetitive DNA sequences. So any doubt? Any doubt here? Any doubt? Are yaar, what will you do with the break now? Let's stretch it. Let's discuss it. And then let's start the biotechnology. Let's start the biotechnology. You people are crying. Yeah, ma'am. Chalo, break is required. Chalo, theek hai, it's okay. So DNA fingerprinting we will discuss with this. What do we say? Biotechnology part. Okay, so this is the chapter that we are going to start, and you know that this chapter is not very lengthy. Okay, it's a very simple chapter. It's a very simple chapter, and here I will ask few questions from. It's a simple chapter, and we will complete it within next 4 hours. You can keep it after break. 4 hours may we will be able to finish it. Okay, chaliye, let's have a break. Then you will not get break for a long time. So break time till 7:45 p.m. Okay. What do you want? Do you want a break for 10 minutes, 20 minutes, या फिर half an hour का dinner break? Let's keep it as a dinner break now. Then we will discuss it. Hello kids. Hi. So let's restart. Hi guys. So let's restart. Welcome, welcome, welcome. So, how are you? How are you? How are you? Good? No, ma'am, very good we are. Chaliye, so let's continue this chapter. Let's talk about the biotechnology. And guys, this is something that I always suggest, that after MBOI, do revise biotech, right? You will find it easy to read it then, okay? Trust me, you will find it easy to read them. Okay, so biotechnology principles and processes. So I will start from the, I'll give you a basic introduction here, right? I will give you a very basic introduction here. So when you are talking about the biotechnology, the word is simple. The word is simple. Bio, living, technology, technique. Basically, we are mixing the living organism and technique. And why are we doing it? Why are we doing it? It is simple. Why are we doing it? It is simple, because we want something good for ourselves. We humans are selfish, right? We want to have the products that can give benefit to us. It can be any enzyme, it can be any protein, it can be any drug, it can be anything. So basically we are mixing the technology, right, in such a way that we can get better organism, or we can get better proteins or better things from that organisms. That is what it is, right? That is what it is. So now when you talk about the biotechnology, right, how can we define it, or how can we, yes. So what do you understand by this? 1 minute. Biotechnology, yes. See, there are two views here. The traditional one, or I can say the old one, or the modern biotechnology is there. So do you know anything regarding that? Do you know anything regarding that? And here in this particular chapter, in the starting, there is an introductory part in the NCERT about the scientists. So I suggest you people to read about it. Okay, I suggest you people to read about it, about the Herbert Boyer and Stanley Cohen. So that's my advice, right, that you should read about it. Okay, chaliye. So what am I talking? I'm talking about the biotechnology, and we are discussing that there are two views here, or there are two types here, the old and the modern one. The old and the modern one. What do you have here? The old and the modern one. So what is this old biotechnology? What is this old biotechnology? Right, what is this old biotechnology? So when you talk about this old biotechnology, here the point is simple. Here we are talking about the natural capabilities of organisms. Right, natural capabilities of organisms. So I am not going to write it, because everything is in NCERT. I will explain it here, right? So this is again a conceptual chapter. The concepts need to be very clear here. The concept needs to be very clear here, right, bache? So what is the point here? The word is old and the modern biotechnology. So for the modern biotechnology, you need to know about the recombinant DNA. So let me discuss the things here. See, so they are saying the natural capability of organisms, we are exploiting it. That is also coming under the biotechnology, but that is the old view. Just say for an example, do you citric acid, we get it from living organism, right? Can you name the living organism which is giving us the citric acid? Yes. Can you name the living organism that gives us the citric acid? Yes, bache. Please name the living organism that gives the citric acid. Yes. Yes, bache. You people take a lot of time to answer a question. Not acceptable. Guys, please. So what am I talking? I'm talking about the citric acid. So yes, you are getting the citric acid, right? So from which organism you can get the citric acid? Excellent, bache. Mandadi, maybe. Mandadi. So it is from a fungus. That is your Aspergillus niger. Aspergillus niger, and other than that acetic acid, right? Other than that, let's say the clot buster, right? The streptokinase. You can also take the example of lactic acid bacteria. So what are we doing, right? You can take the example of yeast. So we are exploiting the natural abilities of the organisms. What are we doing? We are basically exploiting the natural abilities of the organisms. What are we doing, guys? We are basically exploiting the natural abilities of the organisms, and right, we are using them for our welfare. Now when it is the modern biotechnology, the recombinant DNA is the word, rDNA is the word. So basically the DNA will be modified here. Dekho, before that we also know about, you need to know about the hybrid and the something known as GMO. What is the meaning of GMO? GMO means genetically modified organisms. What is the meaning of GMO? Genetically modified organisms. And what is the meaning of hybrid? See, when you are talking about the hybrid only, right, here you will take a superior male and a superior female, right? And obviously when we hybridize superior male and superior female, we are going to see the superior characters. We are going to see the superior characters in the next generation. This is the meaning of the hybrid. But in the case of a hybrid, right, my dear students, in the case of a hybrid, right, no doubt you will get the superior characters, but along with that some undesired characters will be there. Along with that, the undesired characters will also be there. Yes or no? The undesired characters will also be there, right? You are not going to see any new trait here. You will just see the traits of these two parents here. When you are talking about the genetically modified organisms, you are talking about the gene manipulation here. You can introduce a foreign gene here. When I'm saying foreign gene, so it doesn't mean that this gene is coming from America, Australia. No, it simply means that that particular gene, it doesn't belong to that particular species, but still it has been introduced there, right? The foreign gene. So on the basis of that, we say the organisms, they are transgenic. I hope you know. So in the genetically modified organisms, you can introduce a new gene, you can see a new trait, the desirable one, and even some traits can also be eliminated if we don't want that. Okay, so this is what biotechnology is, the old view and the modern view. The old view and the modern view. And when you talk about the modern view, there are other things also. Just I am telling you about the rDNA, the genetic engineering. I'll give you one example, IVF, in vitro fertilization, IVF, in vitro fertilization. So when you people talk about the in vitro fertilization, when you people talk about what? When we talk about the in vitro fertilization, what's the word there, guys? What is the word here? It is in vitro fertilization. What is the word here? It is in vitro fertilization, right? So basically all that things, they are telling us about the technology, the way we are using the technology for our betterment, right, bache? So this is the old and the new view, old and the new view. Now, what is the next thing you need to know? You should know about EFB, that is European Federation of Biotechnology, right? European Federation of Biotechnology. What is this? This is EFB, European Federation of Biotechnology. So when I'm saying European Federation of Biotechnology, it has given a definition of biotechnology, right? And what is it considering? It is considering both the traditional view or modern biotechnology. So it is like integration of natural sciences, right? Integration of natural sciences and organisms. So when you are saying natural sciences, what do you include? All that physics, chemistry, biology, you are including everything there. So that natural sciences are integrated in such a way that from the organisms, right, from the organisms we are getting the better products, right? The products, we are getting the better products. Plus, by using these sciences, we are integrating it in such a way that in the lab also, we can synthesize the things, some molecular analogs that are there in that organisms, we can make them better, right? So if I have to conclude it, that's how we can define. That's how we can define. And why are we doing it, right? Why are we doing it, right? Obviously for our betterment, for our betterment. So MCQ can come from this definition and from this particular point also. Okay? So now when you talk about the rDNA technology, this is happening here. You will have a donor DNA, right, which will contain your gene of interest. You have to cut it. You have to fix it with a plasmid. Plasmid here is what? It is a vector. That's the term that we are going to use. Vector is a vehicle DNA, right? Bache, what is vector? Vector is a vehicle DNA. What is vector? Vehicle DNA, right, bache? right बच्चे? Vehicle DNA it is, vector, and we can also call it as gene taxi. We can also call it as gene taxi, right बच्चे? So it is the vector, it is the vehicle DNA, and it is also known as the gene taxi. So it will join here, and that's what we are going to get, recombinant DNA. So when this DNA is introduced into a specific host, into a competent host, this DNA will multiply here, and we will see the multiple copies of that particular gene. That is gene cloning, and obviously the gene will express. So basically this complete process, that how can we get our donor DNA, how can we fix it with the plasmid, how can we make the multiple copies out of it, that is what we study here in the recombinant DNA. Is that clear? Is that clear? Is that clear? Good. So when you talk about the principles of biotechnology, there are two things here. How many things are there? There are two things here. One is genetic engineering, another is bioprocess engineering. You can also call it as chemical engineering. You can also call it as what? You can also call it as chemical engineering, right? Chemical engineering. So what is the point here? Yes, bache, Bhavya, Shubhan, Arpana, what is the point here? See, principles of biotechnology, and here these are the principles of modern biotechnology, because in the old one we were just exploiting the natural abilities of the organisms. What were we doing? We were just exploiting the natural abilities of the organism. So what is the point here? The point here is very simple. Number one, the genetic engineering. Right? The techniques that can alter the chemistry of genetic material. The techniques that can alter the chemistry of the genetic material, be it RNA or its DNA. Be it RNA or its DNA. So techniques that can alter the chemistry of genetic material, it is the genetic engineering. And that will be introduced into the host organism, right? So obviously, bache, if a modified gene is introduced, your modified DNA is introduced into the host organism, right? Don't you think it will change the phenotype? Yes, Mr. Tillu. Angie, Anshika, don't you think it will change the phenotype? Phenotype means what? Phenotype means what? Physical appearance, right? External appearance, morphological character. If the gene is changed, altered, the protein it will make will alter, then obviously it will give a different phenotype to that host. Now, okay, fine, you have altered the gene, right? You have introduced into the host. Now you need that condition where you can maintain that host, right? You literally need the conditions in which you can maintain that host, in which you can do what? You can maintain that host. So maintenance of sterile, right? That is microbial or contamination-free ambience. So what is the point here? The point here is maintenance. Maintenance of sterile, that is infection-free ambience in chemical engineering processes, right? So that to enable the growth of only the desired microbe. So one thing is we have to alter the DNA. Second thing is we have to maintain the conditions by which, what can we do, bache? By which, what can we do? We can just maintain that conditions by this chemical engineering or bioprocess engineering, so that that particular desired microorganism or host can survive, and we can get the large quantity for the manufacture of that particular product. It can be anything. It can be antibiotic, it can be enzyme, or it can be any vaccine. Is that clear? Is that clear? Now when you see this particular paragraph from the NCERT, this particular paragraph is explaining something very simple, right? Guys, are you okay with the speed? Are you okay the way I'm explaining it directly from the NCERT? Yes, right? Directly from the NCERT. Is that okay for you? Please tell me, is that okay for you? If it is a yes, show some energy in the chat section, guys. Okay, so now what's the point? What are they explaining here? Something very simple, right? They are explaining about the advantages of sexual reproduction over asexual reproduction. Obviously, there are advantages. The very important thing is the meiosis, the gamete formation. And during that meiosis, the crossing over will occur, the recombination will occur, and that recombination is important because it is giving the variation. And that variations are also important to survive in that changing environment, right, bache? Right, bache? So if we want to preserve the genetic information, this line is very important, right? So asexual reproduction is going to do what? Asexual reproduction is going to preserve the genetic information, but when it comes to the sexual reproduction, the new changes will be there, right? The new combination will be there, the recombination will be there. Is that clear? And that recombinations will result in the variation, right, bache? It will result in the variation. Clear, bache? Clear, bache? So when it comes to the genetic engineering, what will be done? rDNA, recombinant DNA. So if recombinant is the word, so basically in the lab, the DNA that we have, we are going to introduce there the gene of interest, and that's how we will get the recombinant DNA. Then we will use gene cloning. Right? Why? Because we want the multiple copies of that suitable gene, and then we have to transfer it into a particular host so that it can express. So bache, yes bache Shizuka, yes bache Anshika, yes bache Shubhan, everybody here in the class. So you tell me that how a gene is going to express itself. How a gene is going to express itself? By making proteins. I know you guys are tired. I know you must be thinking, hi madam, 7 or 8 hours just for one chapter, but that chapter is important, guys. That chapter is important. DNA fingerprinting will be discussed here. So if you watch this particular lecture only to master that chapter, trust me, after the class, read the NCERT, you'll be done. You'll be done. 6 to 7 questions will be done and dusted. Biotech we are going to complete. So I am just requesting 3 marathons from class 12, 3 marathons from class 11. Right? Because when it comes to ecology, it's very basic. It's very basic. In 5 hours, within 5 hours, or I think within 5 hours, in detail we can cover the ecology. 5 hours, human reproduction, complete reproduction unit. 6 hours, let's say. 6 hours, let's say. Right? So in 10 hours, in next 10 hours, the human reproduction, the complete reproduction unit and the complete ecology can be discussed. Again, speed, we have to maintain a speed. Then principles of inheritance and variation, your microbes in human welfare, human health and disease, they are also evolution, right? These 4 chapters, they are also lengthy, not very lengthy, but right, but then we have to complete it. So 3 marathons and the syllabus will be done and dusted. What's the issue? What's the issue? Right? I know you guys are tired. I know we feel bad ki yaar, why we have to study that much. So imagine why we have to take the marathons, why we have to stand for 10 to 12 hours, why we have to teach the same thing again and again. We have already posted the same on this channel, right? But still we understand the importance of the live lectures, right? We still understand that some people, they are not able to connect with that lectures that we have already taken. So it's fine. It's fine. Not just you people are working hard, even we are also working hard. Right? Even we are also working hard, and trust me, it's too cold here. Okay? It's too cold here. And trust me, even I am tired. I am so tired. Even I want to take rest. Even I want to eat my food peacefully. I just don't want to see the watch. Arey yaar, theek hai, my class is about to start. Go. Okay? Okay, so it's okay. It's fine. It's okay. It's fine. Right? I chose it. I wanted to be the teacher. I wanted to be the educator. I was like, I want to do something. I want to teach on YouTube. I want to teach free of cost. Right? I want to help those who cannot afford the education properly, who are not able to go to the, go to the offline institutes where they have to pay the lakhs. They are here. They are trusting us. Right? I don't want to be a YouTube educator who's just coming, who's just giving you a feeling. Okay. This is what we have covered. This is what we have covered. This is what we have covered. No. The thing should be real. If you have given me your 7 hours, then trust me, just check that chapter from the book. Right? You will understand, you will decode NCERT completely. Right? All the PYQs will be answered, and not just the PYQ, even you will be able to answer the new questions if you practice. So that's fine. You decided it. Hannah, you decided to be the NEET aspirant. You decided to crack the examination. You decided to be a doctor. We have to pay for it now. No pain, no gain. This is what I believe. No pain, no gain. Isn't it, Hannah? So if we have decided something, we have to work hard for that. We don't have choice then. Right? I can even switch my career, but I enjoy what I'm doing. So I have to put efforts. I cannot cry all the time. Trust me, sometimes it's very draining to take back-to-back classes. Trust me, it's draining sometimes. Right? I'm not going to lie, I feel so good. Yes, I enjoy my classes, but sometimes I feel tired. Right? 4 years back, I was like, yaar, one more class. Yeah, I was young, now I'm also oldie. Right? So we feel motivated, feel good. Chill. Hi, HSP sir, you are also here. Thank you, sir. See, HSP sir is also there in the class. So next marathon, HSP sir is going to take, I think, day after tomorrow. So be a support, right? It feels very bad when we teach for so long and students they just keep chatting. They don't respond to the questions there. Okay. I'm oldie, yaar. I'm oldie. HSP sir is younger than me. HSP sir is younger than me. He's just 22 years old. He's just 22 years old. So guys, chill. It's fine. It's okay to feel tired. It's fine. Even I am tired, but theek hai. We have decided it. Then let's go for it, na? Right? And as I said, I cannot be a teacher who's just coming here, who's just giving you a feel that, okay, you have completed everything, but in reality, you don't know anything. So I'm sorry, I'm going to take the time. Right? I will take time. Okay? So if you're not happy, please don't waste your time. Maybe you don't resonate with my thoughts. That's okay, that's fine. But if you are like, nahi yaar, madam, let's do it. Let's do it sincerely. 3 marathons I'm expecting. Right? And 3 marathons means 30 hours. Even I can finish it in 1 or 2 chapters. Shh, completed, Ambika, leave it. No, I don't want that. Okay, so let's focus here. Let's go, guys. And you know that you can go to any video of our channel. See in the description box there is a link. This is the link for our fast track reloaded batch, and in that batch also I teach with the same energy, right? So be the part of the batch, right? Revise properly. Practice question, give test. Now come back. So when we are talking about the biotechnology, what you have to do, you need a recombinant DNA, you need to clone that gene, your gene of interest, then you have to transfer it into a host organism, and then what do you need? The recombinant protein. That is what all we require, right? Why are we altering a gene? Why are we changing a gene? Why? Because we want a particular protein that a gene is making. Right? This is what we need. Right? That's our need. Am I right here? So that's what we have to do in the genetic engineering. Right? So here they have given some general things. Okay, that D piece of DNA would not be able to multiply itself in the progeny. Chalo, I'll ask you the question. Okay, I'll ask you the question. Here you have a cell. This cell has picked a genetic material from the surrounding. That is how it is present. So what do you think? This gene of interest, this DNA or this gene of interest, is it going to express itself? Yes? Is it going to express itself? Excellent bache Tina, excellent bache Shubhan. No, because how this gene will express itself? This gene has to be the part of this genetic material. Why? Because this genetic material is having ORI site. You know that if there will be no origin of replication, the replication will not be there, the DNA will not replicate, and if it will not replicate, then you tell me, will it be able to express itself? Will it be able to show the cloning? No. Will it be able to show the cloning? No. So basically what is written here in the NCERT? It's very simple that they are saying that when the gene of interest, it gets integrated, right? It becomes a part of genome, then it may multiply, it may be inherited along with the host DNA. Right, definitely then it will multiply and then it will get inherited to the next generation. Okay, bache? Right. So origin of replication is something very important, because of this origin of replication, application is possible, and then that genetic material can pass to the next generation. Is that clear? So alien piece has to link with the chromosome, with the origin of replication, so that it can show the cloning. Is that clear? Now here they are discussing something regarding the artificial chromosome. Now we will discuss some other thing. Acha, who's the father of genetic engineering? Please tell me, who's the father of genetic engineering? Who is the father of genetic engineering, people? Anybody? Very good, very good, very good. So basically, father of genetic engineering. Now I'm going to talk about certain scientists. So father of genetic engineering is Paul Berg. What he did, he transferred a gene from a virus into the bacteria with the help of bacteriophage. Okay. Now I am going to discuss about, yes bache, I am going to discuss about some other things that basically I am going to discuss about the foundation of biotechnology. What I want to discuss here, bache? I want to discuss about the foundation of biotechnology, right, bache? You can mark it. You can mark it. So whatever I am going to discuss, it is there in NCERT, but the extra points that I am going to discuss here, they are still in NCERT. When you read the, the part where this unit is starting, the Herbert Boyer, about the Herbert Boyer is given now, about the Herbert Boyer and Stanley Cohen. That's the thing I'm going to discuss. So I will not write it. You can just see it from the NCERT. I will discuss that thing, right? So what am I talking? I am talking about the foundation of biotechnology, and here there are two discoveries. One is presence of plasmid in bacteria. One is presence of plasmid in bacteria. This is the number one thing. Presence of plasmid in bacteria. Number two is restriction endonucleases that can be used in restriction endonucleases that can be used in vitro. So what is the meaning of that can be used in vitro? In vitro means that I can use in the lab. These two things are the foundations of biotechnology, right, bache? These two things are what? They are the foundation of biotechnology. Now we have to discuss about the scientist here. So what are we going to discuss? We are going to discuss about Herbert Boyer. So in 1969, Herbert Boyer, what he studied? He was studying the restriction enzymes of E. coli. What he studied? He studied the restriction enzymes of E. coli, your Escherichia coli bacteria. And what he discovered that these restriction enzymes, they cut the DNA in a particular fashion. What he studied, bache? Firstly, scientist name is important. That Herbert Boyer, he studied the restriction enzymes of E. coli, right, bache? And he observed that these enzymes can cut the DNA strand in a particular fashion, in a particular way, right? So Herbert Boyer, what he studied? Restriction enzymes of E. coli. And he found that they cut the DNA in a particular fashion. They cut the DNA in a particular fashion, right? And particular fashion means they cut the DNA in a particular way that what will be obtained? Sticky ends will be obtained. What will be obtained? Sticky ends will be obtained. Means they are the complementary overhangs. You already know A will form 2 hydrogen bonds with thymine, C will form 3 hydrogen bonds with guanine. So the DNA is cut in such a way, I'll show you that also, right? That the strand, the fragments that we are going to get, they are also complementary. Like if here I have A, here I have T, whenever I'll bring it closer, they'll form the hydrogen bond, and then DNA ligase, molecular glue, will join it. This is the first point. This is the first point, right, bache? Then Mr. Stanley Cohen, he also studied something, and as I said, everything is given in NCERT. Read that introductory part. So he studied that there are certain ringlets in cytoplasm of bacteria. What are that ringlets in cytoplasm of bacteria? Yes, please name it. That is the circular DNA. What's that circular DNA? That's plasmid. What's that circular DNA? That's plasmid. What is that circular DNA? That is plasmid. Yes. What is it? It is plasmid. So what are we talking? We are talking about the Stanley Cohen, who studied the small ringlets of DNA known as plasmid, and where these plasmids are present in the cytoplasm. And obviously these ringlets are free floating. So he observed that he developed a method by which he can just remove that ringlets from the cytoplasm of the prokaryotic cell. Is that clear? Is that clear? Is that clear? So he noticed one thing, that the ringlets are present. These ringlets are present in the cytoplasm of bacteria. We are calling it as plasmid. These ringlets are free floating and they are self-replicating. They are self-replicating. Now, what is the meaning of self-replicating? Yeah, देखो. If you know it, good. If you don't know it, please listen. In the bacteria, I'm not saying in all the bacteria, nucleoid is there, and plus there is the presence of circular DNA. We call it as plasmid. We call it as plasmid. And this plasmid, definitely it is also having the genes. And that genes, when they express, they also provide a particular phenotype to this particular bacteria. Jैसे we have in the plasmids, antibiotic resistant genes are there. In the plasmids, drought resistant genes are there. Right, that type of genes are there. So he studied that these plasmids, they are self-replicating. Right, they are self-replicating. They are independent of chromosomal DNA. Means they are right, they are independent of chromosomal DNA. Means their replication, it doesn't depend upon the nucleoid. Why? Because they have their own ORI site. So he developed a method by which we can remove the plasmid. Is that clear? Is that clear? Is that clear? Yes. Is that clear? So now you have two discoveries. One is of Herbert Boyer and another is of Stanley Cohen. So if we can combine these two discoveries, one is of Herbert Boyer, we have some enzymes which cut the DNA in such a particular fashion that you get the complementary overhangs. Number two, you have a plasmid. That plasmid can self-replicate. That plasmid is not waiting for the chromosome, waiting for the nucleoid. Please help me in the replication. That plasmid is like, okay, fine, I'm self-dependent. I'll replicate myself. These are the two things. Okay, that I'll replicate myself. These are the two things. So these 2 points clear. These 2 points clear. Sure? Sure? These 2 points clear, right? Very good. Now if we combine both the things, don't you think that recombinant plasmid can be formed? Obviously. So now let us focus on the first instance of the construction of artificial rDNA. So construction of first rDNA. So don't you think the question can come from this part? Don't you think the question can come from this part? So Stanley Cohen and Herbert Boyer, basically they combine their discoveries. They are like, we have enzyme which can cut a DNA particularly, and we can cut the plasmid also with that enzyme. We can join them and this plasmid can replicate. The gene cloning can be done. Scientists, they can think like this, right? They can think like this. They are not the one just sitting like this. Arey yaar, why? Please complete a lengthy chapter in two hours or complete a lengthy chapter in 30 minutes, right? No, they are scientists. They think, they are like, okay, we are ready, let's think about it, let's do something new. So this is what they have done, right? This is what they have done. So they are like a construction of first rDNA. It emerged from the possibility of linking a gene. Which gene? Antibiotic resistant gene with a native plasmid, right, bache, of Salmonella typhimurium. Important. Bacteria name is important. So there is a bacteria, Salmonella typhimurium. What they did, they first, right, they just first recombinant DNA emerged from the possibility. What possibility? That how to link a gene which is encoding antibiotic resistance, right, with the native plasmid of Salmonella typhimurium. So Stanley Cohen bache and Herbert Boyer, they accomplished this in 1972. Year is important here. Year is important here. So Cohen and Boyer, they isolated the antibiotic resistant gene by cutting out a piece of DNA from a plasmid, right? Right. So they basically isolated from that plasmid, they isolated the gene which is providing the antibiotic resistance, right? And cutting off gene at specific location, it was possible. Why? Why? Because of the molecular seizures. Right, because of the molecular seizures that are your restriction enzymes. So they are also known as chemical scalpels. Done, bache? So the cut piece of DNA was then linked with the plasmid. Okay, so now this plasmid is what? It is a vector. What is the meaning of vector? Vector mane gene taxi. What is the meaning of a vector? Vector mane gene taxi. Is that clear? Gene taxi. Is that clear? Right, जैसे here they are giving the example, mosquito, female Anopheles, can transfer the malarial parasite. Malarial parasite is the one that is causing the malaria, but who's transferring it? Mosquito. Mosquito. ठीक है बच्चे. So this point clear. So plasmid can be used as a vector to deliver that alien piece of DNA into the host organism. So then linking of antibiotic resistant gene with the plasmid vector, right? It was possible with the help of DNA ligase. Right बच्चे. So this make a new combination of circular autonomously, means self-replicating DNA, and it is the recombinant DNA. Right, so they transferred it into the E. coli. So they took the plasmid, right bache, they got the antibiotic resistant gene, they combined it with the vector, and then they finally introduced that recombinant DNA into the E. coli. Is that clear? So this plasmid replicated there in that E. coli. Okay, it made the multiple copies. So this is about the gene cloning. That is how we obtain the multiple copies of antibiotic resistant gene. Understood? So this is the story of the first rDNA. Clear bache? So what we have to do here, the steps and sometimes their sequence, they are important, right? Just in 2023 one question was there regarding sequence. So two options were correct there. So these sequences and all, they are also important to understand. So identification of DNA with desirable gene, number 1. So because you should have that gene na, you should have your desired gene first na, that is important. So firstly you have to identify that, then you have to introduce into the host, right bache, and then you have to maintain that gene, introduce DNA into the host, and transfer of DNA to the progeny. These are the things that we have. Okay, so let's start the tools of rDNA. So even in the steps part, these tools are given. So with this portion only, we'll complete everything. So what is the first word here? Tools, right? So you tell me, you have a cell, you want to take the gene out of it, you want to take out your desired gene. Don't you think firstly you have to break the walls? Right? Firstly you need to reach, right? You need to reach at the level of that DNA, right? Hanji, tell me. Don't you think so? That is more important. Obviously that is more important, na? That is more important, of course, right? So when you talk about the tools of our DNA, firstly we will start with the enzymes. And very first thing that you need to know is the lysing enzymes. So lysing enzymes, they are for the breakdown. Like if it is a bacterial cell, lysozyme will be used, right? For bacterial cell. For if your gene of interest is in the bacterial cell, if you have to break the cell wall there, go for the lysozyme, right? Cellulase and pectinase, and it is used for the plant cell. It can break the cell wall of that plant cell. And chitinase is used for what? Chitinase is used for the fungal cell. So first thing is the lysing enzyme that we need. Second type of enzyme, second category is of the cleaving enzyme. And when we talk about the cleaving enzymes, we talk about the nucleases, we talk about the exonuclease. Yes, bache. We talk about the exonuclease and we talk about the endonuclease. We talk about the exonuclease here, we talk about the endonucleases, right? So cleaving enzyme, the one that are going to cleave our nucleic acid. Any doubt here? It's simple. If the word is exo, even in the molecular basis of inheritance, I told you, what will be the story here? Exonuclease. Yes, bache. Quickly tell me. Yes, please. You tell me. I'll eat a toffee till then. Hanji, what is the function of exonuclease? Yes, it will cut the ends. Exactly. Endonuclease inside, right? Endonuclease inside. So exonuclease will make the cuts at the end. Exonuclease will basically do what? Exonuclease will basically cut the ends. What will it do? It will basically cut the ends. Exonuclease, right? So if it is going to cut the ends, do you think that it will be helpful for us in genetic engineering? Of course not. Of course not. It is just cutting the ends, the terminal parts, If it is the endonuclease, endo meaning inside, it will make the cut within the DNA strand. It can cut single strand or both the strands of the DNA. Usually exonuclease will make the cut if there is a space in between two ends. Yeah, in between a segment, if there is a space, if there is a gap, it can make cut there, or at the ends it can make cut. But it can cut both the strands, single B and double B. Is that clear? So they cleave one strand or both the strands of double helix, right? Right. So because of endonuclease, if we get desirable fragments, we can use it in genetic engineering. So here, what is more important for us? We have one more category here, restriction endonucleases. Under the exonucleases category, we have restriction endonucleases. What is the word here? Restriction, right? So it will cut the DNA, but in a specific way. How will it cut it? In a specific way. From where are we going to get these restriction endonucleases? Definitely from prokaryotes. We can get these restriction endonucleases. They are the enzymes, right? And it's not mandatory that one bacteria will give you only one enzyme. No, it's not like that from one bacteria you will get only one enzyme. Right? It is not like that from one bacteria you people are getting only one enzyme. No, it's not possible. It is not like this, right? From one bacteria you can get many restriction enzymes. Okay, bache. So they will cut the DNA. They cut DNA in a specific way. We obtain it from prokaryotes. Okay, bache. Now I'll give you the example of one restriction enzyme, EcoR1. I will give you the example of one restriction enzyme, EcoR1. As I said, we have to study for our exams, so we will move in that particular way. So obviously it is very important for your NEET. It is a PYQ. So I am giving you the example of restriction enzymes, and I'm telling you that they will cut DNA in a specific way, in a particular way, and they are obtained from the prokaryotes. I will give you the example of restriction enzyme EcoR1, and there is a logic behind its nomenclature that also we have to discuss. First of all, when you talk about the restriction enzymes, but they recognize the sites that they cut, the sites where they make the cut, that are known as cloning site, or we can call it as restriction site. What are they? They are cloning sites, they are known as restriction sites, and sometimes we also call it as recognition sites. They are the cloning sites, restriction sites, and the recognition sites. They are cloning site, restriction sites, and the recognition sites. So EcoR1, right? So they cut the DNA at cloning sites, restriction sites, and the recognition sites. Okay, bache? And what are they going to recognize? Restriction endonucleases. They recognize the kind of palindromic sequences. What is the meaning of palindromic sequences here? It's simple. I'll give you the example. The word mom. If you are reading it from this side, the spellings are same. Mom. Radar. Dad. Ilam. See, from both the directions, if you read it from this side to this side, left to right, right to left, it's all same. Left to right, right to left, it's all same. That type of sequences are palindromic sequences. Now in the case of these restriction enzymes, as I said, I'll give you the example of EcoR1. But it's a previous year question, even in NEET 2020, 2021, this question has been asked. It recognizes this sequence, GATTUS. I call it GATTUS. It is GAATTC. GAATTC. Please repeat. GAATTC, GAATTC. 5' end is having G, 3' end is having C. 5' end is having G, 3' end is having C. G. So basically, जैसे we have the networks 3G, 4G, 5G, 3G, 4G, 5G. That's how you can keep it in your mind. So it is that is GAATTC. So 5' end near to it, but the 5' end is having G. So if in the paper they are mentioning 3' end, then they are saying GAATTC, that is incorrect. If they are mentioning 3' end and then they are saying GAATTC, that is incorrect. Is that clear? That is incorrect then. Is that clear? That is incorrect then. Is that clear? So it's GAATTC. Understood? GAATTC. Understood? Sure? Sure? Okay. So eco R1. Now see, if you are reading it from this side, 5' to 3', 5' to 3', it's same na, GAATTC, GAATTC. So it's a palindromic sequences. So your restriction enzymes, this is what they are going to recognize. This is what they are going to recognize. Is that clear? This is what they are going to recognize. And it's not always mandatory, right? I'll give you one random example. Just let's say if it is ATC, ATC, GAC. ATC, ATC, GAC, right? So it is not mandatory. It is not mandatory, students, right? It is not mandatory that always, जैसे here G complementary to GC, complementary to AT, T, then AA and G. It's not mandatory that always it has to form the stretch like this. Sometimes that can also be the scenario that this sequence is present here. It is not complementary to it. It is present here. ATC, ATC, GAC. ATC, ATC, GAC. So if you are reading it from 5' to 3', it's same. 5' to 3', it's same. So this is the meaning of the palindromic sequences that the enzyme can recognize. Is that clear? That the enzyme can recognize. Is that clear? Is that clear? Now we have to talk about the ends also. So look at the NCERT here. Look at the NCERT here. So when you talk about this, now I will discuss the nomenclature, the first enzyme discovered, right? Then I will tell you about the sticky and the blunt ends. So please, focus here, focus here. In the year 1963, two enzymes responsible for restricting the growth of bacteriophage. What is a bacteriophage? It's a virus. It's a DNA virus. It's a double-stranded DNA virus. It's a virus that infects the bacteria. So, two enzymes responsible for restricting the growth of bacteriophage in E. coli were isolated. In E. coli were isolated. One of these added methyl groups to DNA. One of these added methyl groups to DNA, while the other cut DNA. So it is the restriction endonuclease. So in the NCERT, it is not given in proper detail, but I will tell you. See, here we are talking about the restriction modification system. What are we talking about here? We are talking about the restriction modification system. Anybody in the class who's aware of it? Anybody in the class who's aware of it? Anybody in the class who's aware of it? Yes? Yes? Right, so restriction modification system. So restriction modification system is having two enzymes. One is the restriction enzyme and another is the modification enzyme. Another is the modification enzyme. Again, I'll tell you, understand it, no need to write it, understand it. It's simple. Just say I told you that this system is basically important. This is important for what? This system is important. How is it important? This system is important, but in a way that it restricts the growth of bacteriophage in, right? It restricts the growth of bacteriophage in E. coli, right? Done बच्चे, done. So what is going to happen? जैसे let's say this is your bacteria. Bacteriophage is trying to infect it. Now in this E. coli, you have the enzyme. What do we call this enzyme? We call this enzyme as restriction enzyme. What do we call it? We call it as restriction enzyme. Now this restriction enzyme is going to do what? It recognizes specific sequence of DNA. It recognizes specific sequence of DNA. Because it recognizes specific sequence of DNA, my dear students, so it is going to make a cut in that specific sequence, right? That is the restriction enzyme. So when this bacteriophage, when it will insert its genetic material here into the bacteria, so that restriction enzyme will find that particular sequence and will make the cut there. The role of this restriction enzyme clear? The role of this restriction enzyme clear? Now the point here is, if restriction enzymes are present in the E. coli, if they are present in the E. coli, then why it is not destroying that E. coli's DNA? Due to these modification enzyme, they add methyl group 2 to 3 bases of the sequence. So it adds methyl group to the 2 to 3 bases of the specific sequence, to that specific sequence recognized by restriction enzyme. Just for an example, I told you about the gatus GAATTC. Your EcoR1 recognizes that. So what if I add methyl group? So if I will add methyl group, the restriction enzyme will not be able to recognize it, will not be able to cut it. It will not be able to recognize it, it will not be able to cut it. Right. So that is the role of modification enzymes. So this restriction modification system was discovered, bache. This restriction modification system was discovered in year 1963, right, bache? And this is the role here. Okay? This is the role here. Okay. So the first restriction endonuclease was HindII. Let me mark it. HindII. It gives blunt ends. It's important. It's a PYQ, so I'm marking it here. There is HindIII also that forms sticky ends. Do not ever get confused. Okay, so HindIII will give you sticky ends, but HindII will give you, right, bache? HindII will give you what? Yes, HindII will give you what? HindII will give you blunt ends. Yes, Hannah. So HindII is the first restriction endonuclease whose functioning, right, for identified, and its functioning was on a specific DNA nucleotide sequence, and that nucleotide sequence was isolated and characterized 5 years later after its discovery. Is that clear? HindII. So it is GTCGAC, right? Which sequence it recognizes? GTCGAC. So 5' end is having G, 3' end, 5' end, 3' end. So it is GTCGAC, GTCGAC. So this is the 6 base pair sequence that will be recognized by the HindII. Clear, bache? So it was found that HindII always cut molecules at a particular point by recognizing a specific sequence of 6 base pair. So it is GTCGAC. Quickly write it in the chat section. Just for EcoR1, it is GAATTC. Here it is GTCGAC. GTCGAC, right, bache? Right, bache? So in addition to HindII, we know more than 900 restriction enzymes that have been isolated from over 230 strains of bacteria, each of which recognize different recognition sequences. Is that clear? Each of which recognize different recognition sequences. Done, bache? Yes, please. Yes, please. So HindII today we know more than 900 restriction enzymes that have been isolated from over 230 strains. Done, bache? Right. So now when you talk about its nomenclature, please highlight everybody. Please highlight everybody that first letter of the name comes from the genus. And the second two letters, they come from the species of the prokaryotic cell. Just say here, we are writing about EcoR1. E, this first letter, it is coming from the genus name, Escherichia coli. Second and third letter, it is coming from the second and third species name. They are coming from the species name. Now this R, it is telling us about the strain, right? It is telling us about the strain. The fourth letter is basically the first letter of the strain, right? This is the fourth letter and it is basically the first letter of strain and it is written in capital. So now though I cannot write it in italics, but these two, they need to be in italics. So the nomenclature is important. Then bache, Roman number written at the end indicates the order in which enzyme was isolated from the strain of that bacteria. The order, right? It indicates that order. Is that sufficient? Right, bache? That Roman numeric. So, yes, the letter R is derived from the strain name and Roman number, right? That Roman number, it indicates the order in which enzyme were isolated from that strain of bacteria. Now, see here, they are saying a restriction enzyme belongs to a larger class of nucleases. Exonuclease and endonuclease is there. Exonuclease remove nucleotide from the end and done. Is that clear? Chali. Now I'll give you one example. Then I'll come to this paragraph of NCERT. I told you about the GATTC. GAATTC. GAATTC. What these enzymes are going to break actually. Now, because we have completed molecular basis of inheritance, right? So you understand these are the bases, right? You understand these are what? These are the bases. So the sugar phosphate, these are the arrows written here, right? So this arrow is saying that what this enzyme will do. It will basically break or hydrolyze its sugar phosphate backbone. Right? Sugar phosphate backbone. Important it is. And always remember, when you look at this palindromic sequence, if here, right here, if it is cutting in between G and A, here also it is cutting in between G and A. It's not possible that here the arrow is at G and A, here it will come between AA. No, it's not possible. It will be here only. So what will it break? Sugar phosphate backbone. Right. Now I'll show you. I'll give you a demo here. Don't you think that if this is how it is going to cut, right, then these are the fragments that we are going to get. Yes, people, don't you think so? Please tell me, don't you think so? Yes, don't you think so? These are the two fragments that you are going to get, because it's simple, na? It's simple. All of them are connected with the hydrogen bonding without any doubt. Now I am cutting it here, So if I'm cutting it here, basically I'm breaking the sugar phosphate backbone in between G and A. So this part here will get separated, but still G and C is still connected to each other. Then C, T, T, A, A. Right? Now here, because I'm making the cut here, so obviously this AATTC will get separated. So don't you think this is what I'm going to get? What are they? They are the sticky ends. What are they? They are the sticky ends. And why do we say they are the complementary overhangs? It's simple. This is one stretch, this is another stretch. This is one fragment, this is another fragment. If I will bring this fragment here, don't you think that they are complementary to each other? Don't you think that it will be complementary to each other? Don't you think that it will be complementary to each other? Please tell me. Please tell me. Don't you think that it will be complementary to each other? Yes or no? Yes or no? Hannah, obviously madam, 110% madam, right, no doubt. So when I will bring them closer, they will form the hydrogen bonding, and this G and A, again, who's going to ligate this G and A? The sugar phosphate backbone, DNA ligase. So DNA ligase is the molecular suture, molecular glue that helps in the joining of sugar phosphate backbone. That helps in the joining of sugar phosphate backbone. Understood? 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 Complementary overhangs. This is the arrow. So it is not cutting in the center, a little away from the center. Now, I gave you the example of, I also gave you the example of Hind2. Please tell me the sequence recognized by Hind2. It is GTC-GAC. It is GTC-GAC. It is GTC-GAC. Now, how Hind2 is going to cut? It cuts like this. So if it is going to cut like this, GTC, that's how it will get separated. So these are the blunt ends. They don't have anything complementary. These are the blunt ends. So if we need to use the blunt ends in the genetic engineering in the template independent manner, we have to add the bases here so that we can make them complementary overhangs. So why am I telling you this? Why am I telling you this? Because you already understand how to form the recombinant DNA. Right, you already understand how to form the recombinant DNA. So basically, why am I telling you about these complementary overhangs or not? It's simple. Like, you need to cut. You need to identify your desired gene, then you need to cut it. Then you have to select a vector. That is, most commonly used vectors are plasmid and bacteriophage. Then you have to take it. Then you have to take it. So you always use same restriction enzyme. You always use same restriction enzyme to make a cut in your desired DNA and to make a cut in your vector. Why? Because if you will use same restriction enzyme, you will get same type of ends. Same type of sticky ends will be complementary to each other. You will be able to use it. Okay? Okay? Right? So, enzymes cut both strands at the same site. This is your vector DNA, people. This is your vector DNA. Yes, this is your vector DNA, yes or no? Yes or no? Right, this is your vector DNA. This is the foreign DNA. So, eco R1 you are using, so obviously GAATTC. So, you are using same restriction enzyme to make a cut in the vector DNA to make the cut in the foreign DNA. So, obviously you will get sticky ends. You will get sticky ends. So, because we have to join it, ultimately we have to form a recombinant. So, we need to join the vector with the desired gene. So, obviously if I am using the enzyme that is giving me the sticky ends, they are complementary overhangs. I will bring them closer, so they will form the hydrogen bonding, and later on, who's going to join that sugar phosphate backbone? Who's going to join that sugar phosphate backbone? My DNA ligase. So that's how I'll be able to get the, recombinant DNA. That's how I will be able to get the recombinant DNA. Am I clear here? Right? So DNA fragments join at sticky ends. DNA fragments join at sticky ends. Right, bache? DNA fragments join at sticky ends. Is that clear? Is that clear? Yes, is that clear? Is that clear? Sure? Are you sure? Are you sure? right? So this is about the sticky end, and I told you about the blunt ends also. So please, there are enzymes. Jैसे in your book, PBr322 is given. PBr322 is given, right? That plasmid, right? So in that plasmid, that recognition sites are given. So in the case of that restriction, so I think you should know the sequence that is recognized by that enzymes. So just EcoR1 GAATTC, right? You have this cell 1 Streptococcus albus, the sequence. Just like HIND2, it also recognized GTC GAC, but it gives the sticky ends instead of blunt ends. So HIND3, it also gives sticky ends. BMH1, sticky ends. So they all are, no, not all, some are also giving the blunt ends. So this is the thing that I have uploaded here. Rest in NCERT, they have explained the same thing, bache, right? About the palindromic sequence, they have also given the example of Malayalam, right? So when you read from both forward and backward, you will get the same words. Is that clear? Is that clear? So this Gattus example is also given, right? So restriction enzymes are used. So let me tell you, when you talk about the restriction endonucleases, you have 1, restriction endonuclease 2, restriction endonuclease 3. But the one which is useful in genetic engineering is restriction endonuclease 2. Why? Because it's simple. It's not like that restriction enzymes, they directly know that, okay, this is the part where I have to make the cut. No, they will inspect the length of the DNA. What they do, bache? They inspect the length of the DNA. What they do, bache? They inspect the length of the DNA, and when they find the suitable sequence, the particular sequence, then they make a cut. Then they make the cut. Okay, so restriction endonuclease 1 and restriction endonuclease 3, they can inspect the length of the DNA but cannot make the cut. Again, this is also a PYQ. That restriction enzymes, they cut the strand of DNA a little away from the center of palindromic side. What is the meaning of? It is not this is a palindromic sequence, not here at the center, little away from the center. Little away from the center. And what will it do? It will break sugar phosphate backbone. Okay? Right? And it will always be in between same bases, GA, GA. So these are the overhang stretches. They are the sticky ends on each strand. So these are named so because they form hydrogen bonds with their complementary cut counterparts. Right? They can form hydrogen bond with their complementary cut counterparts. The stickiness of ends facilitate the action of enzyme DNA ligase. So what DNA ligase is going to join? Sugar phosphate backbone. How's the speed, guys? How's the speed? How's the speed? Fine na? Not bad, ma'am. Acha. See, that's the best I can do. Otherwise, I have to be like, restriction nucleus, cut and cut the end. So that you will not be able to understand it. Then you will not be able to understand it. So that's how it will make the cut, right? So restriction endonuclease used in genetic engineering to form the recombinant molecules of DNA, which are composed of DNA from different sources or genome. So when you cut the same restriction enzyme, the resultant DNA fragments will have same sticky ends. Doubt? Diagrams are important. Do not miss the diagrams. Okay, diagrams are important. Do not miss the diagrams. We are next 1 till 10. Nahin, this chapter is comparatively till 10:30. We will finish this part, and 1 hour is required for your next chapter. Okay. Chalo, let's go. So foreign DNA vector. So you will use the same restriction enzyme. That's how we are going to make a cut. Ligase will be used here to join foreign DNA to plasmid, and then the transformation. What is the transformation? When you are passing this particular recombinant DNA to a cell and it is picking it up, so obviously it will replicate, right? When it will replicate, and plus it will express the gene, right? When the gene will express its protein, so obviously this particular cell has been transformed. It is having the new genes which will give it the new phenotype. That's how it is. That's how it is, Okay. Okay, so next thing is the separation and isolation of DNA fragments. Again, here it is not something like this. Ki, "Arey yaar, ma'am, okay, I have this piece of DNA. Okay, I am going to make a cut here like this, like this. Nahin, nahin. You are basically going to mix some liquids, right? You are basically going to check some solutions. You'll put one solution in another, one solution in another, one solution in another, and on the basis of that, you will be like, okay, just say you have the sample. Okay, I'm putting the DNAs. It will digest DNA. Okay, I'm getting this. I will put it there. So it's not like you will be able to see, okay, that helically coiled DNA is sitting like this and DNA is waiting for the enzyme. Oh, come on, restriction enzyme here. Please make the cut. No. Ultimately, from your part, you will see that you are mixing certain solutions. Right? There will be certain instruments under which you will observe the things. That's the point. Now the point is you have used the restriction enzymes. Let's say, imagine there is a test tube. There is a test tube in which you have your DNA, right? And there you are putting the restriction enzyme. And then obviously you assume that restriction enzyme at this temperature, at this temperature, in that particular time, it will make the cut. And then you're like, I have to isolate the fragments. So how that fragments will be isolated? With the help of electrophoresis. How will it be isolated? With the help of electrophoresis. With the help of electrophoresis. So when you are using the word electrophoresis, electricity, right बच्चे? Phoresis. देखो, here basically there is the separation. There is the separation of charged particle under the influence of electricity. So there is the separation of charged particle under the influence of electricity, right? There is the separation of charged particle under the influence of electricity on the basis of size. This line is important, this point is important. It will be done, but how? On the basis of size. It will be done, but how? On the basis of size. Is that clear? Is that clear? It will be done on the basis of what? On the basis of size. Is it clear? Is it clear? So what are we talking? We are talking about the electrophoresis. And what am I telling here? I am telling that there will be the separation of charged particle under the influence of electricity. The particle has to be charged, but the separation is not on the basis of charge. It is on the basis of size. That's the important factor. That's the PYQ. So when you talk about this electrophoresis, particularly the gel electrophoresis, you have two things. You have age and page. You have two things. You have age and page. You have two things. You have age and you have page. You have agarose gel electrophoresis and you have polyacrylamide gel electrophoresis. Agarose gel electrophoresis and you have polyacrylamide gel electrophoresis. Is that clear? Is that clear? Is that clear? Pakka. So agarose gel electrophoresis and polyacrylamide gel electrophoresis, right? Polyacrylamide gel electrophoresis. ठीक है जी, right? So basically we are going to read about this. So what is agarose? It's a polymer extracted from a seaweed. It's a polymer that is extracted from seaweed. It is a polymer and that polymer is extracted from seaweed. Understood? Understood? Okay, so we have to use this. So firstly you just have a look, then we will read it from NCERT. This is the whole setup. This is the whole setup. This is a just like right, this is that setup. This is attached to the source of electricity here. Here you are going to see some wells, right? You are going to see some wells, and in that wells you have to load your sample, right? That is your DNA. Actually what's going to happen, as I said, agarose gel electrophoresis we will read. So agarose is a polymer, so you will get it in a powder form. What will you do? You will mix it in the hot water, and then we will allow it to cool. We will allow it to cool. So once it will cool now, once it will cool, it forms some gel-like consistency, right? The filaments, they will be, you know, they'll be arranged in such a way that we will get gel-like consistency. What will we get? What will we get? Matlab, when we will dissolve the agarose in hot water, the solution will be cooled, the double helices will be forming here, right? The double helices which will form here, they will be laterally arranged in such a way that ultimately the point here is you will get a gel-like consistency. You will get a gel-like consistency, not exactly something like this, not exactly something like this, but yes, some gel-like consistency you people are going to get there. Okay, some gel-like consistency you people will get. Let's say after cooling down, it is forming the double helices. They are laterally arranged, the filaments are there, and this is what you are going to get. Now here, when you talk about this gel-like thing, now here the pore size will also be there. What will be there, bache? The pore size also be there, and that pore size, it depends upon the agarose concentration. Understood? This pore size, it depends upon the agarose concentration. Understood? Understood? Right, right, right. So pore size depends upon the agarose concentration. So now you will get this gel here. You are having the wells here. You have to load your DNA, and under the influence of electricity, it will move on the basis, separate on the basis of charge. So DNA is negatively charged. Here now you have positive end and negative end. Plus end is anode. Negative end is cathode. Remember this. Do not make the mistake here. Plus end is anode, negative end is cathode. Do not make the mistake here. So when you load your sample, your DNA sample here, right, this end is the anode. So under the influence of electricity, it will, on the basis of its charge, on the basis of its size, it will separate. So smaller the fragment, nearer to the anode, larger the fragment, it will go slow. So smaller fragment will move very quickly. Smaller fragment will move very quickly. This part clear? This part clear? So smaller fragments under the influence of electricity, they'll be towards the anode here. The larger fragments will be near to the well zone. Right? So if we are loading the DNA sample, later on we have to see it. We have to observe it. Right? We have to observe it. Yes or no? We have to observe it, or we have to see the fragments now. Right? Once these fragments are separated, we will expose it to UV light. We will expose this agarose gel to UV light. Right, bache? So we have to color. We have to color the DNA so that we can see that bands. So that will be colored with ethidium bromide. Again, this is the favorite question of examiner. Ethidium bromide is the stain that will be used to stain the DNA, and bright orange colored bands will be obtained. Bright orange colored bands will be obtained. So we have to repeat this process for both, for plasmid DNA as well as the host DNA with the gene of interest. Now, I'll ask you one question. If you are cutting the plasmid, how many fragments are you going to get? And if you are making a cut in the linear DNA, then how many fragments are you going to get? If you are cutting the plasmid, how many fragments are you going to get? Right? If you are cutting the linear DNA, how many fragments are you going to get? Please answer this question. Everybody here in the class. Plasmid is circular. So if you are cutting a circle, obviously you will get only one fragment. But if it is a linear DNA and then you are making the cut, you will get two fragments. Keep it in your mind. So this gel electrophoresis, it will be repeated with both. Okay? So the separation is on the basis of size. Smaller the size, farthest it will move. Okay? Okay, so DNA fragments are negatively charged. They can be separated by forcing them to move towards the anode. Anode is positively charged, and it will be done under the influence of electric field. Okay, so most commonly used matrix is agarose, which is a natural polymer extracted from seaweeds. So DNA fragments, they separate right, according to their size through sieving effect provided by agarose gel. Sieving effect means a certain pore size is there in the agarose gel, right? It depends upon the agarose concentration. So accordingly, these fragments will get separated. Okay, so smaller the fragment size, the farthest it moves. Okay, so separated fragments can be visualized only after staining the DNA with a compound known as ethidium bromide, and then their exposure to UV radiation. Is that clear? Is that clear? So you cannot see pure DNA fragment in the visible light and without staining. So we have to stain it. If the stain is not proper, we will not be able to see the DNA fragment. If you are not exposing it to the UV light, you will not be able to see it. So while performing these techniques, you have to be very clear. You have to be very specific over here. Okay? Okay. So you can see bright orange colored bands. Bright orange colored bands of DNA in ethidium bromide, stained gel, exposed to UV light. Exposed to UV light. So separated bands of DNA are cut from the agarose gel. They are extracted. Matlab, jaise let's say this is the gel. You can see this is where your fragment is. Now you will cut this gel. You will cut this part of the gel because you have to isolate your fragment. So this step, the separated bands of DNA, when they are cut out from agarose gel, bache, and then we have to extract this fragment from this gel piece. This is elution. Important. Important. Understood? Important. So these DNA fragments will be purified in a way we can use it for constructing our DNA by joining them with cloning vectors. Understood? Now, on what basis you are saying that something is a vector? Jaise most commonly used is plasmid, right? Even the bacteriophage. Even the bacteriophage. So not just the plasmid, not just the bacteriophage, there are others also. Jaise you have cosmids, you have phagemids, etc., etc. And here we also talk about the shuttle vectors. What do we discuss here, bache? We also talk about the shuttle vectors. What do we discuss here? We also talk about the shuttle vectors, right? Shuttle vectors. What do we discuss here? We discuss about what? We discuss about the shuttle vectors here. So shuttle vectors, they can be used in both in prokaryotes and eukaryotes. It can be used in both prokaryotes and eukaryotes. Just for an example, TI plasmid, tumor inducing plasmid it is. Tumor inducing plasmid it is. Okay, what's that? It is tumor inducing plasmid. It's a shuttle vector. You can use it for a prokaryotic cell as well as for a eukaryotic cell. So these are the different types of vectors. Now on what basis we are saying that something can be a vector, right? Something can be a vector. On what basis we are saying? Please tell me. Please tell me what are the conditions that should be there? Quickly tell me. Yes, people, what are the conditions that need to be satisfied? Yes, bachcho, I'm waiting. Self-replicate. That's it. That's it. That's it. Exactly. So when you talk about the conditions here, right? Yes, you're right. Origin of replication is important, but I would like to mention one thing. See, origin of replication is important because if it is having the ORI site means it can self-replicate, and yes, it is the most important property. Now you are going to discuss one thing here in the NCERT, that is copy number. Just let's say, obviously, why do we need the cloning vectors? They are the gene taxis. We need them for the gene cloning. We need them even for transferring that particular gene, right? To a particular host. That's why we need it. So we are going to read a word known as copy number. Total number of copies. For an example, if let's say you want the 10 copies of a gene, then you have to select a vector, right, that can multiply to produce 10 copies in such a way. Just say this plasmid, if in a cell this plasmid number is 5, means it can give me 5 copies of that particular gene, but my demand is 10 copies, so accordingly I have to select a vector. Right? So let's say if in a particular cell, just giving you some random examples, the particular plasmid, right, it is having more than 10 copies. So this, let's say 11 is the copy number of this plasmid, just giving you the example. So this ORI site is going to decide the copy number. So whenever we talk about, right, linking the gene of interest to a particular plasmid, so we join it near the ORI site so that it can replicate. So this ORI site, right, it controls the copy number as well, the total number of copies. So this is something important here, and the most commonly used vectors are your bacteriophages, and it is your plasmid. And in the Human Genome Project, which vectors are used, guys? In the Human Genome Project, which vectors are used, guys? Which vectors are used? Please tell me, which vectors are used? Name it. Yes, please. BAC, YAC, bacterial artificial chromosome, yeast artificial chromosome. Why? Because they can accommodate large genes as well. Right, large genes as well. So now they are saying that plasmid and bacteriophages, yes, they do have the ability to replicate within bacterial cells, right, and they are not dependent on the chromosomal DNA. They can self-replicate. They don't rely on that chromosomal DNA only, right? So bacteriophages are having high copy number, means number of copies that they can form in a cell is more, right? So some plasmid may have only 1 or 2 copies per cell. Some can have 15 to 100 copies. So their number can even go higher. So if we are able to link an alien piece of DNA with a bacteriophage or plasmid DNA, we can multiply its number equal to the copy number of the plasmid or bacteriophage. Right? Just say, let's say if the copy number of a particular bacteriophage is 20, so I will get 20 copies of my gene of interest. Okay. So vectors used at present are engineered in such a way that they help easy linking of foreign DNA and selection of recombinants from non-recombinants. So that is what I'm going to explain. So in the features, the most important thing is ORI, origin of replication, and you know that it controls the copy number as well. Right? So if we want to recover many copies of the target, it should be cloned in a vector whose origin support high copy number. Then comes the selectable marker. Now, before explaining you the selectable marker, just say I am using the word transformant and I am using the word transformant and non-transformant. Transformant is the word and non-transformant is the word. What is the meaning of it? Transformant. It is transformed, right? Yeah, it is having that foreign DNA. I'll explain. Let's say these are the cells here. As I said, it's not like that you will hold one cell and you are holding one recombinant plasmid and then you are introducing it there. Of course not. Let's say this is a culture where so many cells are there, and let's say you are putting your rDNA here. You are putting your recombinant plasmid there. You're putting your recombinant plasmid there. What are you doing? You're putting your recombinant plasmid there. Let's say if out of these cells, if they have accepted the recombinant plasmid, right? If they have accepted, and now this recombinant plasmid is having a gene. What is that gene? That gene is antibiotic resistant gene. That gene is antibiotic resistant gene, and this gene is antibiotic resistant gene against A. Means if I use the A antibiotic, right, my recombinants will not die, right? So I am basically doing what? There is a culture, I'm putting the recombinant plasmids, and let's say out of all this, these 1, 2, 3, 4, 5, these 6 cells, they have accepted my recombinant DNA. They have done what? They have accepted my recombinant DNA. Yes, bache Satvika. They have accepted my recombinant DNA. Okay? Okay? They have done what? They have accepted my recombinant DNA. That's the point that I'm telling you. Okay, that's the point that I'm telling you. So you tell me that these 6, will they get the resistance against antibiotic A or not? Will they get the resistance against antibiotic A or not? Will they get the resistance against Of course they will. So let's say if in the same culture, if I put antibiotic A, so obviously this cell, they all will die, and the one that will survive will be those who have accepted my rDNA, right? Because naturally this bacteria was not having the antibiotic resistance against this particular A. So I will say now, these are my transformants. They have accepted this new DNA. Now they are transformed. Can I say so? Yes or no? Can I say so? Just say even in the Griffith's experiment, when you talk about the transformation principle, what exactly is the transformation? Even in class 11th we discussed. What exactly is the transformation? When a cell, it accepts a piece of DNA from the surrounding. What is transformation? When a piece of DNA is introduced here into the host bacterium. When a piece of DNA is introduced into the host bacterium, that is the transformation. And why it is the transformation? Because if piece of DNA is introduced into the host bacterium, if it will be the part of its genome, then definitely it will also make the protein. It will give that special phenotype to that bacteria. Are you clear here? Yes. So this is what transformation is, and that is how you will get your transformant. Right? That is how you will get your transformant. And non-transformant, those who die, those who are not able to accept this recombinant DNA. Now, when I'm saying selectable marker, see, I told you this thing very clearly. Very clearly. Right, bache? That it's not like that you are holding a plasmid in your hand and then you are like, okay, plasmid, now you have to go within a cell. Of course not. You are mixing one solution with another, you will run certain experiments, you will take the help of some instruments, and then you will check the things. Okay. Now, if I have a vector, I want to check whether my cell has, now my screen is like, ki bas, I am done. Stop the class. Hai na? My screen is like, I am done. Stop the class. I cannot work further. I am just done. Nahin? So when you are talking about a vector, because this vector we have to introduce into a host cell. So students, if I have to introduce into a host cell, I should know na whether host cell has picked up the recombinant plasmid or not. It's not like that ki host cell is very obedient that I'm going to accept the recombinant always. No, that's not like this. Right? That's not like this. Okay. It's like, so if I will use a vector, the vector which is having a kind of special property, and I can see that special property, if my cell has been transformed, that will show that special property. Just for an example, I am giving you an injection. I am saying that if you will get an injection, if you get that particular infection, you will not be able to. If you will get that particular injection, you will not suffer with typhoid. I'm giving you one example. I'm giving you one example, and what am I saying here? What am I saying here? Yes. What am I saying here? I'm saying a very simple thing there. I am saying a very simple thing here. That is, ki, let's say I'm giving you an injection, and I'm saying after taking that injection, you will not get the infection from typhoid. You will not, Salmonella typhi will not be able to harm you. Then what will I do? How will I check? How will I check that you got that injection or not? I will introduce that particular parasite in your body. If you will not develop infection, if you will not get typhoid, I'll be like, theek hai, the injection is working. Same is the case here. I have a vector. That vector is also having a special property, right? Let's say that special property can be anything. It can be antibiotic resistance. It can be a particular type of phenotype. Let's say after introducing that particular vector in a cell, that cell has started forming spores, the cell has started developing new layers, whatever it can be. It can be anything. It can be anything. It can be anything, right? So if I will select a vector, the vector that is having a kind of special property, that vector, if it is having that special property, so obviously whenever any cell of mine will pick up that particular vector, will show that property, right? And I will be able to recognize that whether that particular cell is a transformant or recombinant or not. That's why the selectable marker, the presence of selectable marker is important. Is that clear? Is that clear? Is that clear? Is that clear? Please tell me, guys, is it clear or not? Dekho, in next 1 hour, we will be able to finish the chapter. So please be a support and tell me. Be a support and tell me. Clear hai? Very good. Bahut badhiya. So that is the importance of selectable marker. Jaise, I'll give you the example. The trick is CAT K. What is the trick? CAT K. Like your E. coli. E. coli, it doesn't show resistance against CAT K, means chloramphenicol, ampicillin, tetracycline, kanamycin. ठीक है, chloramphenicol, ampicillin, tetracycline and kanamycin. So in the case of E. coli, it doesn't show resistance against this, right? What do we have? We have, yes बच्चे, ampicillin है. Firstly we have chloramphenicol, right बच्चे? A for ampicillin, tetracycline or kanamycin, right? So normally E. coli, it doesn't have the resistance against these antibiotics. So in the case of E. coli, if I have to use a plasmid, I'll go with the one now. Which is having these resistances, which is having these resistances. I'll go for that. Why? Because that will tell me whether my E. coli is transformant or not. So they'll be the best suitable selectable markers for my E. coli. Understood? Right? So this is the thing which is given here in this particular part, the selectable marker, a very important thing. And this PBR322, right? It is the cloning vector showing restriction sites for HIND3, EcoR1. Ye dekho. 1, 2, 3, 4, 5, 6, 7, 8. Don't you think 8 restriction sites are there? 8 restriction sites are there. Yes. Multiple restriction sites are present here. Now, when you see this thing, it is the AMPR, resistance against ampicillin antibiotic. TETR, resistance against tetracycline antibiotic. So 8 restriction sites are there, 1 origin of replication. So what is this ROP? ROP is, right, they are for the proteins involved in replication of plasmid. So this region will give me the protein, right, proteins involved in replication of plasmid, right? This is the PBR322. You should know about it. You should know about the name of these restriction sites. Now I'll ask you one question. I told you about the antibiotic resistant, that if we go for the antibiotic resistant genes, right, bache? But in the case of antibiotic resistant genes, when we use the antibiotic resistant genes as the selectable markers, when we do what? When we use the antibiotic resistant genes as selectable marker, then it is a very, kind of very difficult task. Again and again you have to culture it. Again you have to culture it, right? You have to put the culture where antibiotic, that you will introduce particular antibiotic, then you will wait for the cells, whether they are live or dead, then you will take out, then again you will grow. Again and again you have to culture the bacteria, right? That's the point. Now let's say, I'll give you the example. Now let's say the restriction enzyme that I have used is PVU1. Let's say, so bache, if I'm using this restriction endonuclease, it is going to make the cut here. It is going to make the cut here. Yes or no? And if it will make the cut here, I will insert my gene of interest. Matlab, this is the site for the PVU1. Let's say I have used this restriction endonuclease. So if I have used this restriction endonuclease, with the help of this restriction endonuclease, I will insert my gene of interest here, yes or no? I will insert my gene of interest here, yes or no? I will insert my gene of interest here, yes or no? Yes or no? Obviously. But now this complete sequence is, this complete gene is providing the resistance against ampicillin antibiotic. Let's say if I interrupt this part, and if I put a gene of interest there, don't you think this vector will lose the resistance against ampicillin? You tell me. Don't you think this vector will lose the resistance against ampicillin? Don't you think that this vector will lose the resistance against ampicillin? Yes, bache. Yes, bache. Obviously. So what can I call it? Can I call it insertional inactivation? Can I call it insertional inactivation? Can I call it insertional inactivation? Yes, please. Yes, please. What is the meaning of insertional inactivation? You inserted something that results in the inactivation of something. Insertional inactivation. That is what you have to understand. No doubt in NCERT it is regarding that LAC-Z gene. It is regarding that white and blue colonies. But that is not the only thing, right? You need to understand that insertional inactivation means when you are inserting a gene and that results in the inactivation of another. So if I have used the PVU1 as the restriction enzyme, so obviously this vector will lose the resistance against ampicillin, right? But still the resistance against tetracycline is there. So I can use both the things to check whether the cell has been transformed or not. Okay, so third thing given here is the cloning site. Then we will discuss about this beta galactosidase blue and white wala example, and then chalo, let's move quickly. We have to finish it. We have to finish it ASAP. Okay? So up to this part all clear? Up to this part all clear? Up to this part all clear? So cloning site. So in order to link the alien DNA, the vector needs to have very few, preferably. So what is preferred? It is preferred to have single recognition site for the commonly used restriction enzyme. Just say for an example, if you have EcoR1, so EcoR1, it is having the restriction site here. If there will be multiple sites for EcoR1 only, so then to bhai, it will be difficult for us now. Multiple sites are for EcoR1 only in that particular plasmid, right? So that is why it is said in order to link the alien DNA, the vector needs to have very few or preferably single recognition site for the commonly used restriction enzymes, right? So presence of more than one recognition site within the vector will generate many fragments, which will complicate the gene cloning, right? So if there will be many sites for EcoR1, many fragments will be there. How will you go for the gene cloning then, right? So ligation means joining of alien DNA is carried out with the help of at the restriction site in one of the two antibiotic resistant gene. This is what they are explaining. So you can ligate a foreign DNA at BAMH1 site of tetracycline resistance gene. The recombinant plasmid will lose tetracycline resistance due to insertion of foreign DNA, but still can be selected out from non-recombinants, because how can we check the recombinants? If I have used BAMH1, then obviously my recombinants will not show the resistance against tetracycline, but still they will show the resistance against ampicillin. So that is also the property that I can use. Are you getting my point? Are you getting my point? Right, so we can plate, we can culture the transformant, we can just check it. This is what they are explaining. Okay, right? So selection of recombinants due to inactivation of antibiotic is a cumbersome procedure, tough procedure. Again and again you have to set the culture, then you have to wait. It's like that. But we can go for something that can, differentiate recombinants from non-recombinants on the basis of ability to produce the color, right? So color, if color is producing, means there should be certain enzymes, right, that use some chromogenic, means chroma mane color producing substrate. So if that enzyme is there, if there is any enzyme, if it is having a chromogenic substrate, right, so when this enzyme will act on it, you will see the color, a particular color. Like here in the NCERT, they have given you the example of beta-galactosidase. Just now we have studied beta-galactosidase. Yes, do you have any idea? Do you have any idea about the beta-galactosidase? Do you have any idea of the beta-galactosidase? A recombinant DNA is inserted within the coding sequence of an enzyme, and what is it? It is the beta-galactosidase. What is it? It is the beta-galactosidase. It is the beta-galactosidase. Yes, we studied that. Very good sign, bache. Excellent. LAC-Z gene. It gives us the beta-galactosidase now. So this results into the inactivational gene for synthesis of this enzyme. Matlab, let's say you have the DNA fragment here. In this DNA fragment, this is your LAC-Z or Z gene. Now, here within this gene, there is a restriction endonuclease, cloning site, a restriction site. If I will use this particular enzyme, then obviously it will make a cut here, and then I will introduce my foreign gene here, my alien gene here. So definitely I have disturbed that sequence, right? Now the beta-galactosidase will not produce. Now it will not give me the colored colony. Okay, so that's what they are saying. Ki in this, a recombinant DNA is inserted within the coding sequence of enzyme. So the coding sequence of enzyme means the gene for that particular enzyme, that particular fragment of DNA that is giving me this enzyme. So what will happen here? There will be the inactivation of the gene for the synthesis of this enzyme. So this is the insertional inactivation. Okay, bache, this is the insertional inactivation. Is that clear? So presence of chromogenic substrate will give blue colored colonies if the plasmid is in bacteria, doesn't have the insert. If the plasmid is in bacteria, doesn't have the insert. Done? Done? So presence of insert results in insertional inactivation of the beta-galactosidase gene, and the colonies do not produce any color. So these are the recombinant colonies. That's how it is used. Take. So now, so I will explain this part here and we will not discuss it then in biotechnology applications. Then vectors for cloning genes in plants and animals. Okay, so here for delivering the genes in plants and animals, right, we can deliver it with the help of bacteria and viruses, right? Right, bacteria and viruses. So here they have given the example of one bacteria, that is Agrobacterium tumefaciens. That is Agrobacterium tumefaciens. Now what is the name of, see, the bacteria is Agrobacterium tumefaciens. But it is also known as natural engineer. It is also known as natural engineer, this particular bacteria. When you talk about this particular bacteria, it is having a plasmid. It is having a plasmid, and this plasmid is having a stretch of DNA. That stretch of DNA is known as tDNA. That stretch of DNA is known as tDNA. And this plasmid is known as Ti plasmid. What is the meaning of Ti plasmid? Remember I told you it's a shuttle vector also. It's a tumor inducing plasmid. It's a tumor inducing plasmid. What's that? It is a tumor inducing plasmid. Is that clear? Is that clear? What's that? It is a tumor inducing plasmid. Now what's the point? Now what is the point? See, this bacteria, it can infect the dicots, right? Just say it causes the crown gall tumor to the dicot plants. Okay, bache? How? Because this bacteria, with the help of this plasmid, it transfers its tDNA into the normal cell of a plant. It can infect because it's a soil-borne bacteria. It's a soil-borne bacteria. We call it as natural engineer. It doesn't require the 4 years degree, right? BTech degree. It's a natural, right? It's a natural engineer only. It's a natural engineer only. Are you getting my point? It's a natural engineer only. Now, what's the point here? What's the point here? Ti plasmid is there. It is having the tDNA, and this tDNA is infecting the dicot plant. And how? It is transferring its tDNA into a normal cell of that plant. So when this tDNA has been inserted in this normal cell, the cell is becoming cancerous. The cell is dividing actively. The cell is making that tumor, right? So scientists were like, they observed this natural ability. They observed this natural ability. They are like, yeah, it's a good bacteria. It's a good bacteria. It can insert its tDNA into the normal cell. Why? Let's use it in a better way. Let's use it in a better way. So they checked that in the tDNA, in this particular sequence, in the tDNA here, they checked that there are some restriction sites. There are some restriction sites. They used that restriction sites. What they did? They used that restriction sites. So now when they have used that restriction sites, so if that restriction site will make a cut here, so obviously this tDNA, the sequence is interrupted, and then we can insert our gene of interest here. Right? And now, because this is the natural ability of this bacteria, it will again infect the plant. But now it will not pass. Right? But now it will not pass. It will not pass that harmful tDNA. Now it will not make it cancerous. Rather than it will pass our gene of interest. That's how we are exploiting these bacteria for transferring our gene of interest. Right? Then, virus. You can take the virus, any virus, adenovirus. You know that viruses are also having, right? In the case of viruses, they enter the host cell. They use the host machinery. They enter the host cell. They use the host machinery. Yes or no? They enter the host cell. They use the host machinery, and then they divide. They form the multiple copies. They insert their gene of interest. They insert their genetic material there so that they can use the host machinery and they can replicate. Okay? So now let's say at this particular virus, right? If I'll replace this harmful gene, if I'll silence them, if instead of that, if I insert useful genes. So this is the ability of this virus to enter into the host cell and then use the machinery of host cell and then to replicate. So basically that's how it passes the, right, that's how it passes that particular, it will pass its gene of interest. So that's how it can be. So here you have the example of this agrobacterium. Then the retroviruses in the animals have the ability to transfer normal cell into the cancerous one. So what will I do? What will I do? Right? What will I do? I will remove the harmful genes, I'll put the useful one, and that is how I can use their ability. Right? So a better understanding of the art of delivering genes by pathogen in their eukaryotic hosts has generated knowledge to transfer these tools of pathogens into useful vectors. Okay? Okay? So now we have to pass that recombinant DNA into the host. So do you think that all the hosts, they are competent? What do you think? Do you think that all the hosts, they are competent? Do you think that all the hosts, they are competent? Hanji, batao yaar. Tell me. Thank you so much. Tell me. Do you think that all the hosts, they are competent? Yes, please. Yes, please. Of course not. No, all the students, right, those who are saying that we are the NEET aspirants, we are the NEET aspirants, right, we are the NEET aspirants, we are the NEET aspirants, are they all aspirants? Are they all competent? No, they are not. Some are like, we have nothing to do, let's be the aspirant. It's not like that. It's not like, it's literally not like that. Not everybody is putting, right, not everybody is putting not everybody is putting all the efforts to crack this examination. Right? Right, bache? So when you are talking about the competent host, the host that is having the ability to pick up that DNA, not all the cells are having the ability, oh please, DNA is there in the surrounding, please come up. No, we have to make that host competent. So they are saying that DNA is hydrophilic. What is the meaning of hydrophilic? Water loving. And you can also say lipophobic. So it cannot cross the cell membrane directly, right? So we have to force that particular cell to accept this bacteria. We have to treat that cell so that it can accept our DNA, right? But we have to make it competent, right? So for that we have to treat that particular cell, and that too with the specific concentration of divalent cation, right? Divalent cation, specific concentration, like you can go for calcium ion, even magnesium ion can be there, right? So when we treat our cell with a divalent cation, so what will happen, right? It will create some pores there in the outer covering. Okay, so that DNA can enter. Right? So it will create the pores in the outer covering, that is in the cell wall. So recombinant, now we want this recombinant DNA. We have to force it into these cells. So we will incubate the cells with our DNA on ice. Right? So first is divalent cation. Then we have to incubate the recombinant DNA with that particular host cell, right, on ice. Then we have to place it at 40 degrees Celsius. This is heat shock. Then again ice. So divalent cation, ice, 42 degrees Celsius, again ice. Divalent cation, ice, 42 degrees Celsius, again ice. Is that clear? Is that clear? So bacteria will be like, yaar, please do not torture. I am ready to accept what? I'm ready to accept the DNA. Host is like, I'm ready to accept the DNA. Now, microinjection is also a way. These are some vectorless methods where you are not using the vector. Microinjections. Just here, what are we doing? Right? We are basically, our DNA is directly injected into the nucleus of an animal cell. Right? So, just here, do you remember? ICSI, intracytoplasmic sperm injection. Intracytoplasmic sperm injection, right, where directly the nucleus is placed in the ovum, with the ovum nucleus, then the success rate is maximum, right? Success rate is maximum, you can relate it. here. Then next method is, this is mainly suitable for plants, but it doesn't mean we cannot use it on for the animals. We can. So what we have to do, we have to the bombardment has to be done. So let's say we'll go for the high velocity microparticles. It can be gold, it can be tungsten. Why? Because they are not reactive, right? And see, so mainly they are not reactive. Your gold is not reactive. So what we have to do? So we have to coat our recombinant DNA on it, right? So at a very high velocity, right? Let's say these are the plant cells. The bombardment will start, and obviously the DNA will enter in that particular cell. So this is the ballistic method or gene gun method, right, bache? And then I told you already about the disarmed pathogen vectors, where we are using the viruses, but we are disarming their harmful genes. Okay. So now let's talk about the tools here. So though you know many things, but you need to focus on few points here. Okay. We need to focus on few points here. So let's finish it quickly. What say? So process of our DNA technology. Now you are talking about the steps here. First is isolation of DNA, right? Isolation of DNA. So obviously, nucleic acid is the genetic material of our organism without exception. It can be DNA, in some viruses it can be RNA, but ultimately it is the nucleic acid that is the genetic material, right, bache? So firstly, right, if we want to cut our DNA with the restriction enzymes, that DNA needs to be in pure form. And it's not like that if you're cutting a cell, if you are breaking a cell, then you will get the pure form of DNA. No, the proteins are also there, fats, carbs, so many things are there. So if we want to cut our DNA with the restriction enzyme, we need to get what? We need to get the pure form of DNA. So before that, we need to break the outer wall. So here, if you see this part, here in this particular paragraph, this is what they have discussed. Lysozyme for bacteria, cellulase and pectinase for plant cell, chitinase for fungus will be used, and I told you already about it. Okay? So after all this, after treating it with this enzyme, what we have to do? We know that the DNA is present, it is intertwined with the proteins. RNA can be removed. Let's say you have a test tube, you have a bacterial cell, you can say that you have a plant cell, you have added the cellulase is there. Now you know that cell wall will be digested. After that, what you have to do? You want the DNA to be in pure form. You'll add RNAs, that will digest RNA. You'll add protease, that will digest protease. Are you getting my point? Right? So other molecules can be removed by appropriate treatment. And how can you get the purified DNA? You cannot add DNAs. You're not allowed to add DNAs. Why? Because if you will add DNAs, it will digest your DNA. And you need DNA to be in pure form. You have no need to digest it. Right? So what we have to add here? We have to add the chilled ethanol there. What we have to add there? We have to add the chilled ethanol there. So because we have to add the chilled ethanol there, yes or no? So after adding the chilled ethanol, right, the fine threads will be obtained in the suspension. So look at this particular diagram. So you will add chilled ethanol, you will get this thing. But here you use the word spooling. So spooling word is not given in the text, but here it is given. Here DNA that separates out can be removed by spooling. You will see that DNA precipitates, you can use the glass rod or you can use some wooden stick, then you will collect these precipitates. This is the spooling. What's that? It is the spooling. Right? So now you have the pure form of DNA. Obviously, what will you do? You will add restriction enzymes, right? How? By incubating the pure DNA with the restriction enzymes. So see, incubation has to be done. So again, you cannot observe it. This is my DNA, this is my restriction enzyme. How is it cutting? No, incubate it. At proper condition, optimal mane, best suitable condition. Then you have to go for the agarose gel electrophoresis because you have to separate the fragments. And same we have to do for the vector DNA also. Told you. Right? Right. Yes, bache. So you will get your gene of interest. Then cut vector with space are mixed and ligase is added. So your vector plus gene of interest is mixed, DNA ligase will be added, and we will hope that our recombinant DNA form. Now let's say if your gene of interest is not there in ample amount, what will you do? You will go for PCR. What is PCR? Polymerase chain reaction. What is PCR? Polymerase chain reaction. A very important technique for the amplification of gene. Right? It is a very, very important technique for the amplification of gene. So it was invented by Carrie Mullis in 1983 and she even got the Nobel Prize for that. Right? Right, so PCR. So here in PCR, polymerase chain reaction, the amplification of that gene of interest will be done. And you remember, just when people were affected with COVID, PCR is also used for diagnosing disease also, and that too at early stage. Right, that too at early stage. Okay, bache? Okay, bache? That too at when? That too at early stage. Okay, chale, I'll now give you the example. Just RT-PCR, when that COVID wave was there, RT-PCR was done. Are you getting my point? So when you are talking about the PCR, it is for the amplification. So here you have three steps: dye, D, A, and E. Dye. These are the three steps here. What is it? Dye, D, A, and E. What is this D? It means denaturation. It means annealing. It means extension. These are the three steps here. Simple. So basically the gene of interest is synthesized in vitro using two sets of primer. Chalo. Now let's discuss it. Right? So PCR, DAE. So what do you need for this? You need a primer, and that is chemically synthesized. That is chemically synthesized, and it is of oligonucleotide. It is not a RNA primer, it is of oligonucleotides. Magnesium, enzymes, right? Deoxyribonucleotide bases. This is what you need. And yes, of course, the gene of interest. Of course, the gene of interest. Right? So in a very simple way, I'll explain it. Just say, let's say this is your gene of interest. Okay, you want to make multiple copies here. So what will you do? Firstly, you will go for first step. And why that first step is important? Because we have to convert this double-stranded DNA fragment into single strand. So when you talk about the denaturation, we are basically trying to break the hydrogen bond here. What are we trying to make? We are basically trying to make the hydrogen bond here. And here the temperature will be above 90 degrees Celsius. And if I have to be specific, 94 degrees Celsius. So at this high temperature, don't you think the bonds will break? At this high temperature, don't you think the bonds will break? Don't you think that the bonds will break? Yes or no? Yes or no? Yes or no? So because these two bonds will break, now you have two template strands. You have two template strands. Next step is of annealing. Annealing means joining. So here the temperature will be low. Next step is annealing, means joining. Here the temperature will be low. Somewhere in between 50 degrees Celsius to 54 degrees Celsius. Let's be very specific, 54. So annealing means joining. Now here the primer will join, and bache, these primers are chemically synthesized. It will not synthesize there in that setup, right? Because we know our gene of interest, we know the sequences there, so accordingly we know the primer as well. We will chemically synthesize it. Just say we have to order it. Let's say you have to perform this technique. You know this is a gene that I want to amplify. You know the sequences there. Then you have to order it. Okay, this is the primer that I need. So this step here, the temperature is low. Right, here in this particular step, the temperature is low because here you have to join this primer with the strand, no? Here you have to join this primer with the strand, na? Here you have to make the hydrogen bonding, na? So because you have to make the hydrogen bonding, the temperature has to be low. The temperature has to be low. So because it is PCR, it is in vitro. So here there will be the continuous DNA strand synthesis. There will be the continuous DNA strand synthesis. There will be the continuous DNA strand synthesis. Right, bache? Right, bache? So, no lagging strand here. Right, it's not like that the DNA polymerase has to move in two opposite direction towards the direction of replication fog. So, no lagging strand will be there. So, it will be the continuous synthesis. Do you know that? Continuous synthesis. So that will be in the next step. DAE. Denaturation, annealing and extension, and even the What is the next step? Elongation या extension, right? What is the next step? Elongation या extension? Extension. So here again, what's going to happen? The bases will be added further, right? The bases will be added further. So it is extension. The temperature is near about 72 degrees Celsius. So obviously, बच्चे, both the strands complementary to it, the bases will be added. So no leading, no lagging strand, and this is not the RNA primer that is used. It is actually the DNA primer. DNA primer only. Right, oligonucleotide primer only. And one more thing here, the enzyme that is used is Taq polymerase. Here in PCR, which enzyme is used? It is Taq polymerase. Taq polymerase means T for thermus, AQ aquaticus. Actually, bache, the temperature here is very high, so we need an enzyme because thermal stability is a very important property of an enzyme. So we need an enzyme that can survive under high temperature. So Thermophilus aquaticus, yeah, Thermus aquaticus. It is an archaebacteria. You know that archaebacteria can survive under harsh conditions. So Taq polymerase will be obtained from this, right, bache? Taq polymerase will be obtained from this, right? And that's how it will proceed. ठीक है जी? Okay. Right? So it's an automatic process, right? Because this enzyme is heat resistant. So this is again important PYQ. This is again important PYQ. This is the PCR, right? So obviously we can get many copies as much as we want. So ये देखो primers, right? Taq polymerase extension is there, and after 30 cycles, so you can get approximately that number of copies. So this is the PCR. This is the actually my mic is dead. Now it's fine, right? Now it's fine. Hello, hello. Now I hope it's fine. हाँ बस ठीक है. Okay. Is it clear? Is it clear? So there are several methods of introducing this ligate DNA into the recipient cell. You know that we have to make it competent, right? Right, and we have to select the. So if the recombinant DNA bearing gene for resistance to antibiotic means this is, they are talking about the selectable marker. So we can definitely check our transformed cell, isn't it? Isn't it? So about the selectable marker, I already told you. Now why are we doing it? Because we need to obtain a product. Like let's say if you are doing in a lab, you're doing it at a small scale. Maybe it is for some experimental purpose, it is for some research purpose, right? But in the like there are companies, right? Those who sponsor these type of research because they want that product. They want to commercialize it. It can be anything. It can be any drug. It can be any particular protein that is good for human welfare. And what they want to do, they want to make some money from that. Right? So they are also doing it at large scale. So obtaining the foreign gene product. Foreign gene product means the gene that we have introduced there. The gene that we have introduced there. So the point here is that when you insert a piece of alien DNA into the cloning vector and when you transfer into a bacteria, plant or animal cell, what we want? We want that DNA to multiply. We want that DNA to express itself, isn't it? Right, bache? So after having cloned the gene of interest and having optimized the conditions to induce its expression of target protein, we have to consider producing it on large scale, right? So large scale pe. So when you want to produce it at a large scale, so you want this recombinant protein at a large scale, so you have to grow that particular microorganism. So we will talk about the bioreactors here. bioreactors here, that large vessels having curved base. So in that bioreactors, we are going to culture it. In that bioreactors, we are going to culture it. Mr. Tesfai, in the description box, there is a video, there is a link, right? You can click on that link and you can be the part of Unacademy platform. Okay, now please focus here. So, if any protein encoding the gene is expressed in the heterologous host, it is called a recombinant protein. Matlab, you have introduced that gene in some other species. There it is going to express. There it is going to express. What is it doing? There it is going to express. Are you getting my point? Hello? Okay? Right? So it is the recombinant protein and we want it, and we, let's say, if in a lab, small scale, if you want to culture it, commercialize it, we have to grow it at a large scale. So for that, what we need the bioreactor. So what are they saying? The cells can also be multiplied in a continuous culture. Just say, firstly, you understand bioreactors. Just say here, these two examples are given, right? Here, these two examples are given. Let's say in simple language, if I have to explain something, you have this large vessel, a very large vessel. You are calling it as a bioreactor. Here in this vessel, you will put the culture medium, right? And you will put that particular organism, that recombinant one, yet transformant one. Here you will grow it. You will grow it. There are different types, right? There are different ways of culture. Sometimes it is batch culture. What is the meaning of batch culture? You are going to put the culture medium, you are going to put the microbe there, they will grow, right? And you are waiting the time this culture medium is exhausted, and then you will get that product. Batch culture, batch wise. Just say I have started my classes with 30 students, right? I have started my classes with 30 students. Okay, what have I done? I have started my classes with 30 students. I'll complete their syllabus first, and then I will take another 30, right? Another 30, like this, batch wise. Then fed batch is also one of the thing. It is the improvised way of this batch culture. Okay, it is the improvised way of this batch culture. So this is one way. This is one way that you are doing what? You are like, theek hai yaar, right? What are you doing here, bache? What are you doing here? Batch wise, you are putting the things. Theek hai ji? Okay. 2 or 3 things. Okay. Then there is the continuous culture. What is happening in the continuous culture? When it is a continuous culture, continuously you are supplying the culture, the nutrient medium, and whatever is the waste, you are removing it, and whatever, there is also a tray from where you are obtaining your product as well. You are checking its quality also. Right? Continuously things are going. Matlab, it's like your microbes, their food supply will never be exhausted. Always they will get the food supply. So imagine if always they are getting the food supply, they will be at their exponential phase now. They say initially there is the lag phase, right? Remember this growth curve, lag phase, then comes the log phase or exponential phase. So because they are getting the continuously they are getting the food, right? Continuously they are getting their food. Yes, bache, continuously they are getting their food. Yes or no? Yes or no? So obviously they will just divide, multiply, they will feed on that food, they will be in their exponential phase, and obviously continuously things are going on, you will get your recombinant protein. So this is about the bioreactors here. Right? This is about the bioreactors here. So you can see that small volume cultures cannot yield appreciable quantities of product. Okay, to produce in large quantities, the development of bioreactor where large volumes, 100 to 1000 liters, can be processed was required. So thus, bioreactors can be thought of as vessels in which raw materials are biologically converted into specific products. So individual enzymes using microbial, plant, animal and all. So bioreactors, they provide the optimal condition, the suitable conditions for what? For achieving the desired product. How? Because they will provide optimum growth condition, the temperature, the pH, the substrate, salt, vitamin, oxygen, etc. Okay? So look at this part. Two type of bioreactor. Simple stirred tank, stirrer is there for the uniform availability, for that uniform distribution rather. ठीक है बच्चे. So acid base for pH control, steam for sterilization, which is very important. Even in the bioprocess engineering we emphasized on it. Foam breaker is there, flat bladed impeller, cultural growth. ठीक है, sterile air will be passed. Then one is passed stir tank bioreactor. So these bubbles are also present here. They will increase the surface area for oxygen transfer. This is a gas entrainment. So these bubbles, they increase the oxygen transfer area. Right, so oxygen is also available there. So this is important. Directly diagram can come. Be clear about it. So stir tank is usually cylindrical, having the curved base, so that mixing of the reactor content can be there. So stirrer facilitate even mixing and oxygen availability throughout the bioreactors. So alternatively, air can be bubbled through the reactor, bache. So if you look at the figure closely, you will see that bioreactor has an agitator system, oxygen delivery system, form control system, temperature, pH, and sampling pots are also there. Right, so time to time we will withdraw the culture, we'll check the quality, and that's how the things are. Right, so now you got a product, now you have to sell it in the market. Firstly, you will check downstream processing will be done, right? Quality check will be done. Efficacy will be checked in the case of drugs. Okay. So after completion of the biosynthetic stage, the product has to be subjected through a series of processes before it is ready for marketing as a finished product. Right. So what is there in downstream processing? Separation and purification. What is there in downstream processing? Separation and purification. Separation and purification. So it is collectively referred to as downstream processing. You will separate, you will get the pure form, you will purify that. Right, then you have to formulate it with preservatives. Right, so in the case of drugs, proper clinical trials has to be done, and then strict quality control will be done. That's it, that's all, and the chapter will be over. Even I cannot imagine that I have finished this chapter within, I think we have started at 8, है ना यार, ठीक है यार, till 10:15. I was expecting 10:30. So now tell me, good ma'am, very good, we are proud of you. Okay. Done? Good. You should be. है ना? So 2 chapters are completed, and yes, we have completed the most important portion. I'm happy. I'm happy. I'm happy. I know you are not happy, but I'm happy. Okay? So see, I'm telling you this thing honestly. I know you're grasping, right? See, I believe that you are not doing these chapters for the first time. It's just that you need to revise it. So the thorough revision is important at this point of time. For an example, if in the month of January, let's say I'll complete, because what do I need? I need 6 marathons. I need 6 marathons. 6 marathons means this is the first week, 1 marathon, next week, yes, hopefully next week, next marathon, hopefully next week, next marathon, right? So basically, let's say if we complete class 11th here in the month of January, class 12th there in the month of February, right? Then you have the March for the revision. We will have revision sessions. We will have practice sessions. We will have short sessions as well. Like last time, chromosomal disorders, right? You know that from the disorders part, every year, you like one or two questions from the, yes, 2 questions directly from the genetics, minimum. And 3 to 4 will be there from your human physiology part, right? So we had that type of lectures, so we'll do it. So rather than be like, yaar, if it is a 10-hour ka marathon, we are sitting with you for the 10 hours, then please complete everything. So I can do it also. Just from human reproduction, I can just talk about the gametogenesis, I can just talk about the menstrual cycle. You already know about the structure. Let it be. Let's start. Let's discuss the embryonic development. Chapter over. Come to the reproductive health, that hardly needs 1.5 hours. Chalo, over. Biotechnology applications, that is also a chapter of 2 hours. Done. Okay, that is so we can discuss it in detail. Then human health and disease. Okay, this is immunity. These are the diseases. You have to revise it. Leave. No, that's not necessary. That's not important. I know the students, they trust their teachers a lot, right? Few days back, like always, ma'am, you give this example, but it is a fact. Few days back, I got a call from a student. So she was like, we rely on the one shots for the revision, right? And we follow such type of sessions for the revision, the short sessions, 2 hours, 3 hours wale for the quick revision. We have completed our syllabus. So my point is, if I'm not going to discuss anything important in that one shot or in these marathons, so obviously I'm basically breaching your trust. है ना कि यार क्या करूं? Students, they trust me, and what am I doing? I'm just finishing like this. Shh. Okay, so that is why the today's sessions, in today's session, I was clear that that's how I'm going to proceed. Just in ecology, you have to read the NCERT, but it's simple. Biodiversity, mein you have the topics. Again, we can go very fast. We can finish it. In organism and population, main thing is about the population interaction and regarding that population growth. Not a big topic. Ecosystem is also a very simple topic. You can finish it with the quick revision, with the proper flow charts. Yes, 4 hours are sufficient. 4 hours are sufficient. Human reproduction needs time. Sexual reproduction in flowering plants, again, you can finish within 3 hours if you go for the most important one. So these are the things that we need to take care. Okay, so still you have time. So there will be marathons in the month of March that are going to be very quick, where you will only discuss the most important things. But right now you still have time. Okay, you still have time. Right? So it's better to utilize your time wisely. Again, I'll suggest you, right? If you are confused about your preparation, please go for the most important chapters. Please go for the most important units. Okay? Go for the genetics, human physiology, master it. Right? Master it till February. And then in the month of March, whatever chapters are left, be it diversity, microbes, evolution, whatever it is, go for it. Do it. Okay? Do it. Okay. Okay. So my next marathon, you can just follow the sequence. Azim sir, then Yavar, then me. Okay. Okay. So now for the biotechnology applications, tomorrow only you will get a recorded video, right? I'll put the recorded video because again, if I will exceed this chapter, that there is some duration thing na, that if I exceed the duration, then the lecture will not be there on the YouTube. So right now I'll talk to my team. I'll tell them to put a recorded video of mine, okay, regarding this biotechnology applications. And it is a chapter for 2 hours and maximum 3 hours. Maximum 3 hours, and that to just increase the speed and finish it. Okay, so I'll share the PDF in our Telegram group, and I'm not going to share this PDF because it is having other chapters also. So it will be a very detailed one. It will be a very detailed one. Right? I'll try to, but I don't think that it will be useful. And then, Baki, you know that if you want to enroll yourself for NEET 2026 and 2027, na, you are going to get 60% off. So you can use the coupon code Ambika10 in the description box of this video, right? In the description box of this video, you will get the link, right? You will get the link for it. And now you know that, okay. So your HSP sir session, 8th ko. Then 10th ko Wazim sir, 12th Yavar, 14th me. Like this. Okay? Chalo, so see you all. We'll share the PDF. Do take care of yourself. Do revise it sincerely. Don't just cry. Don't just cry. Complete the chapter. Okay? Just complete the chapter. Okay. Marathon PDF.